distancia de la planeta desde...
Sí, Venus es un planeta muy peculiar porque parece ser que, bueno, sabemos que su superficie es muy cálida y eso es debido a que hay un efecto invernadero muy grande porque, al contrario que Marte, es un planeta que está bastante vivo, tiene mucha actividad y toda esa tensa atmósfera ha hecho que la temperatura allí sea muy alta, fundiéndose. También tiene como parte de la mayor parte de su superficie es nueva porque por estas altas temperaturas se altera y se regenera y se modifica. Entonces es verdad que he leído estudios que dicen que precisamente por ser un planeta activo podría ser más susceptible para que pueda modificarse para unas condiciones interesantes para el ser humano, aunque yo lo veo algo más de ciencia ficción, porque realmente las condiciones en su superficie son demasiado extremas. Sí. Sí, eso es.
Feel the people that who are watching us today, we are more in an exploratory dive. We are trying to get some methane seeps that we are also looking for the day before yesterday in this typical subduction area, the fault zone of Atacama Trench. So we are today in 2.8 kilometer of depth of Pacific Ocean near the coast of Chile. We are near to the city of Antofagasta, to the coast. And there have been some geological reports that in the zone that we are looking into now, there can be some methane seeps. What is methane seeps? That we can see some bubbles coming out of air, and possibly they are methane. But when this kind of bubble comes out, the diversity or the area changes abruptly. We can see some deposition of carbonates, some white precipitations that can be also because of the microorganisms that are living. We can see some crustaceans kind of animals living over there, and also the water parameters change abruptly. We can see some interesting animals lying over here, if we can find these methane seeps, it will be a very nice finding. It will be a novel finding and really to get this kind of methane seep in this zone that have not been reported earlier, but there are predictions. Also with SuBastian has a huge capacity to go into the depth of more than nearly 4.5 kilometers, but the thing is, we cannot get all the prediction of from where the methane gases can come out. This is why we are going into a transect and we are trying to look into the area to see if we can find this kind of features or not. And also this kind of methane outbursts, they are discontinuous, so often the area that we are going through, we cannot look into it because that particular time probably the gases were not bubbling out. So this is the reason that today, entire day, and tomorrow also we will be looking into this area to see some formation of methanes if we can find out, and this can also open a new horizon depending on the ecosystem that we will be finding near those methane seeps.
Naparna, if you don't mind me asking, how you get to working here in Chile?
Okay, yes, that's a long history. I was trying to, when I was finishing my PhD, I was working in a very typical microorganism. I was working on Bacillus anthracis that cause anthrax. It's a causative agent of anthrax. So we found anthrax causing organism from a hot spring. I was that time also working on hydrothermal areas. So we found that particular Bacillus anthracis never had any encounter with any other organisms nearby region. We didn't get any event of horizontal gene transfer in that bacteria, that we were also looking into, that this is a possible biosignature, that the area has been unaltered for a long time. So this was a new report, because in microbiology we have studied that Bacillus anthracis are obligatory pathogens. So our work has been a first report of Bacillus anthracis are not always obligatory pathogen. So the non-pathogenic one came first to the world, and then they had some evolution, and then they become pathogenic. That was a probable hypothesis. So I was trying to look into the people who started working on Bacillus anthracis first, and I got that group from UK. They have been first working on it. So I applied in some position, but I failed. Actually, I failed in a lot of positions. So from when we
it's very difficult to get a position in science. Yeah.
Yes, and also when we talk about success, and it looks very brilliant, it looks everything is glittery, and the scientists are working in deep of ocean. We need to normalize more the failures that we have in life. So a lot of people I wrote, but I could not get selected into the call. So that I was looking into the positions in Twitter, and I found a professor of her work I was following in my PhD. She was working in salt flats and acid mine drainage. So I wrote to her, and then she told me that she was not working that time into Brazil. She changed to Chile, and there is a call open. If I want, I can apply. So I applied, and I till that didn't finished my PhD. So I was like trying to find a position, and I thought, okay, I got this position, and okay, let's go like this, let's explore. I was always excited to exploring new areas. But well, Chile was a huge cultural and geographical change for me. I was thinking it's America and people will speak English, but it was not the fact. So I came and I realized, no, people all speak Spanish. And then I started working as postdoc there, and in a year I got some funding of me, and then my contract was changed to assistant professor. Then I continued working in that university for 5 years. I had 4 fundings secured in that university, and last year I have been lucky to change to another university in the same region, and so there I'm working now as full professor in the university, and also I'm working recently. I've started working as the research coordinator in Universidad Autónoma de Chile, sede Talca. So the horizons open, and I have just always accepted the horizons that came to me with opportunities, because in India we call we talk this not in a religious way, but in a more like a way of culture, that God gives us the opportunities and we should always take it. So that's something that I believe. What comes in forward to you, just accept that, and that's how I'm going. And I came here till the expeditions with you all with the indications of superpower.
Yes, as you said, is necessary is to remark that a science career path is never easy. I mean,
No.
it's very competitive, and also there are not that much number of opportunities depending on your field. Will be more or less, but it's never easy. And I'm guessing even coming from another country, it's even more difficult, and also probably being a woman. So it's a long way until you get into where you are now, right?
Yes. I don't know that how it definitely it's more difficult being a woman. I see the difficulty more probably from Southeastern Asia, where I belong to, because the society still needs a little of metamorphism. But talking about Chile, yes, the cultural difference was huge, and being immigrant and working is never easy. Can be science, can be any other field. It's always difficult, the acceptance. But what I believe is we are like global citizen. In order to survive in a place, we always have to adapt. Imagine the time we came into the ship, and it moves, and the initial days were difficult. All were almost in their room. They were trying to survive, but as the time is going through, we are getting adapted to this movement. So in every place where we are new, we have to try to adapt there, and that's something I learned from the extremophiles, because
we need to be more like them, right?
More like them. Yes.
And also not just from the this is a lesson that is not also need to be learned from the career path, but also for research. That not always research is fruitful, but it's always necessary to do. I mean, you need to take risks and to go
Yes.
beyond what is standard to make great new discoveries. And that's why I think the work we do in exploration, both in space exploration on for example, what the people in the Schmidt Ocean Institute is doing here with all these very nice projects is super important, because we need to push the limits.
And all the time also we don't found, we are not going to find positive data, and the negative results are also results. I just found a couple of days back that there have been a journal on negative data on microbiology. So this is like something I found very interesting, that we normalize not getting data also. Because it's not that always we are going to find something. We can fail also, or we cannot get positive results, but those are equally important. We are going into down now. So now we are in a depth of 2.8 kilometer, 2,840 meter. The time that we started, we are almost 2 kilometers more than this of depth, and we can see that the temperature is almost constant in all this area. We can see it's from 1.77 to 1.82 degrees Celsius with very little change, and the salinity is also almost constant in all this transit that we are going through today. The oxygen saturation level is almost also moving into 36 to 38%, which is a very less oxygen concentration, but the life that are growing over here have those adaptability to act as an aerobe or act as an facultative aerobe or anaerobe. So the life adapt to this kind of concentration of high salt concentration, low oxygen concentration, very low temperature, and in this deep of ocean where it's almost 4,000 times more of the atmospheric pressure. So I think if everything goes well, we will be having a transect from now of 8 more hours, and our goal today, if we can find this methane seeps in this particular area of Atacama Trench. So we will be having a long day of diving today.
It's interesting how one of the most promising areas is at the end of the path, and instead of being at the beginning, and that's because the SuBastian it's designed to go upwards instead of downwards. So that's why doing the transits we need to go from the more deep to the parts that are higher, and that's why we are going through this transit in order to try to find something. So let's cross our fingers.
Yes, tomorrow also we are going to have a similar kind of dive to search this, and in the last day of our expedition we will be going to the deepest point of almost 4.5 kilometer of depth.
Yeah, we will reach the limits of the SuBastian.
Yes, right? Yes, we will be reaching to the limits of SuBastian.
So mostly you're going to press buttons up here and try and look busy. Every now and then you're going to bother yourself. That's what I hear.
The area that we are seeing now, we can see a lot some sea cucumbers lying here. Also in this kind of deep depth of ocean, the light does not enter, so the visible wavelength of solar light we cannot see here. This is what is a reason that we can see a very narrow area only where the light of SuBastian reach, and there is possibilities that probably the organisms are seeing first time any lights in that depth of ocean.
It's similar when you do, for example, on the beach with a metal detector and trying to find something. We are doing this kind of looking, going through a narrow line, trying to find something.
Mm-hmm. That's always about any exploration, that we try to catch an indication, and we just try to go for that. You can see a sea star, the echinoderm group is lying over here.
There's one that's on a big wire that sticks out. Yeah, I can have a look, but I can't imagine what we would have there. What do you need it for a motor? Yeah. Well, maybe our motor temp sensor, but that's going to be a part. It's not going to be a thermistor. It's going to be a thermistor inside.
You know what I would check? McCartney bench. Spares.
Hablábamos antes, Aparna, de cómo has llegado a trabajar en Chile. No sé si lo hemos hecho en español o en inglés.
No recuerdo en ese tiempo.
Yo creo que ha sido en inglés. No sé si para nuestros amigos que estén en línea hispanohablantes quieres hacer un resumen de tu trayecto y también de lo que yo creo que también es una lección importante de cómo no es fácil y que también hay que tener en cuenta las dificultades que tienen trabajar en ciencia, ¿verdad?
Sí, claro. Bueno, yo vengo desde una ciudad que es cerca de Calcuta, se llama Satsol, y no soy de una ciudad bien grande, así que nunca tuve como muy claro la ruta de mi vida. Sabía que, quiero continuar en ciencia, pero la ciencia, ¿cómo puedo llegar de ser un científico? Nunca sabía. Y más que cómo representa a los científicos en ciencia, es más como hombres. Hombres son científicos, científicas para mí no existirá en ese tiempo, aparte de Marie Curie o Pierre Curie. Y estaba terminando mi doctorado, y en mi doctorado estaba trabajando con una especie de bacteria muy interesante, Bacillus anthracis. No sé, probablemente muchas personas saben sobre esta bacteria, que en 2000 en Estados Unidos tuvo un ataque de bioterrorismo de Bacillus anthracis. Se envió cartas con...
El anthrax, ¿no?
Sí, de anthrax, con las cartas con las puras de anthrax. Y eso fue un tiempo que el Bacillus anthracis vino mucho en el luz. En realidad Bacillus anthracis ya existía, pero mucho más en infección en animales tipo vacas, cerdos, este tipo de animales. Pero después de 2000 ya empezó esa idea de bioterrorismo. Y nosotros encontramos este Bacillus anthracis desde una región muy novedosa, desde unas aguas termales, y que encontramos que este Bacillus anthracis no fue un Bacillus anthracis patogénico. En los libros de microbiología nos veamos que está escrito que Bacillus anthracis que es un patógeno obligatoriamente. Nuestro trabajo fue el primer trabajo reportado con la biología molecular, con genómica de Bacillus anthracis, que no fue un Bacillus anthracis patogénico. Y también podemos entender que probable que este Bacillus anthracis, para vivir en un ambiente muy extremo, nunca tuvo un evento de transferencia de material genómica horizontal. Y también podemos confirmar con este trabajo que las bacterias patogénicas no vienen primero se vienen las bacterias no patogénicas, y con las evoluciones, con este tipo de cambio de material genética, se cambia como una bacteria patogénica. Y durante este tiempo estaba buscando con los investigadores de Inglaterra, quien primer reporte de Bacillus anthracis hizo. yo postulé en algún financiamiento, pero no logré. Y en ese tiempo en realidad no logré ganar muchas posiciones de postdoctorado. Claro, en postdoctorado buscar posiciones es difícil porque en cada época es como un leaky pipeline. Entre se sale desde la escuela, después colegio, después pregrado, menos, magíster, un poco menos, en doctorado mucho menos y en postdoctorado mucho menos. Entonces cada parte hay algunas personas quien sale. Y bueno, yo no logré ninguna posición en este tiempo en Europa. También tenía una dificultad que en India normalmente se hace la convocación de dar el certificado final del doctorado después de casi un año. Entonces yo tenía un certificado como tipo profesional.
Claro.
Así que me logré, estaba postulando, estaba buscando posición en Twitter, en redes sociales tipo LinkedIn, Twitter, y ahí encontré una científica y su paper anteriormente yo tenía leído y comuniqué con ella y yo sabía que ella estaba trabajando en Brasil, pero me comunicó que en este tiempo ella estaba en Chile trabajando como profesora. Y me mandó un concurso de una universidad en Chile, en región del Maule, Universidad Católica del Maule. Y ahí empecé a trabajar en 2018 como investigadora postdoctoral. Y en ese tiempo, yo terminé mi doctorado en 29 de junio y llegué en Chile en 7 de agosto, así que casi no tuve tiempo de prepararme. Y yo pensé, bueno, América del Sur, vamos nomás. Y ahí tú vas a lograr todo en inglés, sería fácil, porque yo pensé, bueno, es América, pero América Latina es distinto. Llegando entendí que es muy distinto el mundo. Y claro, en el tiempo que yo llegué en esta particular ciudad, Talca, en ese tiempo en Talca casi no estaban mujeres de India, y más que mujeres solas es una otra cosa, así que me costó demasiado, más que en 2019 entramos en pandemia y no sabíamos, tuvimos un inmenso inseguridad que cuando esta pandemia va a salir, yo creo que no solamente yo, el mundo tenía la inseguridad de sobrevivencia. Y bueno, terminó la pandemia en 2021, casi 2 años y medio, y ahí poco a poco empecé a trabajar con los estudiantes más, con técnicos, y en 2019 gané mi primer financiamiento en Chile que se llama Fondos de Iniciación. Y con este financiamiento tuve un contrato como profesor auxiliar o profesor asistente. 5 años trabajé en esta universidad, después me cambié a otra universidad, la Universidad Autónoma de Chile. Yo pertenezco ahora de Universidad Autónoma, de sede Talca, y ahora trabajo como profesor titular. Bueno, hay cambios en la vida y trabajo como el cargo o la jerarquización más alta, profesor titular, pero el cambio fue, el camino fue difícil, porque nunca tuve un contacto así directo para llegar en algún lado. La búsqueda fue muy difícil y el número de rechazos fue tan alto que el número de suceso o número de éxito fue casi no visible. Y bueno, ahora estoy trabajando como coordinadora de investigación de Universidad Autónoma de Chile, sede Talca. Y en realidad Chile fue un país excelente para mí. Me acepto con brazos abiertos. Yo tuve mi vida como una científica en Chile. En realidad yo me estudié en India, pero mi formación completa como mi carrera científica de trabajo fue en Chile. Y muchos financiamientos tuve en 6 años, en últimos 6 años, 4 financiamientos. Y tuve un proyecto gigante de Antártica y 2 años fui en Antártica, lideré todos estos proyectos, lideré grupo y gracias a Dios fue todo bien. En realidad en India siempre digamos que si hay oportunidad al frente necesitamos tomar esta oportunidad, porque no sabemos qué va a venir en otro momento en la vida o no. Sí.
Gracias por compartir con nosotros tu historia en español, que seguro que algunos de nuestros oyentes agradecerán.
We were talking also about the small mounds, that these mounds can be some kind of megafauna. They are like sea cucumbers or some sea worms. They are hiding this kind of small lumps of sediments that we can often see, and majorly they are for this kind of places so that the megafaunas they can hide. We can see a lot of sea cucumbers in the way. In all these days, we have found that sea cucumbers are really diversely distributed in all the area that we are looking into. Also, the sea animals, the sea worms are something that we are almost getting in all the places.
When you think about it, is you compare with the surface, it's like I mean, here you don't have large plants or large vegetation, but the life is super rich. You won't find in that many organisms in the surface, right? It's like most of the places, of course.
Yes. And almost the foraminiferas are everywhere, so we get a huge diverse.
It's like the grass, right?
It's like the grass. This area, yes. Yes, actually, this area, this coastal area of Chile is known for its high productivity, and that is what we are seeing. Almost looking into the sediments that we are working every day, the upper surface is actually not a soil. It's all the organic precipitation and loose organic material that we are looking into, and that is the reason when SuBastian is stopping and trying to collect a sample, we are seeing all those upwelling of the loose sediments. Also, some flocks that we are looking that are coming through the way of SuBastian. Something is floating around there. That's something called marine snow, and this marine snow are formed due to some aggregates of organic material, some carbohydrates, some decomposing material. So these are very common near the sediment of deep ocean.
I guess the main thing is if they touch, when the metal touches. What do you need to work with? I'm sure that brings up other engineering thing.
I would worry with the
Nothing with it?
So they're like we are changing our turn now, and after me there will be my other colleagues who will be taking the charge. So we will be signing off now. In me, Apurna in Divestream, and Dr. Antonio Molina in sea logging. So we will be seeing you in some other opportunities, maybe tomorrow or day after tomorrow. And have a good day, everyone, from whichever part of the world that you are looking into. And we had a nice time chatting about ocean life and the science that we are doing and our scientific trajectories. Así que chao todos los entes quien estaba observando el live stream y escuchando a nosotros. Yo soy Apurna, estoy hablando desde live stream y con mi colega Dr. Antonio Molina en Sea Logging. Estamos hablando sobre la vida bajo mar y también sobre nuestras trayectorias científicas y también en la área de ciencia que nos trabajamos. Así que hablamos en otra oportunidad, mañana o pasado mañana. Y gracias a todos para escucharnos. Que tengan un muy buen día. Chao a todos.
Thank you, Aparna. See you later.
Are we carrying on past 5 then? What's the one? One's in, or we're just gonna put
No, it's kind of been 3 hours on the deep end.
All right, hello everybody. My name is Jess. I'm an oceanographer from Germany. I studied at Geomar. I worked at Ambari previously, and I've mostly been on the background on this cruise, supporting science. Oh, there's a skate. I've been here mainly to support Dr. Lauren Seiler in cruise planning, especially with ROV operations. I've done all the sampling sheets and helped everybody coordinate the projects. We have, I think, 16 different projects on this cruise. So guaranteeing that everybody gets their samples was a big part of it. So in the room we have Adam, who's piloting. Oh, they just had a shift change. Ben is co-piloting. And Carlos is our multimedia person. Then there's Antonio, who's our geologist in the room. And Drew, who's sea logging. Carlos, could you switch Drew's mic on, please? Carlos? Oh, we had a question in the chat. Is that a ghost squid? I think I missed it. I'm sorry, I just got here. All right, I think.
Am I? Yep, there I am.
Hey, Drew.
Hello, how are you?
Hey, doing well. Did you get some sleep?
I did get plenty of sleep.
Nice. Yeah, Drew was on the early morning shift with me here. This is an incredibly long dive. We're doing a transect. We have a bottom time of about 21 hours. The dive we started descending shortly before 5 a.m. and hit bottom at about 7, and we'll be staying down until 4 a.m.
Yep.
So we're trying to cover a large area to look for methane seeps today.
And then from 6 to 8, after we wrap up here, we're going to have a special graduate student takeover where a couple of the graduate students who have been with us on this expedition are going to take over sea logging and sort of dive leading the trip.
I think that's going to be a party.
Yeah.
Well, I hope some folks will stay up with us.
Yeah, we're going to be up till 4 a.m. I think we'll be taking turns.
Yeah, Lauren and I will be here too. So, in the hope of finding methane seeps. So we've been, yesterday and the days before, we've been mapping at night while people were sleeping. And then the day after, we'll look at the maps, we'll look at the bathymetry, and look for signatures or indications that there could be seepages. We're in a fault zone. We're at a convergent margin, in a subduction zone where the oceanic plate, the Nazca plate, is subducting underneath the South American plate, and it's pushing it up. And Antonio, please jump in there. I'm no geologist, so he can probably explain that much better
You're doing great.
than I can.
So that basically that subduction that's happening, that creates sort of like an opening for the methane to come through.
Yeah, the thing that's going on is like we have different kind of plates in the earth. This is the upper part of the crust on the surface of Earth, and we have this oceanic crust that is always forming in the I don't know the name in English, but in the crust I think they are called, and hiding below the continental plate. So in here we are right in that part where this kind of plate that is going down the continental plate that is older. We are now in the part of the continental plate, and because of all these tectonic stresses is why the methane is forming, because part of the mantle is diverts to the different cracks and tectonic features that are caused because of these stresses.
Antonio, sorry, one sec. We have a fishing vessel that is in the way. We have to go up to the bridge really quickly. Antonio, would you mind coming with me? Because we need a Spanish speaker to.
Oh, sure. I will translate. Sorry, Drew.
Coordinate. We'll be right back. Okay. Sorry, guys.
Emergency.
I don't think there's anything I can do there. So. I don't think it really matters if I'm back here or up there. So.
Jump in.
Sure.
Never pass that box in case it's a big show.
Fair. So while Jess and Antonio take care of the fishing ship, I'll take over on the dive lead for a little bit. So I'm Drew Palmer. I'm a professor at the Florida Institute of Technology. We're about 30 minutes south of Cape Canaveral, so we're the people who get the best rocket launches, view of rocket launches anyway. And I'm one of the astrobiologists and biochemists and just microbiologists on the team, so I wear a lot of hats in this group. My group, my grad student Caitlin Hubrick and I are here in part to look for methane seeps, but also to do some sort of comparative microbiology between the Atacama Trench, where we are now, and the Atacama Desert, where we were a few days ago. And that is one of the highest and driest and most Mars-like deserts on the planet, and we are hoping that we can use that comparison to help us understand some of the extreme conditions in which life persists as a way of helping us look for life on other planets, like Mars. I just like saying Mars. This is your computer. We're good.
Where are they?
I don't know, but they can communicate.
And we're back to our original positions.
Whew, thanks for standing by. We are waiting. So we needed a Spanish speaker because I think the fishing vessel is somewhere along the track where we wanted to go. So we'll have to communicate with them what their intentions are. We might have to go around them. We don't really know yet. What is that?
Yeah.
Could we log that?
What do I log that as?
Wireless? Oh.
Oh,
Adam said that's a wire rope.
okay. So anthropogenic. I've seen that a couple of times during the day, and that makes sense. That the circular pattern is, I guess, how it descends. And
Yeah, we were about to update our waypoints because we're actually moving along much faster than we thought we would, so we can cover a bigger area. We see more crinoids. Actually, we have a request from the science team. If we find another crinoid field, they would like to take a couple samples. So, and since we have time, we can do that.
There's a question in the chat about stream schedules and locations and goals are listed. So you can find the detailed descriptions of each expedition on the Schmidt Oceanographic Institute website. The stream schedules are when the ROV is at bottom, which I think pretty much varies from expedition to expedition and is kind of subject to change. So, for instance, we made this decision to do this 24-hour dive sort of on the fly. Not on the fly, but with a day or two of planning. So it wasn't what we'd originally planned to do, but then based on the previous day's dives, it was a decision.
Yeah. So the way this worked is we looked at the map yesterday that was acquired by the ship the night before. Whoops. Ouch, that was the chair.
Yeah, I have it before.
So yesterday we looked at the map and we have a couple of waypoints that we would like to go to, and depending on the structure of the sea bottom, we identified some geological features that we wanted to check out, and then in discussions with the tech team, which is amazing by the way, they assembled all the maps for us, cleaned them up, and helped us walk through a couple of different plans, and then together we came up with the dive plan for tonight that helped us cover the largest region possible. And so the logistics behind it always depend on what the other team needs. Will there be a CTD cast in the area? When does the ROV have to come up? When do we have to be in another spot? It depends on a really large amount of factors. We also have a permit that we have to comply with. So there are certain areas where we can't go, so we have to plan around it and come up with an ideal path for both Sebastian and the ship. But the crew is always helping us there. So, and they've been incredibly supportive.
I think one of the more interesting things about this is that really the trip is sort of real-time data driven, right?
Yeah.
So we're constantly getting data from mapping and making decisions in real time and adjusting our models and our hypothesis of best places to look, which is really incredible to have the technology to be able to do that in real time.
Yeah. So we had a rough plan before that. We had a dive plan, but then coming here and the data. So there was a question in the chat as well: what kind of sensors or instruments do you use to help narrow down an area that may potentially have a methane vent? We use all of the ship's instruments that are available. We used sub-bottom profiling to look at the sediment depositions, and sometimes if it's during shallower dives, if you get a really good resolution, you can see a certain fuzziness in the data, in the profile, and that can hint towards seeps, or you can see large sediment depositions. It tells you where rock formations are, so what the bottom looks like. So that is one part of it. Then we also look at what is called the backscatter, that is the acoustic returns, so we can see changes in reflection. And seeps, for example, if they are venting, which we don't know that these would be, they would have a very high reflectance because it reflects off of the bubbles. The other thing that we extensively looked at is the bathymetry, the map. So we use the ship's multi-beam to acquire data after the ROV dives. So in between dive sites, the texts help us to come up with a mapping schedule or with a mapping area, and that's ideally, and all this data together informs where we dive, how we plan the next dives. So it's very it's very data driven because this is exploratory. We don't know where methane seeps are. We know that they were found north and south from here, and because it's all the same continental margin, there's a chance we'll find them here as well. And so we talked to a lot of geologists. We talked to collaborators that have looked at methane seeps in Chile, and they have helped us come up with a dive plan. Yeah, so we're looking for. We also right now, so SuBastian has a sonar as well that we're looking at. So it would show us if there were any bubbles, but we're also really relying on the visual. So we are looking for microbial mats, certain types of tubeworms, and clam fields. So usually methane seeps are bursting with life. And I think there's another lizard fish. Yeah.
Yeah, I've been
Camera shy.
logging some crinoids. There was a little field there of crinoids.
Yeah.
That particular species we've been seeing all along our dives along this portion of the trench. That's I think that's the predominant crinoid species here.
Yeah, and crinoids can also be considered living fossils. They're very ancient beings, and you can also see them in the fossil record. So you can use different species to date them as well. They're very pretty. They're like sea flowers. Oh, there's the lizardfish.
I think that's the same one I logged. I think he just decided to chill out for a while.
There's this green worm, which name I forgot.
Is that the acorn fish? No, those are kind of purpley. Or acorn, sorry.
Oh, I think that's another one.
Yeah.
So we haven't heard any updates yet regarding the fishing vessel. We'll see
We're
if
gonna
we
hope
have
for
to.
the best.
Yeah, we'll see if we have to change our plan slightly. We'll find out if they're actively fishing or
Yeah.
what's going on.
There's
there.
an anemone.
Oh yeah, a couple.
This is another really sediment-rich area. So some of the spots have been a little more barren, but this is very, very sediment-heavy on these rocks. I logged them.
Oh, thank you.
That little ding sound you heard was me forgetting to turn off my email alert. It's quiet now.
I didn't even hear it.
It was.
Oh, so we have a question. No sample collection on this dive. We were just discussing it. There is a request from some people in the team to sample some of the crinoids, but the primary objective of this dive is to cover a large area. So we will ideally hold off with sample selection until we find methane seeps. So we have a lot of space to take all the push cores that we want there, because a lot of people on this cruise on our team are interested in finding seeps, eventually hydrates and associated rocks. Yes, that's right. We're looking for methane seeps. But if there's something that a group member is very interested in, then we'll take a couple of samples. But yeah, in general, we want to cover a large area.
Well, I think we've found no shortage of crinoids today. Actually, there's
Yeah.
quite a bit of them.
Yeah. Very true.
Again, that's a really nice field of crinoids. They're very plant-like in a way.
Oh,
Whoa,
there's a crinoid field.
that's very cool.
That's beautiful. Can you please log that?
Yeah, absolutely. I normally try to count. I'm just gonna go lots of crinoids. I would describe it as a field of crinoids.
Yeah.
That's spectacular.
Where's SpongeBob? We're just talking to Adam, who said they've got the ship and the ROV in SuBastian dialed perfectly, so they're moving along at the same speed. Because we're thinking about taking samples here, but if we'd stop, we'd have to play around with that again. So we'll, for now, just keep going, and there'll probably more crinoid fields later.
One
Because we
there.
might have to wait for the fishing vessel. That's what he said. So that might be a good time for sampling.
There's also almost 12 hours left in the dive.
That's
So-
very true. Yeah. Yeah. It's a long dive. It's 21 hour bottom time.
No.
So please stay on later tonight. It'll be a party in here. Drew's gonna bring glow sticks. He
Yes,
promised.
I did.
I'll count on those.
Uh-oh.
Oh, we'll probably find something that glows. There's a lot of bioluminescence in the deep sea, by the way, but we can't detect it with our cameras. We would need a special low light camera. But there's I think it's a BBC documentary on life that glows.
Mm-hmm.
They built Ben, my husband, built the low light camera for that one. Oh, there's 3 crinoids on one rock.
So there's a question in the chat about what we learn about methane seeps when we find them. So actually, one of the things we're actually interested in more, rather than learning about the methane seeps themselves, is about the biology surrounding the methane seeps. So here in the deep ocean, where there's no light, driving biological processes and productivity, you're always trying to understand how life survives down here. And these methane seeps are sort of oases where there's a lot of activity and productivity going on at multiple scales, microbiological, so the bacteria as well as the macroorganisms. And so usually you find lots of sort of hotspots of biodiversity here. There could be microbial mats. There should be a lot of large organisms. Jess was telling me that a lot of times they're marked by fields of just tons of clams. So we'd expect a lot of the crinoids and cucumbers and sea cucumbers that we've seen in bigger diversity. And so this is a hotspot area for looking for the biodiversity of the ocean, but also for understanding how microorganisms might survive in these environments. And so some of these methane seeps can provide us with evidence for helping us understand rules for how life might survive in other extreme environments. Sorry, I was answering the question in the chat, Jess.
Oh, I didn't see it.
Oh,
What,
it was
what
okay.
is the question?
It was about the methane seeps and what we had learned, so I was kind of giving the
Oh,
lowdown
okay.
on the seeps.
Sorry, I was chatting with Adam about the fishing boat. He saw that there's a lot of evidence for fishing in the area. Oh no, crinoids!
Well, logged.
Thank you. Yeah, so the rope that we saw that might have been fishing related. There was a little bit of trash too, so we'll see what we find. They're all facing the same direction.
Wow, look at that.
I assume it's the direction of the current, so they're filtering.
Hmm.
It's pretty. Yeah, and crinoids are a very ancient group of organisms. They're also considered living fossils, and they first appeared about 300 million years before the dinosaurs. So they've seen some things.
Wow, this field goes on for a bit.
Yeah. Yeah, we're moving along on the ridge, so we've been climbing for quite a while.
So are the crinoids modal? Do they like unroot themselves and move, or do they?
No, they grow on a piece of solid substrate.
Mhm.
I'm not what I was just thinking about. I'm not sure if they can move their head kind of like a sunflower.
Like a sunflower? Yeah,
can-
I was just thinking
yeah, I
about
was
the
just
analogy.
because that would make a lot of sense, and I know that some corals can do that, but they're I can look that up. It's a very interesting question. Oh, there's a question. What are we looking to learn about the methane seeps when we find them?
Oh, that was the one that I answered.
Oh, that's
Yeah.
oh, okay. Sorry.
No worries.
Yeah, and depending on who you're asking the team, there's a lot of different answers to that question.
I tried to give the overall what everyone was interested in.
So part of the
A lot
reason
sea cucumbers.
oh yeah, a lot of different colored sea cucumbers. Did you see the fancy one that looks like a little punk? A little
Yeah.
punk sea cucumber
Yeah.
with the fancy hair? I mean, it's not hair, but yeah. Part of the reason why we're here is Amando's group found microbes in the Atacama Desert, which is the driest and the oldest desert on Earth. And those are really ancient microbes related to land plants. They're one of the oldest relatives of land plants. Oh, there's a little shrimp. And genetic evidence showed that they were related to some microbes that come from the ocean. So part of the project will be to compare those with what we find here, which could give us hints towards origins of life or land plants in the ocean. So part of the Living Fossil project is about living fossil microbes. And they've actually found in some regions of the Pacific, I think it's below the gyres in the sediments, they found microbes that have been dormant for millions of years, and they can revive them. And they will just start growing again. pretty amazing. So the gyres are like, a lot of the ocean is pretty much like the Atacama Desert. There's not much life there, but there's always microbes. But if those microbes don't find enough food to survive, some of them can go in a dormant state and survive until they find their food source, and they can grow happily ever after.
Oh, wow.
Oh, is that a worm? Is that a? Oh, that's a sea.
I don't see them swimming very often, though.
Yeah. Yeah, sometimes they don't like to be filmed, and then they take off.
Very nice. In answer to one of the questions in chat about acorn worms, yes, we've seen quite a lot of acorn worms down here actually over the last couple of days. They're pretty prevalent.
Oh yes, and some carnets can move, but these are the stocky types. They cannot move. They have a hard substrate. The unstalked ones are called feather stars. You can sometimes see them. But I still don't know if the stalked ones can move their arms. They probably can, I'd assume, but I'm more of a microbiology person. My marine biology classes have been a while ago.
So you're a marine microbiologist, though,
Yes,
right?
yes. But in the past, I've also worked with cuttlefish and cephalopods. But I think at that time, I was considered more to be a neurobiologist, or that was actually during my undergraduate studies. So I was studying biology and then later biological oceanography, with a focus on microbial ecology, and this is where I've done most of my work in since, or after my undergraduate studies. And we went to methane seeps in the Monterey Bay. Those are false seeps that are produced by organic matter deposition, and those are sometimes very, very short-lived. And so the 1 year we went there, that must have been 3 years ago. They were active. They were bubbling. We saw lots of live clams, and we did incubation experiments on, I think it was on 1,000 meters roundabout. That was right by the canyon. And a year later, we wanted to recover those cores, and the whole clam field was dead. The methane had stopped seeping from that area, and it's probably moved because we found other areas where there were still live clams. But it was incredible to see how fast such an environment can change, how rapidly.
Yeah, one of the things I know that we found over the last couple of days has been some, I think the term we used was extinct methane seeps, where, that was intriguing because we wanted to look at what's essentially the microbial population that remains after that, and then some of the macroorganisms there as well. But today we're looking for the live seeps.
Yeah. And we're also looking for the live seeps, but having found evidence for ancient methane seeps already is super exciting, because that means we have been looking in the right area, but we're still waiting for the final confirmation from the data. What is that? Was that a little jelly? So I think they will look at the carbon, the isotopic carbon composition to see where the, so we found what is likely carbonates. They will look at the composition, and then they'll find out if those were actually methane seeps, or where they came from, whether it was from organic matter deposition, a benthic siphonophore. Yeah, but today we're looking for active methane seeps. So we're mostly looking for visual indications of methane seeps, for example, microbial mats, and we can see microbial maps usually by discolorations of the seafloor. So sometimes they appear white, and we looked at pictures from methane seeps that are south from here, near Concepcion in Chile, and they're whitish, almost blue along fault lines. And so what we did was we compared maps, and together with collaborators, we identified regions of interest, and this is why we picked this dive site today. And we're going to cover a really large area within a 21-hour bottom time. So our dive will be until 4 a.m. and hopefully lots of people will stay up with us.
Just a reminder, so Jess and I are going to be on until 6 Eastern Standard Time, and then we're going to have a couple of the graduate students who have been working so diligently on the samples we do bring up. They're going to take over and do the data logging and dive lead for a couple of hours.
Oh, is that a Dumbo? Yeah, down there. There was a Dumbo octopus, I think.
Oh.
Yay! Oh, it's a tiny one. We'll get that in focus. Aww. Aww, a little Dumbo.
Nice.
Oh, it's very cute.
When we found earlier, Jess.
Yep.
Maybe.
Yeah. Yeah, this one is much smaller than the one we found earlier. Ben just reminded me. That was the one we found earlier was, I don't know, maybe 5 times the size of this one.
Wow.
It's really hard to tell because distances and sizes.
Yeah, scale
Are-
can be challenging.
yeah,
in these
scale.
pictures.
Yeah. Yeah, going back to the students, huge shout out to them. They've done amazing with all the sample processing. They've been collecting all the water samples, filtering all day long, taking the core samples, organizing themselves. It's been amazing. Thank you guys so much.
That's a lot of cucumbers.
Yeah. Remind me, Drew. What are you interested in the methane seeps? What are you looking
Sure.
for?
So we are predominantly more interested in the astrobiology component. So these are methane seeps are the interesting spots of biodiversity. Methane-related microbes were probably prevalent at one point on the surface of Mars in an earlier time point. Part of the geological history of that planet involves methanogenic microbes, and so that's one of the things we're looking for is signatures for those organisms that live in these kind of environments that then we could extrapolate for helping us detect the presence or former presence, so the after effects, the shadows of microbes that lived on Mars in the past. But we're also just generally interested in these hotspots of biological diversity in rather challenging environments, because that can create really interesting metabolism, chemical processes that we can mine for a variety of potential benefits, right? So there may be new antimicrobials down here, new antifungal compounds, different types of treatments that could be mined out of this environment.
That's really interesting. Yeah, fingers crossed that we find them tonight.
Yeah.
And if not tonight, maybe tomorrow, because tomorrow we have a similar dive with less bottom time though, in a slightly different spot. I think it's 20 kilometers from here. It's closer to shore and a little bit shallower. We're kind of moving slowly over there, but we won't reach this point on this dive. But there's another chance of finding methane seeps if we cannot find them today. But we might have found ancient methane seeps that used to be methane seeps at some point, but are not currently active anymore. But our geologists can hopefully tell us more about that once they analyze the data.
There's a question in the chat about Martian microbes and methane. There's some really good work. There's a researcher at NASA, Andy Sugar, SCHUG. Nope. Andy Sugar. I don't have his spelling, but I'm gonna get it wrong. I get it wrong every time, and I'm not sure. Some people call him Andy Sugar, and some people call him Andy Sugar. So I know it's not SUGAR, but Andy's done a lot of really good work looking at Earth extremophiles surviving in Martian environments. So that's some of his work. So, and he's done a series of papers looking at methane microbes from Earth and using those as proxies to try and understand maybe the past geological history of Mars.
Do you know where his research was based on, or is it theoretical?
He does theory, but he also does applied. He's he's actually originally a plant pathologist, but he's a microbiologist who works up at Kennedy Space Center. He did this really fantastic study of basically an autopsy of a failed plant growth project on the International Space Station. So they had a problem with the veggie system where they used to grow the lettuce up on the space station. Literally, that's the acronym veggie. And when there were some fans that broke down, there was a fungal contamination, and he really just laid out this really nice paper, but kind of dissecting what went wrong and how, kind of through the process, through the mechanical components and the engineering fails, like what went wrong there, and then how that led to the biology. And it was interesting because it helped us understand a little bit about life in microgravity, which is something that my group thinks about a little bit. of course, astronauts train in deep water as part of their process because it kind of helps them simulate long periods of weightlessness.
Wow. How deep do they? How deep do they?
It's a big pool.
Oh, okay.
So yeah, they're at I want to say it's at Johnson. They've got like an enormous pool, and basically they submerge like different rigs that they have to work on, and make them work on it like basically in a spacesuit underwater to
Cool.
simulate like the difficulty in movement and some of that stuff.
Does it simulate it pretty well? What's the feedback from the astronauts?
I have no idea.
Have you tried?
No, definitely not. I've seen them do it, like not live. I've seen that on video and stuff. It's I think it's unpleasant.
Oh yeah. I bet.
Yeah.
Yeah, I've done some science diving back in Germany. It's very cold. I hope their pool was at least heated.
Oh yeah, it's a heated pool.
Oh,
But,
okay.
well, probably not, because I think if you're not out in space, I imagine
Yeah.
those spacesuits get hot very, very quickly.
Oh yeah, that's very true.
It's it's very interesting, the relationship between microbes in these environments, in the dark, or like in the desert, in the Atacama Desert where it's really, really dry. These environments are actually really great proxies for thinking about life on Mars and ocean worlds.
Oh, octopus. Is that a pearl octopus?
A Dumbo, yeah, that's an octopus.
But is that a pearl octopus? I don't think that's a Dumbo.
No, that's, definitely a different species.
Yeah.
Look at its eye!
Oh,
Wow.
oh, it looks like a little alien.
Hi. Oh my god,
Oh,
it's an alien!
it's an alien octopus. Oi, cute.
Can we get rid of the...
Oh, and is that a
Getting
brachiopod right next to it?
It's one of those snails. Hi, friend.
Oh, oh no.
Oh wow.
Bye bye. Can we zoom in on? Okay.
Stayed long enough for me to get it on camera.
Yeah. Oh, we have a question. After taking ocean floor samples, do we have to maintain pressure levels in the lab? No, we don't. What we do is we freeze the samples. Ideally, what we did in the past is and it depends who you ask. So for microbiology, we usually, in the past, we snap froze them. Unfortunately, this time we didn't have liquid nitrogen on board. So what we used was some chemical compounds to preserve the DNA that is in there. And then we just mix it and freeze it. And then it stays intact for quite a long time. And then we can take it back in the lab, extract it with some different chemical processes. And there are lots of steps after that. So taking samples is one thing, but then it is month of work to actually extract everything, send it out. Then there'll be an organization that usually does the sequencing, unless you have your own machines for that in the lab, which a lot of people don't. So we send it out, and then we get the data back, and then the fun part, the analysis starts.
Yeah, from our water column and our core data, we'll be doing microbiological and chemical analysis. And the chemical analysis could take a really, really long time.
Yeah. How long does your analysis usually take? Drew?
Well, so we've never analyzed samples like this before, so that's going to be, in a way, we're going to be doing taking little bits to start with and kind of getting down the samples. So we've done environmental sampling before, so we're not, completely foreign. The water sample analysis will actually not be that hard. What we're doing is, rather than bringing too many water samples home, we can take about 1 liter of seawater that we collect and filter it through these old cartridges we have, and those will trap most of the kind of biomarker molecules that we're looking for, what we call the organics. Then we can take the cartridge home with us, and the cartridge is like, I don't know. I'm of course realizing that holding up my fingers is not a good way to. So it's like 4 inches, 5 inches, and those.
That's like 15 centimeters. Oh, I think there's some trash on the left-hand side. Did you want to lock that?
Yeah, and so then we can take that back with us. So instead of taking like 50 liters of seawater, we're going to have like 30 cartridges, and then we can just drain those off with some alcohols and start running them for some testing with some high resolution instrumentation we have.
Okay. And is that all going to be Katie's job? Do you have more students in your lab?
So right now, I think Katie's going to be sort of spearheading the discovery process, and then we'll see like how much we have to work with, right? And then she might get a spin out a cohort of students on the project. This is really why, for us, finding a methane seep to compare all this to would also be really exciting, right?
Yeah.
So.
Will you
I
also
think-
be?
Oh, go ahead. Go ahead.
No.
All right. So I was going to say what we've already found, right, even in the absence of an active seep, there's a lot of interesting things for us to look at that I think is going to keep us, intellectually engaged, so to speak.
Yeah, that's.
Yeah. So.
Would you be comparing it to the ancient methane seeps if we get the confirmation? I think that was a siphonable.
100%. Yeah, that's absolutely kind of what I was thinking about last night.
Yeah.
Was that's really what would be the goal would be to compare places where we cored at the very beginning where we really had no evidence of seeps, a place like yesterday where we thought we saw some ancient ones, and then following up with an active seep. I think that gives a great range. We also collected some interesting profiles along the water column. So through the oxygen minimal zone, which is sort of a high productivity region, and down here to the floor. So we can make some comparisons about the microbial populations and the metabolism that's going on in those environments, which again, that in a way actually contributes to how we think about biosignatures. How do you characterize the footprint of biology in really, really rich environments? It's very easy to do. And then as you get to more and more sparse environments, that becomes a more challenging goal, a more challenging issue,
Did you, you guys went to the Atacama Desert, right?
Yes.
Before we went on this cruise, did you take any samples there, and will you be comparing those as well?
We did, and we will.
Oh.
Yeah. So we got a couple of interesting rock samples from a couple of different sites in the Atacama, and we even found a couple of rare, hard to find water samples. There is water in the Atacama. It's just in very specific places. And then one of the sites that we went to that was really fascinating was pretty high up, and there it does not rain, but it looks like, we were talking to Armando, every morning it's like clockwork. There's a fog comes in, and then it lasts for a couple of hours, and then gone, and the sun beats down, and like the temperature goes up like 30 degrees Fahrenheit, 30 degrees Fahrenheit it goes up.
Wow.
It gets, and then the fog's just gone. But they think that during that time, we found green on the ground, right? That was not copper, and so it's a pretty good indicator that that moisture in the air is enough to sort of give these organisms the a sufficient amount of water, then they get the light, they can do some photosynthesis, then they
Yeah.
get into the shade, right? And so they have a very short period of time based on how some of them are on the slopes where they would get both moisture and sunlight, and then they'd be out of the radiation. So UV in the Atacama is also really high, which is also problematic. And there's some models on from a sneak in Mars. There are models on Mars for different surfaces that might go through like these kind of freeze thaw periods where ice is most of the time you're not going to have liquid water on the surface. You could get brief periods where sun shines on a particular spot on a crater that would allow that particular amount of water to melt. That could allow the biology to run for a little bit, and then the sun goes by and that stuff gets cold. You could have these freeze thaw cycles, which has actually been proposed in Origins of Life chemistry. Sorry, I got going.
Oh, yeah, no, I'm processing. So the freeze thaw cycle in microbes, I missed that part. Can you?
Yeah. So the idea would be that one way that the microbes could persist, right, is that they
Yeah.
basically go into a
Desiccation?
in a frozen state, right? It may not be completely dried, although liquid water on the surface, the pressure on Mars is so low that at room temperature pressure, liquid water would not last on the surface of Mars. It'd literally go into the gas phase, like just evaporate right off. But the temperature is much lower on the surface of Mars, so it's possible that there could be standing water very briefly under the right conditions, and so it could be that water briefly goes into a liquid form and allows those microbes to kind of do biology for a little while, and then they freeze over again, and then it thaws again, and freezes and thaws, and they could kind of survive through these kind of cycles like that.
Interesting.
Yeah.
Would they also use glycol in their cell plasm, in their cell fluids?
Oh, so
For
like as
antifreeze?
Yeah, as an antifreeze. So that's possible. They're also finding nowadays, and this is actually something that Armando and I have been talking about for the bacteria that live in the Atacama, is there's a growing body of research that shows that a lot of microorganisms that live in these kind of unusual water conditions where water can suddenly be gone or unavailable, they maintain these things called coacervates, which are like almost like oil droplets inside of the cell that's made of amino acids or other materials, and that allows them to make almost like an antifreeze. And they're interesting because they become their structural properties are really interesting and kind of the reverse of what we normally see. They've been found as these kind of proteins have been found in tardigrades actually as possible protectants. They think it's one of the things that allows tardigrades to survive.
I love tardigrades. Little gummy bears.
Yeah. Everybody loves tardigrades.
Yeah, and those microorganisms that they found in the Atacama Desert, they also had, because we had a question regarding viruses, they found evidence for viruses in those, and those had a marine origin. So yes, we will be looking for viruses. There is a person on the science team who unfortunately couldn't make it, but we're taking samples for her. And she will be looking for giant viruses in the water to compare those to the ones they found in the Atacama Desert. And it is true, we're not very far offshore. We are currently about 43, 45 kilometers. That is in nautical miles. Around about 24. We're in a subduction zone looking for active methane seeps. I don't actually know the answer to this question. Is there a public catalog of the usual suspects of fauna we're coming across?
My assumption would be know because this is an unexplored area, really. So
Yeah.
and we're cataloging it for the first time. There's a few of the marine biologists on our group who are we've been taking samples the last couple of days, and then there's going to be 50, 60 hours worth of video to peruse for identification. So I think with that, maybe they'll be able to kind of establish a diversity of at least this portion of the trench.
Yeah. There might be a Schmidt catalog. I think maybe we can ask Hannah that question because she's in the room. Hannah, is there a catalog for common species from Schmidt?
Not that I'm aware of. I think it depends on where you are in the world. Because while these are common deep sea species,
Yeah.
we don't know if they're different from other common deep sea species elsewhere.
So Hannah was just saying that some of those species are very common deep sea species that we find in other areas of the world. So there will be a catalog for those. There's none specific, or at least from what we could find, there's none specific for this region yet. But that's part of the reason why we're here. This particular area of the coast of Chile hasn't been explored very much. So in the last couple of days, during the night, we've been mapping the area and then deciding based on the map where we wanna do our dives.
I would add that SOI, which does have some catalogs.
The where?
So Schmidt Ocean Institute doesn't. The Center for Ecology and Sustainable Management of Oceanic Islands, or SOI here in Chile, does have catalogs. But they're a little more specific to the Salas y Gomez Ridge.
Oh, it's more specific to the ridge?
Yeah, the Salas y Gomez Ridge. And
Oh.
the Juan Fernandez Islands.
Oh,
And
so
the
that
sea
is
mounts,
the
they're a
sea
little
mount.
more-
That's on the Pacific side, so that's on the Nazca Plate.
Yes.
I think so.
We're not on the Nazca.
No, we're on the South American plate still.
Yes.
The Nazca plate's on the other side of the
Yeah,
trench.
so we're on the continental plate. The Nazca plate is subducting under the continental plate and kind of pushing it up. And this is why we're looking for methane seeps kind of on this side, because they're fault lines where methane can escape. And that's what we're kind of looking for. But there have been previous cruises in here in Chile, a little further offshore, that have looked at sea mounds on the Salas and Gomez Ridge, and they cataloged species. They found an incredible wealth of life around those sea mounds.
So I saw in the chat a comment about this being like the surface, the bottom of the ocean floor on Titan. So the only difference is that in order to get to the ocean on Titan, you probably have to go through, I think, about 50 to 60 miles of ice.
Wow.
Then you'll get to the ocean.
Jess, my dad was watching the live stream earlier when I was dive watching. You were dive logging and we were
Oh, yeah.
chatting. He said that you sound really nice.
Oh, thank you.
And he wanted me to tell you that listening to the two of us talking together made
Oh.
it a very fun and interesting ride.
Oh, thank you. I love to hear that. Well, I hope you're listening later too. We have the late night shift, so
Oh, yeah.
please stay on.
Yeah, it's gonna
Thank
get wild and crazy in here after
With glow
some little
sticks.
sleep.
With Drew's glow sticks.
Oh yeah. We're gonna have a mission control rave. It's gonna be great.
Yeah, and then we'll find methane seeps, and then the party is going even crazier.
Oh yeah. No sleep till methane seeps.
Yeah.
It's not European. It's not American for sure.
Right?
And, well, yeah, it's
because I forgot my dog's paperwork. Yeah.
Where'd you go?
The falls, Niagara Falls.
Excuse me.
So Ben, co-pilot right now. So sorry, they had a shift change. We didn't mention that. He just made me aware. I have a funny accent. I almost sound Canadian. I'm German, but I've only once been to Canada, and I crossed the border, and I forgot my little dog's paperwork. So you, in order to come back to the U.S., you need to show like the vaccine record, and I had remembered the day before, but the day we crossed the border, I forgot. So I crossed the border, and the officer there told me, or asked me, do you have your dog's records? And I said, no. He said, well, you can't go back. So kind of panicked a little bit. But they sent me, a very kind friend sent me a photo of his vaccine records, and then we walked. After a couple hours, we walked back over, so we went to the Niagara Falls. I spent a couple weeks in upper New York State. So one day we did this excursion, and then on the way back, the officer on the American side didn't even ask for the record, so I panicked for no reason.
I love when that happens.
Yeah. They're like, yeah, come through. Come on in. Which is usually not the case, by the way. I've been interrogated many, many times at the border. I don't know what it is. But we currently live on a sailboat in Mexico, so we're traveling quite a lot for work. And so I think it's all the stamps and the passport. No sleeps till seeps. Yes, yes, I like that. Just more coffee.
Nice anemone.
Yeah, pretty. Did we have a chance to sample any of those crinoids, or are we waiting until later?
We wanted to wait until we hear back from the fishing boat if we maybe have to stop or do something or diverge, or if we have to spend some time somewhere.
Oh, okay.
I know we crossed this one beautiful crinoid field. We were hoping in the next 12 hours there's probably another one.
Yeah, we'll probably see more.
So when we figured, have you heard back from the fishing vessel? Can we still proceed as planned?
I did not know we were waiting to hear from the fishing vessel.
Oh,
I
okay.
just stepped in, so.
They left.
Oh,
They
okay.
left? Oh, okay.
Oh, that's nice. That's very kind.
Ah.
So yeah, we will update all of our waypoints. We're making really good speed. We're much faster than we anticipated. So we will cover a larger area and check out some of those ridges and fault lines in the hope of finding methane seeps. We're also looking at the SuBastian sonar. So if there are any bubbles, which we don't know that there are, they would show up on the sonar as really high reflection, really high returns. But we're primarily going with visual indications. So we're looking for microbial mats, so discolorations on the seafloor, or specific animals such as clams. So sometimes you can see those clam beds, or tubeworms. We might have found ancient methane seeps a couple days ago. I think it was 2 days ago. We were waiting for the geologists to for their analysis to see if those were actually methane seeps. Some of those rocks look like carbonates, but they haven't confirmed that yet. Oh, so we sailed, there's a question, where about in Mexico? We sailed in the Sea of Cortez. We left last year from California and sailed down the coast, and now we're spending some time in the Sea of Cortez, which is very beautiful but also very hot. Yeah, we spent some time on Isla Isabel. That's a marine protected area where blue-footed boobies, brown boobies, and frigate birds are nesting. And they don't even mind you're there. They don't really care. You can take really great pictures. Ben flew his drone there, and a bird got really upset with the drones. So they're more upset about drones than about people visiting the island. Unfortunately, they don't allow dogs though, and there's a couple of fishermen living there, and they make sure that you don't bring your dog on the beach. So we didn't stay for too long.
There's a question about how do you tell the difference between an ancient seep and an active one?
Great question. So an ancient seep, you can still find the carbonates and the carbonate depositions. Those were the shelf-type structures those might have been the shelf structures we saw. But none of the species, none of the microbial mats, nothing that hints on this oasis of life that methane seeps usually are. So methane is used as a food source for lots of different species, and those would be the ones we would expect to see there, like microbial mats, riftworms.
Yeah, methane seeps are not permanent.
Yeah.
They're actually pretty transient, so
Yeah.
they can run out of methane, and then the species run out of energy and food, and so
Yeah.
they just kind of leave a fossil behind.
Yeah, I was saying earlier we went to a methane seep in that must have been 2020. And it was active. There was a clam field. We took samples and we left them there for incubation. Initially we thought they're going to be there for 6 months, I think, and so we had another cruise planned, but that got delayed because of COVID, and then we went back 18 months later in 2021 sometime. Recovered those cores, and by the time we recovered them, the methane seep was dead, was not seeping anymore. All the clams had died off. So we think it probably moved somewhere else.
So my question is, when we say they're transient, are they typically transient on like more geological timescale, or like more of a relevant biological timescale? I mean, relevant for like what I do.
Both. So it depends what kind of methane seep you're looking at. So the ones that we were sampling in Monterey Bay, those were some call them fall seeps. They're there because of organic matter deposition. So it's a lot of organic material that is being piled up, and bacterial degradation and decomposition produces methane that other species will use. And the ones that we're looking for here, well, we'd be happy with either one, but there's tectonic ones, and a lot of those will change on geological timescales. But it always depends. So different regions have
So then, is it? Sorry, I think
Yeah, no.
intrigued. So is it possible then that a previously active methane seep can produce sufficient productivity that after it runs dry, essentially could create a fake seep by virtue of the organic matter it leaves behind. So a real seep leads to a fake seep.
Interesting question, and the answer is I don't know.
Maybe, I wonder if maybe Antonio knows.
Maybe Antonio knows. Yeah.
So my question was, if you have a real seep that's driving biological activity, when the real seep goes away, could that essentially drive a fake seep because the organic material, all the biological matter that's left behind, then dies, and essentially the same process occurs that creates a fake seep. So a real seep becomes a fake seep, then becomes an extinct seep.
Maybe. I'm actually not sure. It's a great
definite
question.
maybe.
Yeah.
Yeah, I'm definitely not an expert on methane seeps. I just did a little bit of microbial studies on them in the past. But this methane seep hunting for me is new too. We learned a lot in the last couple of months in preparation for this cruise. We were looking at data from seeps north from here and seeps south from here and tried to compare that. This is how we picked the dive sites.
I mean, obviously, this might also be a complicated question to answer because with seeps not being everywhere in the ocean, it being hard to find, then you have to find the perfect storm of conditions to be able to observe this.
Yeah. Very true. It also depends on how much methane is being released, I would guess.
Yeah. That's true.
Yeah. But here, a lot of this organic matter is produced by marine snow coming from the upper layers of the water. There's a huge seasonal upwelling that is also driven by the enso cycle, the El Nino and La Nina. So sometimes the upwelling is very strong, so nutrient-rich bottom water or deeper water layers is brought to the surface, and everything that lives there just kind of goes wild and uses these nutrients to grow. And then this uses up all the oxygen in the water, so that causes a huge oxygen minimum zone. And from the CTD casts that we did, we saw the profile, and I think, was it in this area where the OMZ had the largest extent, about to 500 meters?
Yeah, it seemed like the closer we got to the coastline, the deeper the OMZ ran, the larger it was.
Yeah. Yeah, it'll be interesting to analyze the data because we're also in a transition phase from El Nino to La Nina that might have also influenced our data. But the ocean, upwelling the ocean, what happens in the ocean is usually a little bit behind. So it takes a little while for the water masses to heat up, and so the ocean is a little bit behind the atmosphere in that cycle. But yeah, lots of exciting data, lots of analysis after this cruise. We're very excited.
Lots of sediment to extract DNA from.
Oh yes. Yeah, hopefully there'll be lots of excited students after this cruise too. So the students have been amazing, and they took over all the sampling from the ROV. They subsectioned the samples to look for differences in the different layers, and they did an amazing job. What age classifies methane seeps as ancient? I'm not a geologist. I would pass this question on to...
He left the room.
Oh, he left the room. I'm sorry. We don't have a geologist present. If we can remember, we will ask him when he comes back. I think there'll be geologists on there later.
He knew it was coming.
Yeah. He escaped. So there are differences between ancient seeps and and newer seeps or active seeps. Right now we're looking for active seeps. So we're looking for evidence of methane seep associated organisms. So the organisms that live there because of the methane. Ancient seeps are seeps that were there in the past, and we can find the geological evidence for it. So it's usually certain rock formations. It's carbonate that is being deposited there because of the organisms there eating all the methane. So we will only find the carbonates and some evidence for past life there. So sometimes you can find the clam shells if there were clam fields there. You can find fossil records of microbes as well. I saw a paper regarding that. But active seeps, you can really tell by the organisms that live there. So you find microbial mats, sometimes tubeworms or clams. And there's also some living fossils associated with methane seeps.
That's again why one of the reasons why the seeps are so interesting is it's really a convergence of all the different goals of the project.
This is why we dedicated a lot of dive time today and some tomorrow to really go look for them. Because from the mapping data alone, it is unfortunately not possible to see where they're at. At least not the ones that we have here.
Yeah, ancient's probably not quite the right word because we have no idea how old they are. It could be more recent, but they're inactive. I should say.
We started saying extinct yesterday.
Extinct.
Oh, extinct. Okay.
Yeah, that's a
term.
good way of putting it.
I think inactive is probably more accurate.
Inactive, yeah.
Yeah, inactive.
Yeah,
Probably the best way.
But we are talking about living fossils, so the
we get
extinct
confused.
terminology kind of works.
Yeah, I did some research yesterday and I couldn't really find a good term for it that is consistent in the literature. It was really hard to find good data on that.
Ex-seeps.
Ex-seeps. Extent seeps. Extent. So
So
we'll
are we
be
beginning? Oh, sorry. I was going to ask if we were ascending, going up this ridge on the map.
So I believe John updated the waypoints, did he?
Yeah, we're just.
Okay.
The first one.
Okay. That's the new waypoints? Awesome. So we are faster than we expected to be. So
Which is
we
awesome.
That's amazing because that means we can cover a bigger area. So we're moving up the slope, then we're checking out, what was that?
There was an anemone that went by.
Oh, okay. Didn't like it. We're going up the slope, and then there's a small, I wouldn't call it a canyon because it's not very large, but there's a fault line that we will want to check out, and then we will keep moving up the slope, and then we started the dive at about 3,000 meters, 3,200 meters. We climbed up, went a little bit down. Now we're coming up. And then once we're up on the upper, it's not the continental slope, but up on the upper, I think it's the continental margin. Then we will make a decision if we will keep climbing up because there's a ridge line that we haven't seen yet, or if we go towards a shallower spot that is closer to the coast. That depends on how fast we will be there, because the time we leave bottom is at 4 a.m.
Mm-hmm.
And there's a spot we would like to go to at the very end. So this kind of determines where we go. Well, there's a question: how many waypoints did we have for this dive? Let me pull up the dive plan.
We just added a bunch.
Yeah, we just changed it because we were faster. But
Whoa.
initially, we put in 7 waypoints. Oh, how many do we have now?
19.
19.
19.
Wow, now we have 19 waypoints, but they're much closer together than they were before.
Yes.
So,
We're kind of zigging and zagging here.
yeah. We're checking out geological features that are hopefully interesting and might harbor methane seeps. So,
At the very least, I feel like we're seeing a lot more life around these rocky outcrops than we
Yes.
are on these flatter bits. So
yeah, absolutely. What do they call again? I have a xenophytophores.
Xenophytophores, yeah.
Xenophytophores. It's hard to pronounce. Xenophytophores. That was a very interesting anemone, I agree. What are the things you science people do to prepare for an endure 24-hour dive like this? Lots of coffee.
Yeah.
What else do we do?
Naps.
Naps. Yeah,
Ideally.
we work in shifts, ideally.
Yeah.
Naps, juice boxes,
What
cookies.
is all that?
What is that
What
up
is
there?
that? Could we check that out? Oh,
Ooh.
I don't have my little pointer thing. Oh,
Looks like something.
oh,
What is it?
that looks
Other
like
mussels.
that looks like a clam field. Oh my god.
Oh, could it be?
I'm guessing you don't want to stop here, right?
We do want to stop here. Yes, absolutely.
Can
Oh
we
my god.
Can we stop?
This looks like the thing.
This looks like
Great.
Oh my god! I think we did it. I think we might have done it.
Yeah,
Maybe.
good lord.
I'm gonna give you a different waypoint. We're trying to sleep here, so we're gonna be stopping for a while.
Woohoo!
Yay!
Okay, the sampling starts. Yeah, so those were the types of mussels we were looking for.
Oh my god, that's
That
wonderful.
is
Those are making
It's a yes.
Can you get Alex?
Yes.
Yeah.
the giant clams also.
Those are the methane seeps.
Oh my god.
Oh wow.
I should put a mark here.
Yes.
Yeah.
Uh, wow, look at that.
Oh, okay, let me just take some.
Oh yeah.
Right over, pretty much right over it.
Can you
We're
send me
getting
that
some-
picture
some people-
you took?
Yes.
I'm not at
of
a
course.
good angle to take any from where I'm sitting.
I'm not at a great angle either, but I'm gonna wait.
Oh,
I'm
well,
gonna
we can
send
take
it
lots
in
and
we have a
lots
group
of shots
chat.
from the
So,
ROV camera. But
I'm gonna
oh my
mark this as an
gosh.
observation.
Yeah, I think that would be a good idea.
Absolutely. Oh, yeah. Some of our collaborators already congratulated us, so they were watching the live stream.
Awesome.
This is amazing.
And
Oh, there.
so this is one of the things we said was that
Yes.
there may not be obvious puzzles. Puzzles.
Yeah, I don't see any bubbles on the sonar.
No.
No.
Nothing that seems like super standouty. Wow, look at that.
So that, I believe, you could be an extinct decline in methane seep.
Well, it just could be a very small one.
Yeah.
It doesn't necessarily have to be
Oh.
extensive, but wow. Seeing bubbles? Maybe they're just like super small.
That
Wait, you see the
Racing?
bubbles?
There might be small amounts on that.
Oh wow.
Oh my god.
What an incredible. There they are.
That's,
Yeah.
That's
Oh, they're
amazing.
bubbles.
They're just
Where?
they're
They're just
teeny.
very, very
they're
tiny.
just very
Because
teeny.
the ground is so packed. in,
Yeah.
Probably.
Oh my god. This is amazing.
Wow.
Wow. Yeah, we just stumbled right over it.
Okay. Yeah.
Literally.
Oh my god. Oh my god.
Oh, look over there. Wow. Can we pan up a little bit too?
Yeah, I mean, is there more? I mean,
I think there's more.
Yeah, we
Up-
should
up the mountain a
definitely
little bit.
check this whole area out for sure.
Oh yeah. We will be spending some time here.
Oh boy.
No.
Yeah, there could be tons around here. I don't really see any obvious ones right now, but besides the ones right now.
Yeah, I mean, we'll sample here and then we'll just keep going.
Do we want to do water before we start doing cores?
Oh yes.
We'll take some clams for you. Oh my gosh.
Do we want to consider water first, then cores, so we
Yeah,
don't disrupt
probably.
the sediment?
Yeah, that's probably a good idea. Put the
So
Niskin first, and then.
I guess the question is how many Niskins? Since we found one and we were all looking for it, do we want to fire all of them and just get the water here while we can?
That's
So
a
we're
good
gonna-
question.
We're gonna start with putting the temperature
Yeah.
probe in there.
Yeah,
Adam
that's
was,
a good
was just
idea.
asking if we should do that.
Let's go for
Te tengo que dejar porque tengo que
Ah,
hacer algo que-
and now we don't have the oxygen sensor on the arm anymore.
We don't?
No, I think it's back behind the camera.
Oh, no.
Yeah,
Oh, you're going to grab it. Oh,
that's all right.
it's all good. It's all good. Oh, my God.
Oh, let's see.
Oh, man. I'm feeling really emotional right now.
Yeah.
I'm getting a little choked up. Oh, this was a
Wow.
long time coming.
Yes.
This is a lot of
Wow.
work.
Months and months of looking at data.
Yes, and looking at the bathymetry and stressing and going, are we sure we're going in the right place? What if we don't find anything? Oh, this is so cool. This is so cool. Oh my God.
Party the controller.
Party on the
So the temperature probe is going in. The ambient temperature was 1.4.
So for those of you who are just joining us right now, we have the mission control room for we are going to take some heavy sails. We've been in the water since about 5 a.m. this morning. So it's now 5:30 p.m. here on the ship.
12 hours.
So 12
Yeah.
hours of
Wow.
scanning. We've seen some really beautiful primary fields, but we have a bunch of scientists on the ship who are very interested at the methane.
Yes. Teamwork makes the dream work.
And it's a shot in the dark trying to find this kind of ecosystem, especially so it's really rewarding, it's so exciting, we're all very excited to get to the
No.
balancing network for
Algo muy bueno, lo que seguro no es indetectable. Hay que procesarse para
kind of learn how these organisms survive versus
Era una acampada con el gráfico. Ese no da contenido para sobre todo da contenido emocional para y ahora sí ya tenemos todo lo que
So this is the deepest methane seep site that was found off of Chile. And the northernmost. That's what we just heard from a collaborator. That is amazing.
According to Javier's. So? Not only did we find one,
So we are at 2,835 meters.
In terms of sampling, I think since we have
And-
one, I think we should. But right here, I would say we should fire all the Niskins and get all the water before we start collecting samples and kicking up sediment.
yes. And then if we look around and we find more, we can always do another dive, come
Yeah,
back for more water,
right.
do
I
a
mean,
CCD cast.
we could in theory just stay here.
Yeah, we could.
How we-
We could just hang out on this ridge, looking
Yeah.
around for more. Oh my God. John. Oh my God. John.
All right.
Alex is having a field day.
Amazing.
More highs.
Amazing. I gotta catch my breath. I'm all for clump. Oh my god.
Cool.
Oh, 6 a.m.
Water.
Oh my gosh. This is so cool. Hang on, I gotta touch my husband and
So all we just do first, grab the water before we kick up sediment, and then everybody, I mean, basically, we're just gonna, everybody's push force.
I think after taking all the photos, we'll be nothing.
That's true. There's a lot there, because there's at least 2 in this area.
I think it was all of them, right? Did we have all of the residents?
Do you want to save too, so if we create this area, you find an actual like side message maybe on its way out, or I don't see any bubbles or anything
Oh,
to change, but we can
as
save
many,
some for
as
many as you can.
Yes, yes.
Oh, the
No, of
pores.
course, it's there.
Pores all around. Samples on me.
I don't care if we are here at the one of the
We
ship.
can take up the
So,
same
like
so
maybe
I don't want to just.
maybe we fill the 5 liters for sure, and we can keep 2 of the small ones.
Probably, I mean, it depends on if we're satisfied with the sample.
That depends on what everybody wants.
I don't know, like, I feel like if we find a bigger one, we can.
No, it's job for everyone. It's job for everyone out here. I'm very happy for you. Great.
We know a very good seed with temporary agents and bubbles, and then you have your net skins, and you can always come back to it. Now we know where
I need to know.
you may get an idea.
So
We're here now.
You can. I can give me the location for my
So,
apartment.
one idea is to put down a marker. One idea is to put down a marker around and then come back and collect.
And they're asking you a huge question because they're not understanding why we all are happy.
We
It's gonna
we
take a
found a methane seep. We've been looking for days. We've been looking at the bathymetry data and planning this for months. And we weren't sure if we were going to be able to find one this deep. And not only did we find one, we found the deepest one found off of Chile. So this is incredible. We're going to sample the heck out of it. Get sediment cores, get water,
And are there more deeper than this in the world?
I don't know about worldwide, but off of Chile.
Worldwide, there are some
Sorry,
people-
guys.
20 years ago,
How many
discovered the
discs?
first methane seep
So
in
this
Chile.
was
20
the,
years.
so
20 years ago, they discovered the first methane seep off of Chile.
the
Almost
question
so this
from
is-
is do we want to put a marker here and not sample right away and explore? No, but look around this area. No, it's fair, because if we fire the bottles now,
That's true.
what if we find something bigger right here, right? So
Yeah.
we're not talking about like moving around. We're talking about like just looking around.
Yeah, we have
What?
plenty of time.
long for that look around?
I
Oh,
guess
we can
we could make it quick. I mean,
we can
yeah,
also
I'm-
make it for the next couple of hours.
What-
We've got plenty of time, it's not gonna go anywhere.
That's the problem.
Why don't you guys send a little bit here?
To me, I'd say like 15 minutes to just peruse
Another periphery.
and see,
Yeah,
right,
let's
if there's
look
any
around
more
and
in this
see.
area.
Maybe
Yeah,
there's
yeah.
more, maybe
Let-
it's more
Yeah,
extensive than we
let's
think.
consider, imagine that for whatever reason the rover fails and has to go out. Yeah, so we're here. Let's consider if whatever reason that something in the rover fails. we
Yeah,
have
I
to
think,
leave.
yeah, because
So,
if
just a
we
little
fill
around.
up,
Just
if
a
we
little
fill up
around.
with samples, we have
Yeah,
to go up to
and
the
then-
surface, so.
That's that
mark
was the
this
suggestion
spot.
is we mark it,
Yeah.
look around a little bit,
Look
and
around,
see
maybe
if,
we'll find
you know,
more. But this is, for those of you in the chat, this is incredibly exciting. This is an awesome source of energy for microbes and other life down here. This is all life from chemistry. This is a place in the dark. There's no light from energy from light. So they're feeding off of this chemical energy. So this is incredible. And we could learn a lot about how life in the dark can exist. So
we have 15 minutes while it's still our show. Okay.
Also, please, y'all, don't forget it's dinner time. I know it's
Oh,
exciting.
think about food at a time like this.
Dinner first for later.
I'll be safe correctly.
All right, I am taking over on dive watch. I was sitting in the background, just lurking in the shadows, but
The student take over now.
all right. This is so beautiful.
I think it's in 15 minutes, so I think we can
Oh my goodness.
turn around and then we can
Wow.
something and take a request.
Well, I've got about 15 minutes left, so I'm gonna sit down on this.
Do the party.
Yes.
It's
Yes.
you were the no, you were not here. No, who was it?
Wow, look at that.
You're one of the we're watching.
If you zoom in really close, you can see little bubbles of methane escaping from underground. They're so tiny, we would never have picked them up on our sonar.
Alright, so we are gonna
Oh, thanks, Jeff. Hi.
Sorry, ¿Estás sacando fotos? Are you taking pictures of this?
Yeah, wow. So we may find more of these seeps on this particular ridge if it's a good spot for it. So we're gonna put a little marker down, take some pictures here, and then I think we'll do a little bit more exploring before we start filling up the ROV with samples. But we can always fill it up and come back for more. This is incredible. This is super amazing. Wow. I'm trying to get my heart rate to slow down. I'm so excited. Wow.
Yeah, look at that.
So beautiful. Lot of life here.
Yeah. I see.
Lots of life here.
No, I saw it and I said that I didn't think it was important.
Lots of bacterial production here. Yeah, it could be some fecal casts as well. We've got these bivalves, we've got these amazing crustaceans here, all feeding off of this energy. Living out their lives in the dark, feeding off of geochemical energy from under the seafloor.
So we're gonna.
This was a long time coming. We spent countless hours looking at the data, mapping the seafloor, trying to figure out where the best possible places would be to find these, looking at fault lines, looking at ridges, and we just stumbled across this one along the path that we chose. We're so lucky. We are so fortunate right now. And you're all really fortunate to be sharing it with us. I'm so glad that people are here watching this right now. This is incredible. I think these are squat lobsters. We've been seeing a lot of them on the way up here, but we didn't see any seeps. And so we were like, well, we keep seeing the lobsters because they're often associated with the seeps, but we didn't see any seeps, and now here they are. There's lots of other little critters here too that we can check out. Yeah. Little sea star there. Some isopods. Oh my gosh. Wow. Wow, I'm just speechless. I'm so happy to be here right now and to be in this moment. We worked really, really hard for this. Yeah, so there's most likely a biofilm. I think that there's probably some detritus covering it. There's some greenish color around, or maybe gray. It's kind of hard to tell. But we will definitely take tons of sediment cores here, and if we find other spots, we'll take them there too. Get some of that surface sediment. Yeah, that green teal, I'm not sure if it's actually green or if that's a trick of the light. Over on the left there, around that other mussel bed, could be a biofilm.
for us to start flying around?
Can we just take a quick look at the one over on the left there? I don't know where the pointer ran off to. And put the light on that. I can't tell if there's a biofilm over there or if it's just shadow.
Well, 'cause you're right up against it. Take out the channel.
We have a partner in the room, maybe she can help.
Oh yeah.
Lauren,
Yes.
if we live here, don't we want to throw a lot of sediment?
Will it be okay if we lift up? Will we disturb it?
They seem to be
I think we can be.
gentle,
Yeah, we can just kind of very slowly and gently back away. But we have a marker down, so we know where to come back. So, but yeah, it would be great if we could zoom in on the clam bed on the left here, because it looks like there might be some biofilm on the surface. It's kind of hard to tell, or if that's just changes in the sediment. Yeah.
I think there's some on the one in front was on the right also. You said it was kind of gray.
Yeah, it's a little difficult to tell if it's a biofilm or if it's just differences in the sediment. There's like some white, sort of powdery stuff. That's a good question. Why are the squat lobsters here smaller than the bigger ones we've seen elsewhere? I don't know. No idea.
That photo we need to take, that white spot, because that is something that
Yes.
applications.
Yeah, definitely. We will definitely. Can we take a note of that white area above the clams there? Zoom in on it. Take a picture. And after we fly around a little bit, we can come back and take a core.
You wanted to stop and look at this one though, right?
Yeah, if we can just see. Yeah, we can just zoom in on that for a sec. Whatever works better for you guys.
Yeah, there's just some stuff underneath us, so if we're concerned about these shells and things directly below us,
Why don't we just? Can you just zoom in on it for now?
just float here and zoom in.
Yeah, that's fine. That'd be perfect. Oh, and there's a sea pig. Hi, sea pig.
Are you pushed out? You know,
Yeah.
Lauren,
Yep.
Javier said that she ice.
Ice?
I don't know, it looks more like a biofilm, doesn't it?
No, it's like a biofilm.
It looks like a biofilm, that white. Yeah. Why would there be marks in the sediment between the two clam colonies? Could be animals that were scooting around in between. Gorgeous. Wow. Oh my goodness. Yeah, I think. Oh, thank you. Yeah, hiking trails. Yeah. Now I think some of that, those changes in coloration might be microbial mats. We'll know better, of course, once we've sampled them, and then we can do some sequencing.
Oh, you want an observation of it?
Now that I'm awake.
Please stop there to take a
Thank
photo.
you.
Can we get a close up of the white real quickly?
Oh yeah, a close up of the white, sort of powdery substance there.
So talking with Nick, probably this is an inactive this is because you can see most of the remnants of tubeworms that are highly dependent on active methane being produced. So you see most of those guys dead, half of the clams dead as well, so it's something like everything is going down, but once they're active, who knows?
Well,
Month
we
ago-
saw a couple bubbles coming out, so.
Yeah, but you see a lot of dead worms.
Yeah,
on the
there's a
tubes,
few- there's a-
and
there's
a
a
few
few,
that are still
yeah.
like there.
Yeah.
So maybe it was active, I don't know.
Maybe it was more
Month
active
ago?
before.
Right?
Hard to say.
Months ago, maybe.
Maybe we'll find ones that are more active as we
Yeah,
keep
we'll look.
looking. Yeah. This could be a seep on the decline. It's on the smaller side.
Okay.
We are seeing little bubbling out of here, but it's very, very small. That's, yeah. Oh, that is very exciting for a partner.
Oh, wow, that's
Look at that.
amazing.
Wow. It looks like cobwebs.
Lauren,
Yes.
do you have a sense of what kind of bacteria? I'd be really curious.
Oh gosh, no, not on site. Certainly, we would expect to see anaerobic methanotrophs at a site like this. Those are generally very common at methane seeps. There could also be aerobic methanotrophs, so microbes that feed off of methane. And there are a couple of different clades of those. Yeah. That I don't know. I mean, generally when you see any kind of bacterial mat, what holds that together would be what's called an exopolymeric substance. Or exopolysaccharide. So these are, it's literally like a goo that bacteria secrete to help them all stick together and form structure. So these are just big sugar molecules, basically, that can help bacteria stick together and form structures like mats. I've seen microbial mats that even have like almost like a tower-like appearance. They grow upwards, not just outwards. So depends on how dense the growth is.
The cobweb structure on that is kind of different than what I've seen in the past. I haven't seen one like that.
It's possible that it's methane hydrate. I don't know. Either way, we need to take a sample of that.
Do we want to decide that not to look around and just grab stuff here?
I think that we should look around a little bit more, just to see if we find something. Like there could be, some more extensive seepage, just outside of our field of view right now. But I think we should definitely, mark it, plan to come back. We have so many hours for this dive planned out, and we can just sample until we're ready to come out. So.
Okay, so it is almost time for the grad student takeover.
Oh, the grad student takeover is coming up?
Yeah, 6 o'clock.
They got lucky. This is an exciting time for them to be taking over.
I know.
All right, well,
Hello, how are you? I'm good. I'm even better now, so now I can go to dinner and be like woohoo.
Go upstairs to dinner and enjoy.
There you
I
go.
have already messaged my wife and kids.
Victory dinner.
Because at 7 o'clock this morning, I was like, I really hope we find a mat today. And she texted me a couple times over the day, mat, and I'm like, no mat. Seep, no seep. But seep.
Yeah.
Sorry.
We did it.
So,
All right. Why don't we continue going along our planned path for a little bit, and just have a look about.
you want to do a search pattern? Down, up?
Yeah. Maybe zigzag along this little area here, because we're right at the base of a little ridge here. So.
We can go up about 5 meters and scan the area.
Sure. Yeah. Maybe we'll see something bigger. Maybe this is just kind of the outskirts.
Yes, for too much.
Yeah, I'm gonna. So we're gonna get stuff in the bottom. Just gonna lift off. I can grab the bottom and back to this dimension. Put this everywhere.
Little rocky outcrop here.
That's interesting. That outcrop looks like there might be some interesting stuff on it, so
Oh, yeah.
we'll want to look at that too, probably.
Yeah, we could certainly zoom in on that and
So
see if there's some
is-
animal samples.
The depression that we're seeing in all the lines on there, is that like kind of a secondary sign of the seep potentially?
I think generally what, like, physical marks of the seep would be like pock marks.
Okay.
Those could be animal tracks, maybe.
Mm.
So, methane can be used by microbes as a source of carbon and as a source of energy. And there's a couple of different pathways that they can use for this. But it's basically a chemical form of primary productivity. So rather than using light and carbon dioxide, using methane. And so this is a small example of a seep. There are certainly larger ones out there that are much more actively bubbling that we may stumble upon. But this is a great example of geochemistry fueling life, which is super exciting. So before we do a ton of sampling here, we're just going to take a quick look around. We kind of zigzag around this path that we had started on and see if we can find anything else as we go.
You guys want to take a look at this formation over here?
Where's that at?
It's dead ahead of us.
Yeah. I can't quite. Okay, yeah. Sure, we can head in that general direction. Yeah, so we expect that if we find life on another world in our solar system, like Titan or Enceladus or Europa, that life will be fueled by chemical energy, because those worlds are much further from the sun, covered with ice, and the life may be under meters and meters of ice, and so chemical energy would probably supply the energy necessary for life. How do the clams find or get to the seeps? Oh, I do not know very much about bivalve dispersal, because I don't study mollusks. I know that their larvae disperse and are transported by water, but
I don't know much about bivalves, but if you look up the concept of
We have,
yeah, let's go ahead up there to that white. Sorry, Soph, go ahead.
I think Nick has something he might want to say about the bivalves.
Oh yeah, Nick, please.
I didn't see him in the room there.
Oh, yeah, we have a bivalve expert here.
Bivalves have a veliger larva, which has a planktonic stage. It really depends on it's species specific, and usually they reach a stage where they search for chemical cues to be able to settle down, and usually they like to settle down where the adults are. So, in a way, the adults release chemical cues for the larvae.
Oh, wow. So they have like a chemical language. That's very cool. Bacteria have a chemical language too, so they have something in common.
Some of the cues for those larvae, actually, there's some evidence that they might be quorum sensing related in some way, like some of the stuff that Aparna and I work on.
Zoom in on some of that white material. I think that might
There's
be
even
another
more.
mat there.
Fantastic. Yeah.
Definitely more of those clams, which is a great sign.
There's a lot of it. This
Yeah,
spot.
yeah. All right, we'll have to think about, maybe put a marker down here. We'll have to think about how many cores we can take. Depending on how extensive this is, this might be a multiple dive. Yeah, you zoom in there when we'll make a note of that in the sea log. Yeah, I don't
I'm
know.
gonna take a couple pictures here for the observation.
That to me looks more like a biofilm than ice.
Seep 2, you just want to keep going Seep 1, Seep 2, maybe this one.
Yeah, sounds good to me. Could be a vent and decline because it is kind of small.
Can you zoom in on the...
And there's some dead clams here.
Can we zoom in on the worm, please?
Oh yes, can we zoom in on the worm, please, for Nick? Long boy.
It's a little distant. Let the tentacles go out.
It's pretty.
And that's a polyking parrot, and there's a limit.
If it's possible, I prefer to get the code at Seep 2 because the maps are more wide.
Okay, yeah, we can make a note of that. Seep 2, Aparna would like a sample from.
I'm going to put it on the back of a sheet paper as we're starting to take notes.
And then once we've had a pretty good lay of the land, we can figure out what's going where. Yeah, that I see that spot up ahead. We can head over there next.
So we have hit 6 o'clock. So do we want to let the grad students take over for a little bit and share in this discovery?
I think that would be very exciting.
I am going to go upstairs and force myself to have dinner. I hopefully can put the food in my mouth while watching the TV and not hit myself in the face with the fork.
Yeah, of course we find it right at dinner time.
So
We were asking the chef to put aside a plate for me because I'm not leaving this room. Oh, the bandana fell off. I'm so excited.
well, I'll be back on at 8.
All right.
Bye.
We'll see you in a bit, Drew. Yeah. Good call. I like it.
Okay.
Release the grads.
Yes, I'm looking at some of the comments from the viewers, and it looks like this is a vent in decline. However, still, this is an amazing discovery of the day.
Very happy.
So.
And we may find something more active further ahead. These kinds of seeps, as we said earlier, are transient. They may not last for very long. They come and go, so we may find more active ones later. We've got a lot of area that we could still potentially cover, so the night is young. We had 10 more hours planned for this dive, so we're certainly in no rush. Sea cucumber. He's doing that little, the Tina Belcher dance. I don't know how else to describe that. Looks like there's another white mat over there. Yeah, that one up ahead. Aparna? Yeah. Oh, Aparna's gonna have a field day here. She's all about microbial mats, so. Yeah, looks like there's some more along the other side there.
Check out this one.
Yeah.
You want to speak down and check those out?
Sure.
Another indication that this may be a seep on decline is I see normal animals that I would see in any other part, like within meters. Again, people should appreciate the ephemeral nature of these ecosystems.
He's had a big couple of days. We don't have a thermal plot of this area. These seeps seem, the one that we did measure the temperature on seems somewhat cold, so that would be another indicator maybe that it's in decline. But it's still interesting. Yeah, look at all that material there.
We are not in Mexico. We are in northern Chile, of the coast of Atacama Desert.
That question was directed to Jess earlier because she lives.
Oh, okay.
Yeah, she lives off of Mexico on her boat. Yeah, no, that was from an earlier question. But yes, we are off the coast of the Atacama Desert in Chile, and we found what we believe are evidence of the deepest yet methane seeps located off the coast of Chile.
It's really cool. So
That is super cool. Look at all that fluffy, feathery. Yeah. And there's like some gray material and then the white. It may have been more active in the past. The color variation may be different species in bacteria possibly, or also differences in the sediment. Yeah. Keep it under seep 2, I think. Yeah, that's so close. It's very small. I love when I find fuzz too, yeah. All right. Do we have that logged? Can this?
What do you want? Do you want to add a marker on this one? Yes.
We can keep it under seep 2 if we want to, because it's a teeny. I think I saw some more stuff further ahead, so.
It's on the right.
Yeah, on the right.
Check those out.
Yeah, let's do it.
Let's do it.
Let's do it. Let's do it. We have all the time in the world to explore this ridge here, and maybe as we go further up, we'll find more active seepage. Armando.
My grad students in the lab are celebrating, and they seem to be more energetic than me.
Your dream. Water coming out of the hole.
Yes.
Is that something?
But let me just say that this was the stubbornness of my colleague here, Lauren, that keep going and going and going until she found it. I'm just behind her now. She's
Thank
the
you,
front
Armando. I am nothing if not stubborn.
Just keep going.
So we're just gonna keep looking until we find them. Look at all this.
We've got some matting to the left, and we got all this, and there's some bigger stuff dead ahead. Do you want to go
Yeah.
matting first? and kind of work our way
Yeah,
through?
let's go cruising.
You can notice here that all the bivalves are dead here, so this is, I don't see any sign of alive. Well, perhaps a few.
Maybe a couple, yeah,
Yeah,
but
I
they
see
mostly
one right
look
there,
dead. Yeah. Oh, were you here the other day when I was narrating as Linda Belcher? Or was that just a strange coincidence? Look at that go! All right, Bobby. Oh, wow.
There's some good wording from the audience there.
Yeah.
Precipice of an ancient ship.
Yeah,
that's pretty nice.
That's beautiful. It's poetry right there. Oh boy, a brachiopod. I'm so glad y'all are here for this. Yeah, why don't we scooch ahead and see what's over this little ridge here. Because we could definitely sample here, but there could be more up ahead. Wow.
This goes pretty far. Yeah, this looks mostly inactive, but I mean, I couldn't say how recent. Few clams here still, and some growth, some worms. Sticking his tongue out at us. Yeah, let's keep going. No, I don't think we're gonna go any deeper today. We're gonna climb a little higher, if anything. Right now we're at about 2,800 meters, a little more than that. It's pretty deep for a methane seep. There's some more.
Will you focus on that?
Yeah, can we get a zoom in on that? There's definitely
One of those.
worms.
These are calcium carbonate polychaete tubes.
Polychaete tubes.
I see some that may be still alive. There is some. You see that little pink?
Oh yeah, yeah.
Can we zoom in there?
Yeah, down the bottom left there. There's a little pink worm that looks like it's still alive. Yep. Where's the pointer at? The pointer grew legs. Thank you. Right there. Katie, could you bring me my phone? Thank you. Check and see if my husband got my all caps text from a moment ago. He's probably putting the baby down for a nap or something. You're like, what'd I miss?
Okay, that's a shrimp. Okay, sorry.
Oh, shrimp?
Yep.
Okay. Never mind. Well, no, look, check it out. Check it out.
Those are alive. Okay, no, those are alive.
Wow, look at all those guys. And their little feathery bits. Wow.
Can we flag that also? Polychaete field or something?
Yeah.
It's good. You got a nice photo of this.
Yeah, we'll take a picture, make a note of this one. This is very, very cool.
Polychaete field?
You want a marker on the map called Polychaete Field?
Yeah.
Yeah.
My dad just texted me. He's like, "Are you still on the live stream?" I was like, "Yeah."
I would call this Polychaete Fields forever.
Polychaete fields forever. I like it. Wow.
Polychaete is Polychaete.
CHET, yeah. Yeah.
Whatever you want.
Polychaete.
Polychaete worms here, and
You see the dead.
tube worms. Itty bitty tube worms, and we see some former ones over here too as well. The former shells. And these clams.
Because I would really great experience with those.
Oh, stopped article?
No, because what? No, just
I don't know why I should think I saw it.
new record in new species. No, but
And we'll definitely come back and get plenty of samples from here. I'm also hoping to study the microbiome of some of these animals, so that'll be super cool. Are they static? The worms? Yeah, I think they form these like solid casings, and then their mouth parts come out and move. Can we go out and further to the right?
all these animals are benthic, So at the adult stage, they cannot move. However, they can move at the larval stage.
Yep.
They can disperse, not
Right.
really move.
Right. They're dispersed in the currents, right? Yeah. Here? Oh, can we zoom in on this thing here?
Bump over there to get a really good look at it.
Sure.
It's a little bit out of our way. Should be a view.
Yeah.
As they grow, they secrete calcium carbonate and they make those tubes. Correct.
That's so cool. Life making rocks. Rocks making life. That's what we're all about here. Oh.
Not much excitement there.
My grandmother would have said, I dropped my watch. She made a really loud noise. Hope she's watching this somewhere. She'd be so proud. She'd be like, Lauren discovered a, what do you call it? A seek. A seep. Maybe some biofilm material down there. I think the green thing might be another one of those sea cucumbers. Yeah. All right. Do we want? I think we keep going to the right. Was the direction we were going? It feels like we're kind of circling this spot, but that's probably a good.
Yeah, is that a clam or is that?
This here it looks like another clam, but. There's definitely some kind of microbial mat next to it. Thank you. All right, why don't we, you want to keep going?
Yep.
Yeah, let's keep circling around this spot, and then maybe we can start doing a ziggity-zag.
If you want to go up and take a scan. I think there's more stuff in there.
What will come here first, the bacteria or the clam? Well, there's always bacteria everywhere.
Yeah, I would probably the bacteria, because they're kind of the base of the food web. And then the clam larva, I would think.
As for the sea cucumber question, if we look around, I really think we're going to find the same species we're finding in other parts. It's just
Yeah,
you're focusing on the
Yeah, we've seen a
one
few of
species
these
we see.
here and there. Yeah. This looks like more. There's a few live clams, but I think there's a lot of dead ones here too.
This is not as interesting.
Yeah, I think we can maybe kind of continue to go in that little arc. Maybe.
The acorn worm could be used as an indication that the benthic environment is, let's say, back to normal after the.
Oh, yeah. Kind of a transition away from this type of ecosystem. On the other hand, if these are in decline, we'll probably see more carbonates, potentially. So, tough to say.
You want an overview?
Yeah,
Go up a
yeah.
little higher?
Yeah, can go up a little bit higher. Get more of a wide angle shot. Yeah, this looks pretty extensive, but there's a lot of dead clams. I'm wondering if we go up higher, if we'll see, like higher in elevation, I guess, or but more shallow. Yeah.
You have been amazing to see this area at its peak activity.
Yeah.
Who knows how many years ago?
does make me think that perhaps we'll find more activity close by, but who can say?
I'm not an expert on this type of ecosystems, but I have visited shallow water hydrothermal vent in Taiwan, and I was talking to my colleagues there, and apparently the life expectancy of that shallow water ecosystem, for the people that know, that was next to the Turtle Island, northeast Taiwan. They were measuring it in decades, so the system would just shut down.
Yeah. These can be pretty short-lived, so who knows when this was more active. There's definitely like lots of mats of bacterial growth though here that we could potentially sample. And of course the clams and the worms. To our track. We're still going uphill, so.
This is about the edge of it, there's not a lot of structure after this.
I'd love to talk to the geologists about the path, because if it's, you see, that's associated here with this
Yeah.
little hill.
Yep.
Could either go around here
Mhm.
on the other side.
Yeah.
Check out the whole area. And we can spend a lot of time there actually.
Yeah. So right now we're kind of going up a little hill here. So we could just kind of continue to check out the slopes of this hill. Right. It's almost like a little scarp, which is typical for finding methane seeps. They're usually found in places where the topography changes rather sharply, or the bathymetry, I should say. So I think it would be probably a good idea to just sort of scope this area out, see if we've really seen the full extent of the activity here, and then maybe consult with the geologists and see what they think as far as the possibility of finding more in this same area.
Start working my way back, and then unless you want to go up further.
Let's see, I'm just trying to get my bearings here, like where we are in relation to this hill. We're going up this way, and the waypoint is behind us.
Waypoint is to our sport right now.
Okay.
There's no heading on this one, so.
Maybe.
Not to see it,
Okay.
it's not pointed the same way, but you can
Right.
see here, we actually are pointed that way.
Okay.
See here, the waypoint along the ridges.
Right. Maybe it would make sense to check out this little area to the left of this scarp rather than going over the top of it. Yeah.
We could also, we should probably also go over the top of it, maybe check out the sides first.
Okay. All right. Why don't we go straight up over the top of this then, and kind of see if we can check out the sides. What do you think? What do you think would be best? Just
But it's either this way or here, kind of the same height, or the same
okay.
distance to this, or the other way.
Mhm.
It seems to be here. There could also be more here.
Yeah, I'm not sure.
We have so much time. We can just
Yeah.
go check it out.
Yeah, why don't we just kind of like check out this general region, whichever way would be easier for the ROV to travel. you guys are flying. So I'm saying, hey, pull over and look at that thing over there. So. We are in a subduction zone right now, so we are on the South American plate. The Nazca plate slides under this one. So we're on the South American plate side, which is the continental plate. And the Nazca plate is the ocean plate that goes underneath. That's what forms the trench. I'm so sorry that in my excitement I forgot to introduce the students. So we have a number of grad students in here who have been doing incredible work throughout this entire cruise. We have Miguel, who's a microbiologist. And we have Katie and Amber, who are astrobiologists. Well, they're all astrobiologists. Katie works on space biology, and she's interested in producing food for astronauts on Mars. Is your mic not on? Put on Katie's mic. I want to hear all about what she does.
Unmute me.
There we go. Now I can.
Hello, I am the Katie that has been mentioned. Yes, I do study, unrelated to this, I study sustainable space-based agriculture, but I also have interests in life on other planets, and that's kind of how I found myself here.
Hi, Katie.
Hello.
Amber. Do we have another set of headphones for Amber?
Can we get Amber a headset?
Do we have instruments? I want to study this little hump here, but if we could check out, there's a good way to check out either side. It's a little steep. Yeah, maybe if we kind of go this way. The deep sea flappy thing is a skate.
I think the side matters.
Amber, why don't you introduce yourself?
Hi, my name is Amber Wu. I'm an astrobiology student, and I study the formation of cell membrane before, I guess, it becomes a lipid membrane.
Ooh. Very cool. And Miguel is here with us again. He's been in and out.
Yeah, hello. I've already been talking a decent amount these past few days, but just in case, I'm Miguel Arribas. I'm a grad student from the Spanish Center for Geology. We got some more methane clams, which was nice.
Was that the one we started with? Okay, yeah. No, sorry, Miguel.
Oh, no, don't worry. And I'm also technically an astrobiologist. My PhD is more focused on biocommercial and mostly doing microbiology in here.
That makes more sense.
Why are the organisms at seeps and vents white, like the crabs? Nick, do you know? I'm not sure about that.
I can't see the Facebook message all the way, but that's directed at me.
Right now we're just kind of zigzagging along our path to see if we find anything else, or if we want to circle back around to those first spots that we discovered.
Also, we got a Twitch message a few minutes ago. Don't want to ignore you. You're asking, how can our movements be so smooth? The ROV is hovering and swimming, if you would. We land occasionally, which you'll be able to tell because we kick up a little bit of sediment when we do so. But the reason why the feed looks so smooth is because the ROV is smooth sailing a few meters above the surface.
Yeah, we call it like flying, really. Flying over the seafloor. I have never found a whale carcass. I'm sure some of the folks at Schmidt have found a whale carcass at some point in their lives.
I have a request from my graduate students to find a whale carcass. So
How is Sebastian powered? So the pilots have an answer. By Whimsy. Whimsy. So it's electricity. It's connected to the ship. Runs on electric.
I see that I've been specifically asked a question from the Facebook chat. An idea for Martian food. How could you find out if it's a viable concept? I actually got into it. You could Google this yourself. There is an international competition held by the Institute of Competition Sciences called Plant the Moon Mars. I competed in that for 3 years, and that is a global effort of trying different crops and different methods of growing in simulated Martian as well as simulated lunar regolith. I recommend that the simulated regolith is also commercially available through the University of Central Florida Exolith Lab. You could purchase your own regolith and try it home even.
Loren, look at those sponges.
Oh wow, pretty sponges. Can we zoom in on these real quick?
But my recommendation for you is to look into literature and just give it a try. It's a lot easier than it looks.
We may want to consider collecting them because I haven't seen them anywhere else. It's up to you.
Yeah, those definitely look different from other sponges we've seen. I'm happy to stop and take a sample. Yeah.
It's verified sponges, huh?
We'll take a couple sponges for Nick. I was also hoping to get a couple of these crinoids at some point, so if we're here, now is as good a time as any.
One species, two species. Two species.
Ian, you're back. Did you see the seep?
You're welcome.
Yeah, they're hexactinellid sponges. Hello to Andy Portalatin from Texas, an ex-student of mine.
No, hi. Yeah, I'm thinking of grabbing a couple of those crinoids since we're stopping. Another great living fossil, crinoids. They appear in the fossil record 300 million years before the dinosaurs. So, pretty old. Oh, a whole bunch of people giving shout outs to Nick. That's great. Aw, you missed it. We had a big moment in here a few minutes ago, Ian, but it's okay. We're going to go back. We found a methane seep. So we're just kind of scouting out this area to see if there's evidence of more before we start sampling. Ask Nick. It's for him.
If you can pick up two of those tubes.
Sure.
Entire sponges?
Yes. This is probably the same specimen, but we don't need to take the whole thing. Just two of those, two of the tubes.
Two of the tubes. Okay. What would be the best way to grab those? Suction?
With it's possible to manipulate to get them both, maybe.
Okay.
Okay.
Yep.
I don't want to take the
Oh,
whole
we don't
animal.
we haven't put anything in any of the bio boxes yet, so. Don't really. Whatever's easiest to put them in, I guess.
Yes.
Probably one of the smaller ones.
They're delicated.
The
Maybe we
biobox
can
one.
consider a coral cleaver.
A coral quiver? We can do that.
If we get any bivalves, they'll be very heavy.
Yeah. Okay. Coral quiver it is.
If it fits, yes.
Can I actually get an extra sponge for me? I'll sequence the microbiome. Never seen this before ever. Take two.
We don't want to take the whole animal. That's
No, I
why
don't
we're
wanna
just
take the
taking
whole three,
a
yeah.
partial sample.
Yeah.
Hey Danielle, how are you? Good to hear from you. Hi Griselle, how are you?
Good question for you, Nick. How do you decide what samples to take?
This is from my students, so I choose not to answer any question from my students. Hi Griselle. Yeah, that's right. I mean, so the hexagonal sponges is not just we're taking a sample for the sponge itself. It forms a habitat, and sometimes the association with other crustaceans is very species specific. You may find shrimps that just completely associate with the sponges and are not found anywhere else.
Wow.
Griselle, for your question, the sampling, well, okay. So we have to go back. Every day is planned the day before by the principal investigators. I mean, they take ideas and then they form a plan. And we follow pretty much the plan, let's say, to the dot, unless something extraordinary happens. What we do is we have some idea of what we want to sample and where we want to sample, and we just follow those transects that were laid out from the night before when the Seep maps the general area. Okay? Now remember, this is a discovery expedition. We don't know exactly what we're going to be finding, so a lot of the actions are determined on the spot. Okay? Species ID. Species ID, as you know, for your projects, you know, and for the stuff we do in the lab is difficult. Unless you have an expert, for example, if you got an expert on vents, this person will help us more. Most of the work to take it down to species is happening in the lab. And other studies, this isn't the first sheep that has been found in Chile. Somebody just sent us a paper on the first report of a sheep in Chile that was 20 years ago. However, for our group, this is a very exciting moment. This is a very rare habitat, and if you have been watching the video, this is a relatively it literally a needle in a haystack. Okay, this is complete by chance. You should have been here when the lead group discovered the habitat.
It really was something. Even with all of the bathymetry data and everything else, we were really taking a chance, planning out a course and just hoping that we tripped over one. So that's beautiful. That's a beautiful thing.
For the sampling itself, we of course we try to be extremely mindful of what we're collecting, and we do understand that some of these species are very rare. So keep that in mind, okay? That we don't collect. There's a reason for every sampling.
Can I grab one more for me, please?
Finish. Yes, thank you.
One for me. Yeah, same. Same sample quiver is okay. Yeah, just one for me to do the microbiome, and we'll get a shot of it as well. Make sure you get a picture. Glamour shot. It's a beaut. Thank you much. Yes. Betcha. Ooh, isopod. Yep. Yep. We'll just keep jumping around. Okay. Once. Oh, my finger was caught in my headphones. Once we get this we would like to get some of these crinoids as well. And put them in a different box. I think a bio box would be more appropriate for these. I don't know if they'll fit the quiver. It's tough to sample these and to be delicate when you're using a robot arm that's literally thousands of meters away.
You guys too for.
Nah, that's all right. And we'll leave the rest to regrow.
So we got a glamour shotgun, some firebox then. That'd be tough to stuff in the coral quiver.
Yeah, you can put it in the coral quiver if it'll fit.
Doesn't just any firebox, the small
Yeah, one of the smaller ones.
ones. Glamour shotgun.
Just thinking the clams are probably gonna need to go in the bio box. That's good.
That's good.
Yeah.
Dos especies, sí.
How do sponges regrow? They don't really have tissue differentiation, so you break a piece off, it can grow back.
They have different cell types, but you're correct. Yes, they don't have tissue differentiation, but they have several different types of cells. Plus, they have structure. They have spicules inside their tissue, which makes them keep the form that we see.
Right, that gives them the structure.
Yeah.
Yeah.
Similar to, let's say, the iron, and you lay down the iron and then the cement. Something similar to that.
Oh, yeah. Rebar.
Yeah, rebar. Yes.
Yeah.
It's more difficult. Sponges do regenerate. We know that from there have been several studies already. You can cut a patch and you can follow how they replace the lost tissue. So indeed, since they don't have specialization in the tissue, they can regrow the lost area. Okay.
To answer the question in Twitch chat, that is a sponge that we are currently collecting.
So instead of the audience asking us, I'm going to ask Andy. So since he's listing two of the different cell types that you can find in the sponges, guanocytes and pinacocytes. Andy, what other cells are we expected to find in sponges? You don't have to answer to me. You already got my master's, but the master's for me. But just for the audience. For his master's, Andy used machine learning to identify sponges from pictures from the web.
Oh, wow.
So that's we're trying to submit it in a journal right now.
So next we'd like to get some glamour shots of the crinoids and then sample them, please. And then the geologists have chimed in and given us a potential flight pattern spot to look at. So I'll show you where once we get those crinoids. Rad. Love it. Yep. Stocky boys. What if a flower had a skeleton?
Now we're gonna get the two.
Okay.
Okay, first of all, we're gonna get let's get the mix them. Yeah, but I gotta see them a little. I gotta see them. This is a different species. You can see the.
Okay. Okay. All right. Can we zoom out a little bit for Nick? Nick needs to identify. He says there's 2 different species here, actually. And he would like to get both. Mm.
That may be a possible title.
Oh, okay.
But, so, okay, so these 2.
Those 2?
are
Okay.
different species
Uh-huh.
for the branching order.
Oh, I see.
For example, you can see here that this is, all of them are bipinnate.
Uh-huh.
Those start, yeah, this is definitely different species. Yes.
Okay, so one of each of those.
One of each.
Should they go in a bio box? I don't know if they'll fit in a quiver.
I'm not very good with this.
Yeah. We're looking finance in my house.
Bio boxes? Okay. Okay, so we need... Hi, babe! My husband just jumped in. How are you? All right. I will catch you up in just a moment. We need this crinoid and this one. There are 2 different species, and they can go in a bio box. They can go together, right?
Yes.
Okay, they can both go in a bio box. So, if you're just tuning in now, we found a methane seep moments ago. It looks like it is potentially on the decline, but we did see little teeny bubbles of methane coming out from the sediment. And we found a whole bunch of clams and tubeworms. And so we marked those on our map, and we will come back to sample them. But first, we're going to move a little bit further ahead and see if we can find some more activity. We're just repositioning ourselves so we can sample these crinoids for Nick. We're not in a submarine. This is a remotely operated vehicle. So it's a robot that's connected to the ship, and we are controlling it from on the ship. So there's no people inside of SuBastian. That's wonderful. So exciting. I love crinoids. One of my favorite fossils. Every time I see a fossil crinoid, I'm like, oh, look at that. It's so beautiful. I want it. All right, we're just getting ourselves into place here so we can sample these crinoids so that Nick can study them. Would it be possible to take an extra of each one so I can do some microbiome sequencing on it? I don't know how many of each species are here, and I don't want to over sample.
So they want at least 2 crinoids. Right. Another one, just tall one, and then this one here.
Perfect.
Yeah.
Yes, bio box, please.
So you want
That one, Nick, can you point these out again for us?
Tall one. Let's get the tall one.
The tall one?
I have a message in the YouTube chat from my partner. Hi, Ty.
Aww.
Thank you for stopping in.
Let's go to grab it from the as close to the base as possible. It should be easy to break.
Yeah, right. Close to the root, the test? What?
Base.
Not test. Base, just base. Not test. What was the word I was thinking of? Hold fast, I guess. Nice. Nicely done. Ooh. Pretty. Oh yeah, I do see the difference between them. No, wherever we think it'll fit. It's kind of a long boy, so. A crinoid is a type of echinoderm, so they're related to sea stars and sea cucumbers. But they are pretty much living fossils. They're very, very ancient organisms. So we have fossils of crinoids that are, I think, appeared 300 million years before the dinosaurs. So crinoids have been around for a very, very, very long time. A skinny sea cucumber with a brittle star attached at the top. I don't know if that's accurate, but I like it. I don't know if that's anatomically correct, but it sounds good to me. How big are the crinoids? They can get pretty big. I've seen fossil ones that are huge. I don't know how big modern ones get, but this one's pretty long. We may have a bonus organism of some kind on the crinoid, but I can't see it from here.
It's an amphipod.
Amphipod?
Yes.
Yeah, amphipod. So yeah, my husband is now on the stream. If you could just tell all of our friends and loved ones that I found a methane seep, that'd be cool. I do it myself, but I'm a little occupied at the moment. And I think the other one he wanted is one of these guys. Can we zoom in and get a little picture first? I keep forgetting to ask.
Loaded?
Yes. That one. There, right?
Yes.
Okay, yeah, this guy. Yeah, I can see what you mean now. That is different. The branching is different. Yeah, beautiful. Oh, thanks, Ian. Thanks for sending Jax a message. I had no way to notify everyone, so I just sent a hurried text to Keith to get on the stream. That's beautiful. Thank you. Yeah, let's try to get as much of it as we can.
Guillermo just mentioned to me that there has been reported only one crinoid species in Chile, and we have already doubled or perhaps tripled that record.
Oh wow, that's awesome! So we've found a few new living fossil species today as well as yesterday. That's outstanding. I love this. This is beautiful. Yes, I would. Yeah.
Same bio box, yeah? Yes.
Yeah, I think they go in the same way. So pretty. Very pretty. If you were here earlier, you might have seen fields and fields of these. Almost like a field of flowers, all pointing in the same direction to capture food from the current. Oh, okay. Give the babies a kiss for me, hon. Love you. husband is at home with our 5-year-old and 7-month-old right now. Oh, a sponge? Where? Can you point it out to me? Oh, okay. Do we want that as well? Okay, sure.
That's for the museum, yes.
Okay. Can we also take? I'm sorry. I want one more of these crinoids for myself for the microbiome. Doesn't matter. I think these are all the same species, right, Nick?
All the same species.
Those are all the same. So any one of those is great. And then Nick wants this sponge for the museum. Nick, what are the crinoids made of? Like, what's the? Is it?
What are they made of?
Yeah, like.
I mean, just what kind of tissue?
Yeah. It's a question. Like, do they have an exoskeleton?
Well, these are invertebrates. They don't have an exoskeleton. They have a rigid body.
Mm.
Well, I guess you can say an exoskeleton, but that's a interesting question like this. But a regular crinoid, there are two different types of crinoid. So the anchored, or the sessile crinoids that they are stuck there, and they have this, they have substrate to form the base, and then the stuck is not just keeping the animal upright. And then the living parts, all the living parts of the crinoid are in the tentacles.
Oh, yeah, same one's fine. Thank you.
Another group of crinoids that they have what we call the cirri, and these are appendages that just keep them in one place, but not stuck in one place. They can actually move, and then many people may have seen them as they swim around as a Spanish dancer. So. The majority of the organs are within that feather-like structure.
I think we're getting a free hitchhiker here.
You're looking at a big amphipod.
Amphipod.
Will stay. It's there.
There we go. We get a little amphipod for free. And the sponge can go in the same bio box.
Yes.
Thank you.
Crinoids are filter feeders. They collect microzoplankton or other detritus with their tentacles.
pH at the bottom. Do we have a pH sensor on? No. No. So the average pH of the ocean is slightly alkaline. Wow! Nice grab. Got the whole thing. That was beautiful.
Nice.
So yeah, the pH of the ocean is slightly alkaline on average, around like high 7s or low 8s. And yeah, ocean acidification is certainly a threat to ocean life in general. I don't know. I know that it leads to things like coral bleaching. I'm not sure how it affects sponges and echinoderms.
It seems that we have a well-versed person in hexactinellic taxonomy, and are calling the sponge we just collected of the genus Volteria. If you know more, please let us know.
Yeah, some of the fans in the live streams are extremely knowledgeable. Certainly more so than I am as a microbiologist. I only know how to ID these these organisms in a very general way, which is why we need folks like Nick and Guillermo to help us out. Yeah, we got some nice specimens here. Okay. Pilots, do you know where you're going next? Oh. We have a request to zoom in on this rock over here quickly.
There was a possible chiton.
Maybe a chitin over there.
For Lisa in the audience, I'm not confirming, I'm not expecting exactly name, it's Lisa. That's why I was asking you back.
So we want to kind of go to the left of this ridge towards that next waypoint.
Okay, so stay to the left.
Yes, please.
Head up down. Okay.
Yeah, that's what the geologists are saying.
Okay, no problem.
What's up, Emin? Can we go over this way? Keep going to the right. Maybe up.
You want to see it
Oh.
better, we just reposition.
Yeah, we might need to reposition.
Yeah.
Oh, I got called a young lady. Thank you, that's very sweet.
Dr. Lauren Saylor is the lead person, and then there's a group of 2 or 3 people around that have additional opinions.
Yes, we have quite a few amazing students, graduate students on board this expedition who have been absolutely invaluable to this work. So we have Amber, Katie, and Miguel in here right now with us. This was supposed to be their dive watch, but I sort of had to take over for a moment.
It's okay. Drew said we can stay through his.
Aww. Drew is a mensch. He's a good guy.
Did you see Drew?
Yeah.
I'm right here.
You're a good guy, Drew. We love you.
Oh, I love you guys too.
Aww. All right, where are we looking? To the left?
Just said we were out of stuff.
Here, let me give you the pointer. Oh, that's right.
I'm sorry.
Getting your left and right mixed up. No worries, we're all pretty excited right now. Maybe around the other side if I'm wrong.
Say what?
Zoom out a little bit. I don't see it. Maybe it was that. No? Something different? Oh. Oh. Oh, is it that down there? Maybe? Let me zoom in the bottom left here. At the base of the crinoids here. Maybe that was it. That's a sponge. I think they're both sponges. Okay. All right. Let's keep going. We're gonna go up to the left of this ridge, because this looks like a potential place to find more seeps. Possibly they'll be more active. But in any case, we've marked the location of the first ones that we found, so we can always come back to those.
Sorry. How's that? Is that good, Tyler?
To the Twitch chat, we are very excited to be included in the mission control room. I personally have spent the last few days in the wet lab filtering up near 200 liters of seawater, and because our vehicle is down for 24 hours today, I have the time to come have fun.
Yeah, this has been, because this dive is so long and we haven't taken any samples yet today, it's been a very relaxed day for us. We've just been flying across the bottom of the ocean, keeping our eyes peeled, and our patience has paid off. So it's very exciting to have everybody here to see this.
Yeah, new update. We're just going to be jumping to the left of this track line and heading towards waypoint 2 on the left side.
Tomorrow's going to be a very exciting day to see all the samples surface.
Yeah. Yeah.
Okay, understood. Thank you.
Wow.
Updated waypoint.
ROV watching as a hobby sounds like
Updated waypoint.
an awesome hobby to have. I think I need to add that to my list of hobbies. Just put them on in the background while I'm doing stuff. That's fantastic. It's so great to know that there's fans of SOI that are so dedicated. The meteor-like rock collected yesterday. I wasn't on Dive Watch yesterday. Does anybody know what rock they're referring to?
What about your sticky rock from yesterday?
Sticky rock from
The one
yesterday?
that was hard to get? You have a question in the
You're
chat?
asking about it in the chat.
We're asking about Miguel and his rocks.
Tell us about your rocks.
So answer, it was not a meteor. We looked into it. It was not a geologist, but a couple of our geologists looked at it. So it broke upon collection. It's had some metallic parts inside, but most certainly not a meteor. I have kept it safely, so.
Very cool. Someone was asking what keeps the water in the Niskin bottles and the bio boxes from popping out like soda at sea level pressure. We do rise pretty slowly, so I think that it has a chance to equilibrate.
Those biobugs are not 100% sealed, so there's always.
Yeah, they're not air or water tight exactly. I mean, not the bio boxes. The Niskin bottles I think are pretty watertight, but we do rise up pretty slowly, so they probably have a chance to equilibrate to the pressure. Yeah.
Do you have a distance on the other side of this line you'd like to go?
We're aiming to be on this side of the ridge, so to have this like on our right. Yeah, if we can. Yeah, if you can aim to be in that little divot there. I like thinking about how dark it would be down here if we just turned off all the lights. Just this whole world in darkness.
It's spooky.
It's very spooky. Have any clay samples been taken? Yeah, I would say that our sediments have had some clays in them, for sure. The geologist could speak to that better than I can as to what the sediment cores have looked like so far.
I can't say with certainty, Ty, but I think some of what I'll be bringing back to our lab will have some. Once again, can't say with certainty. I'm not a geologist.
That's the buzz phrase of this entire cruise. I'm not a geologist, but thank goodness we have a few.
Drew says hi, Ty.
Alright, we're just gonna make our way alongside of this ridge here. The backscatter data for it looked really promising.
People in the chat are questioning how cool the uniforms are, the ones that we have, and they are comparing us with the cartoon show called Sea Lab. And I suggest to the PIs that we need to have a nice uniform, encompassing the mission.
I agree. I would like to have a speed suit like on the Venture Brothers. Now, it's not a jumpsuit, it's a speed suit. Oh, that is a shame. Venture Brothers is a great show. It's a very, very funny show. Hi, Dad! My dad is in the chat. Hi, Dad. Everybody says hi, Dad.
Hi, Lauren's dad.
Oh, the forbidden calamari. I did see that get captured. That looked pretty gross. I wasn't there when they took it out of the ROV, but I imagine it was pretty stinky.
It actually didn't smell very bad, and it still had
Really?
its beak completely intact.
Wow.
So we were looking at that for a little bit.
That's cool. It didn't look like it was that complete, so that's awesome that you got the beak and everything.
Its tentacles were a different story, but
Yeah. Well, I saw those were like broken off or chewed off or something like that. We did not slurp the forbidden calamari. We pinched it. We grabbed it with the grabbers. That looks like more biofilm. Can we zoom in here and get a little picture? What are those little guys? There's some clams here, but it looks mostly like shells and ooh. Beach? Those are fun.
Long dead bivalves.
Yeah, those are definitely toast. But there's some biofilm looking material here. If we could get a shot of that. Log it. There's also some stalks looking things sticking out of here. I don't know what those might be. Maybe crinoids. Yeah, drop a marker here. Maybe we could come back to it. Because this is a pretty extensive mat. Well, that's up to Aparna if she wants to take a sample here or somewhere else.
Okay. See?
Okay, Aparna would like to take a core here.
The first phase I don't want to disturb because that was so it's fine.
Okay. Brad, we're gonna stop here and get a core.
Yes. Yeah, in the highest concentration of the white here. Here, yeah. Oh, sorry, I pushed too hard.
Okay.
And we're gonna have to be gentle so as to try not to disturb that top layer there. Yes, please. Start with 1. 1 and 2 are reserved for Aparna because she needs to take her cores off first.
Sorry, you did say start with 1.
I did, yeah. Yep, the better. Yeah, okay. Can we just go very, very slowly on this? Thank you. I'm going to try not to disturb that top layer if we can.
For Andreas, that is commenting on the chat, bacteria are the beginning and the end, not just the end.
Yeah, that's a very good way of putting it. And that applies to lots of things, not just methane seeps, for sure. Beanie weenies.
Aparna.
I'm sorry, that just made me laugh. It's got nothing to do with microbiology or marine biology, it just made me laugh. All right. Yes, Aparna, can you show us exactly where you would like this? This is your show. Yeah. 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 That looks like a good spot right there. Oh, a little over to the right. Oh yeah, that looks really good. It is definitely a bit like a space operation. We are using a remotely operated vehicle to take these samples. Oh. Okay, yeah, we're trying very hard not to disturb this top level layer here. There's another clam there over on the left too.
Is it possible to suck it all these white things?
Suction it? I don't know.
But then the problem is in which box you will be keeping it so that it will be totally.
I don't. If it goes into the suction, it'll go into one of the jars, but there's a mesh at the bottom of those, so I don't.
I think it would say at the top. I think you got the hard core stuff. Doesn't matter.
Yeah.
It's not fine much.
Right. I don't know if putting it into one of the suction sampler jars is the best way to do it. I think a core would be better, probably.
But the thing is, that time it was all went out. So maybe here, but we have to put it in one because it's very closely bound. So
Right. Maybe if you just take instead of pushing all the way down, maybe just take the first little bit.
So that's another question. The depth is 5 centimeters, 3, 2?
3 is fine.
Just
5.
want
My
that
interest
top
is
layer.
just that part.
Yeah, she really just wants the top layer, so.
I thought we could include as the segment more, right?
Yeah, I think right about there, please. Yeah.
City cover with the feeding tentacles out. Usually these things buried in the sediment and all the tentacles out.
Oh, that looks beautiful. Awesome.
For the size of the mess, that matters. Of course, it matters. That really depends what the individual researcher wants. So a mess for one person may not be suitable for a mess of the other person. But you are correct with pointing that out.
We just, I think, very, very carefully and then gently lift. Ooh. Oh. I think this is the first time we've actually had a failed core. The material here is just so loose.
Oh, I'm wondering if there's a better way to do this. Yeah, I don't know if a core is gonna be the way. Oh, maybe it's still in there?
The core.
I don't actually know. I don't think it failed.
No, the core did not collapse.
It didn't.
It's fine.
It did settle. Okay, okay. I thought we lost all of it. Wow, okay. Sorry, I won't jinx it. I'm not gonna say a word. Yeah, I say, yeah.
Lauren.
Oh, is that just fluff? Oh, yeah. Oh, yeah, that's all like suspended. Yeah,
What about the scoop?
maybe the scoop, but then that's not gonna keep it intact.
Correct, correct.
We could sediment scoop it, but I don't know if the top layer will stay.
The scoop, yeah, the scoop
Stay
is
together.
We might be able to if we just go around. Maybe we can get some, and maybe it'll settle in the bottom of the jar. We can try.
Uh-huh.
Suck it very carefully so we get it
You want to try down there? Maybe it'll be a little bit more firm. We can try it if you want to. Right about there? Okay.
Come on.
Come on. Is this where you want me to go? Before I
Ye-
start going down?
That was the spot, right, Aparna? Yes. Oh yeah. Oh.
He ran away.
It's a rock or that's solid.
All right. Just have to be slow and patient, I think. And we'll see. We'll see if it pays off.
For the audience, isn't lubriculus an oligochaete? I don't think they have marine representatives. Please correct me if I'm wrong.
Aman.
Ah. Okay. Okay.
This is claw machine level 1000.
Lauren,
Yes.
if this core is successful,
Mhm.
I have a request with Armando to do a core on top of a living bivalve a little to the left. That is going to include the bivalve and the sediment down to 5-10 centimeters.
Okay, yeah, can do.
Core number 3.
3. You got it.
Yeah, because number 2 is 4.
I think we need a round of applause for the the ROV pilots right about now. It does look like a Winogradsky column. All right, we have a request for another core with a living bivalve on top, if we can. Yeah, core number 3, please. Yeah, this one's for Nick. I think we saw 1 or 2. Okay.
We're waiting. Are you going to share your folder? Danielle wants a code. Does he want a separate one, or is that you're going to share?
No, that's first is first.
Okay.
I want to take the one for
The voices that you hear in the background, these are scientists of the and graduate students of the expedition, and we have meaningful discussions on how to pick sampling sites and what priority we have at that particular moment. So, slightly to the left.
Slightly to the left? Oh. Oh. Did that have a live bivalve on it? Where? Oh, the little guy. Oh, okay. No? Okay. Left? Left? There. Okay. Oh.
Hey Lauren, just to clarify, did Armando mention that Drew and I want to take a core here of the film as well?
Yes.
Okay,
We're
thank you.
taking 2 more after this one. One for Daniel and one for you.
Perfect, thank you.
You are so welcome.
Yeah. Katie just did. Where?
Oh. Actually, could someone take over for me for just 5 minutes? I need a biological break.
Sure.
Thank you.
Okay.
Direct core sampling. I'll be right back.
Okay.
3?
Yes.
Okay. No. I've got a request for another core, number 4, on top of the white bacterial mat.
No one's listening to you, so we can just they don't have.
It's a
Look at here.
work for them.
Let's finish with that first. Perfect. For the pilot,
Sorry.
Perfect.
Fish core number 4. It's behind the arm, but in a really dense white area. We're also doing the
That's for
And
my
4 is my lucky number.
you have to appreciate the excitement of people trying to get us, as many samples as we can, as we possibly can. Now, again, remember, a sample, of course, it takes a huge amount of effort to be here and be able to get those samples. But the vast majority of the work is done back to the lab.
Here?
Yes, please.
Andrew, are you guys going to be doing 16S or metagenomics here? So, we sequence everything. Okay. Looks like a very successful core with microbial biofilm intact.
Beautiful. Big thank you to the ROV pilots. Yes. There is a lot of white on the top of that. I'm going to have lots of fun.
Car number 5, look at my finger.
That's why if you just go out in one spot, they come back.
course is this?
Geology. This one.
Geology. Okay.
Maybe here. I don't know if
You're gonna get, you see all the gray matter, that's from the bottom of the other core. So
Mhm.
Oh,
to get
okay.
rid of the mix, you know, you see what I'm saying? Where it's like dark gray on top of the white? You're okay with that mix. But it's okay, otherwise we can reposition if you want to move to the left.
Yeah, I think that is better to check out another one here.
Yeah, we can move, just see the clean patch over there. We just move right over there.
There's more.
Yeah.
But wait, there's more.
There? Yeah, okay.
If you can take this, would be great, please.
That's there?
Yeah.
That's what you're looking for? Okay.
I won't be able to tell you guys on this stream, but maybe on the next stream, I do smell every sample that comes up because all 5 of our senses are really important when it comes to things. So minus one sense. I am not tasting anything. I would like to specify.
Good night and thank you for watching us and commenting.
Technically, you can taste everything at least once, but
That all switch towards you?
I think that's all it is.
It's all here?
The horse?
not from this spot.
I think that's, no, we got ours, I think we're all good.
No, we didn't do number 2.
We will know how it smells by tomorrow when we process, of course. We'll report back.
Dear Celso, I am from Greece and I assure you, people in Greece do not eat sponges. I do not know who told you this. Maybe perhaps you mean that there are some islands, for example, like Calyptus, that traditionally people do collect sponges for the commercial sponge that we use in the bath.
You've deputized yourself?
I guess so. Leaving temporary, the non-Dr. Lauren.
Okay, so when we last left our story, we were working our way up towards this marker right here.
Sounds good.
Sounds good?
All right, we're gonna continue moving forward. I am briefly taking over for Lauren. I'm Drew Palmer.
She doesn't want to hear the report.
I want everybody to go to Calyptus. It's a beautiful island.
The species.
Okay. All right, Lauren is back.
She's back.
I'm back on the date.
Was it good to be the king? It's all good. I'm more than willing to give this seat up in a little bit so that I can go eat something. Haven't had my dinner yet.
Oh.
Dr. Drew, you're popular.
You are popular, Dr. Drew.
So I won't be eating anything that we take from here, but in my work off of the ship, we grow different crops in simulated Martian regolith, and I can say I have actually tasted tomatoes that have grown out of regolith. So there are some times where it is okay for us to taste our work. I just don't think this is one of them.
In this case, do not lick the science.
Don't lick the rocks.
No. Unless you see a geologist do it first, I guess.
Only Oscar.
Oh no, they discovered the haunted shipwreck while I was gone? This is why you never take bathroom breaks.
If the geologists start licking rocks, we're in trouble. That's why we never leave them alone.
No. You need to be supervised.
At all times.
No, that's, I think, the Falcor's point. We're going up along this transit here.
Deep sea gastronomy may be a relatively young field. However, there are established recipes. Let me remind the audience, and I'm sure many of you know, that there are some great recipes for bathynomos. For the people that know, bathynomos is a giant isopod, a scavenger, and it's about the size of a lobster. Deep sea gastronomy is a real field.
Ah, the forbidden gherkin. I love those. The color, this is so silly, but it reminds me of like a green velvet sofa. Like I don't know why it makes me think of that.
Better put the plastic cover on.
Yeah.
That's right.
Can we stop and grab a crab?
Grab
a crab. I'm guessing with the suction sampler is probably going to be the easiest way to do that.
I agree. Some of those bathynomos isopods are amazing. When you put a little, you're just gonna cook them either on the barbecue or on the boil them. Make sure you empty the stomach, okay? Because remember, these are scavengers, so they're gonna eat everything that they see from them. You can just Google bathynomos recipes and you will see several ways to prepare those isopods.
Yeah, that's fine. Jar 1. Crab grab. Operation crab grab. I know it's a lobster. Squat lobster. You can eat a giant isopod? Well, that's news to me. I didn't know that. I believe that they taste like a crustacean, though. I mean, that would make sense. I do like crab and lobster a lot.
Escape response is backwards for the squat lobster.
Yes, it's going to. It swims butt first, so.
Yes, he has nowhere to go but up.
Booty first.
Can't go through the rock.
Oh no!
Or that way. Definitely go that way.
It is Doug on the manipulators, and Tyler is flying at the moment. The chat wanted to know.
He's on to us.
Look at that shrimp. He looks like a Muppet. Oh, that's a good strategy. Yeah. Can someone jump in for me for just a sec? Drew?
Let's just take over 82 worms.
Yeah. Moriel needs me.
Thank you.
All right. This time I'm back for real.
Yes. Awesome.
Ooh, it's close. I don't think we have a net. Please, crab. We just want to be your friend.
It's a very crafty crab.
I think it's a squat lobster, but we just want to be your friend.
For those of you who are just joining us, about 2 hours ago, 2.5 hours ago now at this point, we actually identified this fairly sizable area that appears to be a methane seep. Yay! Right. And is that going into sampler 1? Oh, just too soon. I'm watching the jar cam.
Where did our check? It just disappeared.
Is he stuck in the sampler?
He's holding himself in the tube around the metal ring. Yeah. So please. Oh, he's still there. That's a strong little lobster.
We're trying to catch this little lobster here.
Oh, perfect. Thank you.
No, he's still there. I can see him. You can see it's all.
While
Yeah.
you catch the crab. I'm gonna go.
Right, so while we were working on getting this crab, I'll just again say we about 2.5 hours ago identified what appears to be a declining methane seep, but it is an active one. And so that was one of the main goals of this expedition. And so here we are. We've been on a dive since about 6 o'clock this morning, trying to identify one of these. And we found a mound here that appears to have a fair number of them. And currently we're doing a little bit of sampling, but we're starting to explore the area a little more in depth while we capture a few sediment cores here to look at the microbes and whatever metabolites might be leaving behind, but also trying to catch some of the larger fauna of the area. We've got a lot of excitement in here.
Andrew, did Lauren have a plan on how long she wanted to rover around until we go back to the original site? Did she mention it?
I believe there probably was one, and I don't think I know. So if somebody in here wants to go ask.
Sounds good.
I think the tube narrows. I think the tube narrows at that point.
This is the craftiest crab that ever lived.
I think the leg is stuck.
Is the tube able to eject? Could you eject into a bio box?
No, he's like holding on. Wow, that is spicy. I respect that crab.
Can you hover over a bio box and turn the suction
And
off?
eject it, I wonder.
Yeah, eject or turn the suction off.
Would it be possible to try ejecting it into a bio box?
That's a good question. I think the problem is it's holding on pretty tight. They were wondering if you can eject it into a bio box. Can you eject it into a bio box and reverse section it?
There is no reverse suction.
There's no reverse
no.
section.
Okay. Yeah. Actually, the tool is not capable of reverse suction.
There's suction and no suction.
Yeah. Yeah. 2 options: suction, no suction. That's what Hannah in the room just said.
Can you hover over the bio box and then turn off suction and then like shake it?
He's
Well,
holding on.
they've been turning the suction off, and it's just not going
Yeah,
anywhere.
He's just holding on.
Hey! See one?
Is it in?
I don't see it in the jar yet. There's a lot of sediment in there.
Yeah, I can see the shadow. He's fighting it. See him right there.
He's trying so hard.
That's all the shadows.
Oh.
That's suction off.
Are we stuck in there?
He's probably stuck. take this or this or this or this.
Yeah.
That's okay.
Looks like he's in a bit of a pinch.
We're getting a pilot change. So with me in the room, we have a couple of graduate students who are riding along with us. I wonder if you guys could introduce yourselves, since we might have some new people on.
Okay, I can go first.
Okay.
My name is Caitlin Hubrick. I actually study underneath Dr. Palmer, the one who just introduced me. I am an astrobiologist. I am currently working on my master's degree. My research is primarily focused around sustainable space-based agriculture, but I also have interests in desert microbiology, as well as life in other planets and moons in the solar system, which is why I'm here on this trip. I'm going to pass the microphone off to our next grad student.
Hello, my name is Amber Wu. I'm a PhD student studying prebiotic chemistry on the origins of life. My work centers on experimental and computational chemistry, looking at how coasperated droplets can be a precursor or, I guess, model for cell membrane, cell compartmentalization before lipid membrane forms.
And Miguel. If Miguel would introduce yourself to the viewers at home.
He's passing that.
All right, and next up is Miguel, who's also my data logger today. There's far too much joviality in this room.
I am Miguel, sea level extraordinaire, microbiologist, zoologist, some microgeology. I'm going to start that and really focus too much on my microbial side, which is something I'm actually doing here currently. My piece is focusing on how this organic matter and specific molecules degraded under the extremely damaging conditions of radiation on the surface of the planet, which is assuming has absolutely nothing to do with this. I am also a microbiologist by training, which is why I'm here. We're going to focus on studying the transcriptomics, the specific proteins and processes that are currently being favored by the organism. It's not in comparison with genomics. That genomics will tell you everything that an organism has and can do. Transcriptomics will tell you what it's doing at a specific time. It will analyze the messenger RNA of the cells. It's been pretty nice to hear. We're still trying to find the lobster, so we may still hear
Before a little bit.
But I think we'll try to keep quiet.
Just to kind of continue up along that line.
I just want to say thank you for all the...
Did we lose the suction sampler? Okay. Okay. So, Miguel, can basically, for right now, just make a note that the crab is in the suction sampler. We can still use it to bring other stuff across, but he's somewhere in the tube, so we'll just be on the lookout.
I'm going to draw a tube in the ROV SuBastian.
All right, so we're going to continue moving forward up this ridge. We think there may be even more methane seeps here. We've still got several hours left planned on this dive, so we're going to continue pushing forward and see if we can find some other sites, and then make our way back here to collect more. Okay, ready to go up?
Yeah.
Not like all the way, but up a little bit. Yes, move forward. That's how I should phrase that.
have just been notified that I have a really squeaky chair, so I'm gonna try to stay still.
I didn't want to say anything.
I was adjusting my legs, I'm sorry. Miguel says I have to go, I must go. No, just kidding, just kidding. I'm staying. From
I had to summon the squeaky chair the other day.
Yeah. We're just following that line up. All right, and we're continuing to follow this line up. The geologists in our crew, based on some of the data we've been seeing, think that this is a good possible site for finding some more of this seep that we're tracking. So, for those of you who have not been on for a little bit, these methane seeps offer sort of a spot of high energy and productivity in these marine environments. And so these are sort of reservoirs or hotspots of biology. Oop.
Sorry,
Hold on one second. I think there's been a change.
About what our plan is for tomorrow's dive. So right now, I would like to continue along this path up this ridge, which is going to take about an hour, and see if we find 1 hour. Well, yeah, and then we're going to come back.
Then we come back.
So,
No.
yeah. So we'll go up that side, see if we find more active seepage. If not, we can turn around, come back to the original spot, and do some more searching.
That'll put our
And then
sampling
even
at
if
10
we go
o'clock.
with the ROV, we can spend the rest of the night just looking around for more spots, put dots down, and we can plan another dive if we want to come back.
So to wrap that up, we're going to spend an hour moving up this ridge line here, and we're going to be looking for possible additional methane seeps. And then as we figure that out, we will either stay in those spots and sample, or we'll turn around after the hour and come back down to this area, which has been shown a lot of productivity, and we'll grab the remainder of our cores, our water samples, any animals of interest, and then kind of move, then start to look around a little bit more at the remainder of the evening. So this dive is planned until 4 a.m. I'm going to be on here until about 10, so I'm just starting. And thankfully the students have agreed to hang out with me for a little bit longer. Not that everybody else isn't awesome too. And then we're going to see if there's any more exciting seeps to find.
And there's going to be 6 or 7 long line of crabs in each.
The crab chain.
For those of you who missed the previous evidence of the methane seep, there was just a lot of oysters, clams.
Bivalves.
Yeah, collection, a high density of organisms there. These spots, we zoom in, we can see there's very light amount of bubbling occurring. Some of these sites have a lot more bubbles, but that's not always visible.
It's really loud. Yes.
I'm just going to zoom in these bivalve looking things.
Okay, one of these?
Yeah, these were the one.
Okay.
To the. I wouldn't sure, just look at this bivalve.
Is it possible for us to get a zoom in on one of these, or is that? Yes. Yeah.
Yes.
Yeah.
Yes.
I think it's because it's pretty thin. Hopefully, it'll just sneak around.
No. We just want to zoom in on.
Oh,
We don't want to. Yeah. Sorry. Okay.
With the reality check, none of the stuff.
Okay. All right, we're good to move on. Thank you, guys. So with the range that we've already seen on this particular seep, which kind of appeared in a few spots that were a bit spatially separated, it's not completely clear the extent to which the seep is influencing this area. So it's kind of one of the reasons why we're running through a larger area to look. Oh.
I think we'll have things in the back.
Yeah, I got it. We could take a look over there. I think we might have another possible sign here. You can see the shells collected there. That's been the telltale sign. And there's some there too. Awesome. Cool. Could we zoom in a little bit on that? Please. All right, I'm gonna zoom in a little bit. It's looking pretty good. So this is the right kind of materials. So, I mean, it looks similar. I think there's a lot less here than there was in the other sites, but yeah. This one looks like it's, yeah, I think this one's passed already. So if this is a declining seep, then this is probably one that's already, this part's already past its prime a bit. That one?
This looks very much like trash.
I already talked to them.
Plastic one.
I was saying that it's quite interesting that we're finding evidence of methane seeps so far away. Not so far away, but this could be a way away. So.
Yeah, we can proceed. Sorry. So that would be actually, can someone get Oscar potentially? Is he available?
He's right here.
Yes, that's why I was asking. Can you grab headphones real quick? I want to ask you a question.
Yeah.
So I was kind of making a comment, and I wanted to get your thoughts from the geology perspective. We're looking at this, and it seems like the seeps are sort of spatially distributed along the mound as we're going up, and so is that sort of a common phenomenon that we see sort of a patchy network? And do you think that is a sign of it potentially being in decline, or is there insufficient data to make a?
So we try to find the methane seeps in this area because this similar place that all the place that we are know in Earth that exists these methane seeps in Japan and in China, in the front of the coast of the U.S. and also in the south of Chile we found evidence of the methane seeps. So we try to find the same feature, the same structures that we found in this place to try to recognize these amazing places. So I don't know if I can answer well your question, Drew, or you need something else?
No, it's okay. I think that's a pretty good answer, though.
Okay, thank you very much.
Could be. Are we headed up that way? Yeah. Okay. All right. Yeah, I think that's taking us up that way. Found whatever path Lauren set for us. So this has been very exciting couple of hours, having been on the hunt for multiple days, and then today alone we started the cast I think at about 7:15 this morning, and been going nonstop since. It's been exciting.
Maybe we could explain why are these methane seeps interesting, and how microbes that feed on them are actually interesting, how methanotrophy works, maybe compared to methanogens as a comparison. That would be interesting.
So without getting too complicated, I mean, one of the reasons why these areas are of real interest is that a lot of the benthic environment here at the bottom is reliant upon this organic snow that you're seeing falling here, which comes from basically it's the detritus of material in the more productive layers of the ocean, in what in this area is the oxygen minimal zone, where there's so much productivity from photosynthetic organisms, and that kind of crashes down as they deplete the oxygen and pass away. So in these methane seeps provide a different source of carbon and energy to the organisms down here, and that creates a new biological hotspot that's driven by energy from below as opposed to energy from above, from the sun. So this is sort of geologically powered, if you will.
Yeah, like what I meant is that maybe for all of us astrobiologists, it's more interesting to see how life organizes around these methane seeps, which can be abiotically, that means generated regardless of life, although they can also be generated through bacterial activity. Because from an astrobiological perspective, most of the ecology on here is centered around organic matter that falls from the top of the sea, and it already assumes a very developed ecosystem that we may not find in other places, while an ecosystem that solely relies on methane may be more easy to be generated in maybe more extreme places, or it does not rely on the presence of a hugely developed ecosystem as most of these sea environments do.
Gotcha. Yeah, no. So I was thinking about it from the earth biology perspective, as the astrobiologist. Yeah, our interest in a lot of these environments is because of the relying on energy from a different source, just like you said. That's a perfect explanation.
Maybe that way it's a bit easier to understand why Lauren, who's an astrobiologist, was so excited and so interested in finding these methane seeps, as they're not by far, but they're some of the more interesting places to study astrobiologically, while just studying the sea sediments may not be so much astrobiologically relevant.
Well, some of these bacteria are also interesting. That might be in these environments may be interesting for understanding the past history of certain plants. So whether or not the possible role of methane on Mars over geological time. There's some models that microbial produced methane may have been responsible for sort of a global cooling of Mars, which ultimately might have made it uninhabitable, not habitable. And so that's something that looking at the microbes here and how they're able to shape their environment could give us some insight into those processes as well. And we're continuing along this trail for a little bit longer. We're going to stay on this path up this ridge for about an hour. And then at that time, as we near 9 o'clock, we'll come back around if we haven't found any richer seeps, and we'll do a bunch of our sample collections probably. Students have any thoughts, comments?
They don't.
You're also a student.
Yeah, but I already had comments.
For the trained eyes of the pilots, we're also looking for chitons and brachiopods. It looks like your eyes are just amazing when it comes to chitons.
Yeah, on yesterday's dive they found two.
I'm just trying to connect.
Amazing.
So the green line that we're just passing by, can anybody guess what it is?
I cannot.
It was a long, thin green line,
Is that
biological
laser?
material.
Oh,
I did not
I didn't even see
see it.
Oh, this?
Is it one of those medium
Yes.
things?
No, it's one of those weird green ones.
So the hint is at the end. So I would like the audience to give it a shot. We have seen several of these specimens. Oh, somebody said it already. I heard that.
Oh, what was it?
A lot of scientists bring their own page.
Is it? Well,
So, Tyler, I asked someone who works for SOI. They said
well, it
they're
looks
not
like we
sure.
have a very
Normally,
educated
scientists bring their own.
audience. That is correct.
Katie, what was the answer to the pH meter?
The question was they are not sure. Normally, scientists bring their own and test it when it's been surfaced. Which we are bringing water samples back to our lab, so that's something we could potentially do.
Oh, look at it swimming. We're moving up the same line. This line had the same, the data we were getting from this following the last night's mapping. Basically, this is a line we think might also be possible sites for seeps. So we've marked the ones that we've already passed. We're looking to see if we can find any larger ones.
And do the grab to the left, to the right.
To the right.
We just passed it.
Okay. There's something over here on the right? I didn't see anything. But
Slightly backwards.
okay. Can we try moving a little backwards?
Right there.
Oh.
Most likely a pycnogonid, a marine spider.
Oh, cool. Do we want to?
We don't want it, right?
Alright.
Let's get the pycnogonid with a sac. Well, I don't know if the suction is working.
It has squat inside.
I think the suction sucker will not work with something that big.
No.
This is a very slow moving marine spider. So.
Do you want to put it in the bio
Yes,
box?
please.
Okay. Yeah, we'd like to request a little pickup.
So I think this is maximum speed for the pycnogonid.
We want to get a human pick before we pick it up?
Yes.
The sea spider gets its close up.
I think you might appreciate the alien aspect of the walking for the video since maybe. And also I like
Absolutely gorgeous.
That's is a very nice specimen.
With a white amphipod on its right side.
Hitchhiker.
All right, we're going to grab that into BioBox and then we're going to move on.
Does it eat? Yeah.
Yeah, that's fine.
Oh, wow.
He's faster than we.
Yeah, I thought he was pretty slow. He is not slow.
That is correct. The stomach is in the legs of the pycnogonid.
He must have heard us say he was slow, so he's like, watch this.
All right, we're taking a brief stop here because we're going to get this Pictogonid spee spider while we climb up this ridge to look for more evidence of methane seeps. Yeah.
Can we detach Amber's headset and give it to Miguel? He's logging and doesn't have the audio. Miguel needs it to log.
You can see a couple of these purple stars next to it.
But Nick also needs it because he's answering questions. That's okay. Oh, and are you okay switching spots with him? That would just be easier. Okay. We're getting it figured out.
So the pycnogonid has two amphipod hitchhikers.
It's okay. They flipped seats.
Are you sure? Because, like, technically, if that's like your system, I want to make sure. If it has your mind.
Oh,
If it's easy to switch,
all right.
I can just do that. But if it's complicated, I can just sit here.
If it's easy for all of you, then it's okay.
Working this thing.
I can move the keyboard.
Wherever you think it'll go.
BioBox 1's okay?
BioBox 1 should be fine. It's empty, right? Miguel, the BioBox is our empty still, right?
Yes, we only have things in BioBox 3D.
Okay. So
This guy shouldn't be able to escape easily, so...
Let me move over to give you space.
there's nothing in 3D, right, Miguel?
We have many things in 3D.
We have many things in
We have many things. As long as you don't care about the pycnogonid eating the crinoids and the sponges, it should fit.
He's not going to eat the crinoids and the sponges.
Try 3B or 3A, doesn't matter.
3A.
Do you have any space?
one of the reasons we stopped for this one is it was on the move and we didn't want to lose it. Okay. All right. Uh-oh.
I have been collecting smaller pycnogonids from the Caribbean and Bermuda, and I will agree with what one of the what Kevin says. They lose the legs so easily. This one seems to be pretty sturdy, but it's very common for at least smaller pycnogonids that I know of. You may not get the intact specimen.
That's amazing. All right. Thank you guys so much. We're totally ready to move forward. Thank you.
Yeah, it might be good to try to see the end of the ridge. There's something interesting.
We're gonna try to bring the suction on again to see if that crab that was there is finally ready to move down the tube a little bit. Before we continue on, we're trying to clear an animal that's been holding on to the suction unit here. All right, and we have liftoff again. We're moving forward up our ledge. So we've actually already identified a couple of spots of methane seeps. So we will be continuing on with some collections tonight. Right now we're trying to follow this particular line and see if there's evidence of more active or newer seeps. Okay, we're going to take a look at some of these lower area here now in between think, are you guys looking at over this way?
Yeah.
Yeah, that sounds good. You can see just how dark it actually is here. So which, on the map, which way would we be headed?
It's that one inside the slot.
Okay. So we're still continuing our path. All right.
There's a question here that I think the pilots can help us. Somebody's asking about the accuracy of the multi-beam around this depth. Is it 50 meters, 25 meters? Do we know?
Josh can answer that.
There's a question about the multi-beam.
The accuracy of the moment. Sure. Wait, I mean, it's 15. There's a calculation. It's
An anemone?
Yes.
Yeah.
That's correct.
Yeah, we're just keep moving forward. Right here.
Followed by another blue sea animal.
That's a fish, not a shell. So yeah, we're just moving forward.
Rat-tailed fish.
There's some shells there, but it's not a significant amount. So I'd keep going forward. Yeah, we're good.
Is it better now?
Yeah. So we haven't seen, for a little bit now, we haven't seen any real strong evidence of any additional seeps. But the same data that led us to those earlier ones are basically suggesting there could be some here. So we're going to continue up and keep looking. I'll stick by the line.
Just a minute. But the default answer is
We just want to continue up along this line.
sí, that's probably I would say number
I'm just confirming with one of the geologists on the team.
Pelagic number
I think Miguel wants to know when we're going to be turning around to core. But we really want to give this area a little bit more evaluation.
Yeah. We might find something amazing.
We might.
A huge methane seep.
I mean, we really, after had been marching around for several hours and before we found the seep. There's a nice outcropping here.
Yeah, and all these rock outcrops have been by far some of the most, I don't know if by the birds, but interesting parts we've ever seen as of now.
Yeah, for the nemertine enthusiasts, I would love to collect that nemertine, but we're on a mission right now. That seems to be like a very nice species to study. Nemertines is a phylum of predatory worms, mostly present in the benthos, but there are a few species in the water column.
Nice. So it's been kind of a very interesting expedition for us. This is marine biologists, geologists, microbiologists, astrobiologists, and we all find ourselves interested in these little spots in the ocean of biodiversity.
Zoom into that white fluff. It's a sponge, obviously.
Picture. Overall, we've moved quite a bit of distance today. The first couple of days, we only moved a few hundred meters. We've moved kilometers today in the search of these seeps, and it paid off.
Andrew, those green spots that I see, those green blobs. Just zoom in a little bit, just to get an idea.
These guys,
nice. Spots. I see the tunnels. I see the tunnels, so they're sponges.
Okay, sponges.
There's a. Can you zoom in a little more on that plane that you have? Right there.
No.
That's okay. Thank you.
Yeah.
Thank you.
Thanks.
Ideally, you want that as a place.
Alright. Yeah, we can.
So if you see any channel-like structures, immediately that's a sponge. There's nothing else like this. It's kind of it doesn't look like a bacteria shape. And usually there's a hole, the osculum, where the water is coming out.
Oh.
Wow. To the left side, it's scrolling down. It looks like it's moving like a crab.
Yeah, it's a crab.
It looks like a crab.
It looks like it has some fun stuff on it. Like an amazing back then.
Not seeing really shells. I'm not seeing any biofilm really. Can you zoom in on that? Right there. It looks kind of.
Just below, you have a crab walking with some interesting anemones on it. Just on the right side of the.
Okay, no, I don't.
It's a hemicrab full of sea anemones.
Huh.
The association is well known, but it doesn't mean that this is not an interesting.
Look at it go.
Wow.
It's a beautiful pink.
That's cool. That's definitely good. That's definitely photo worthy.
Wow.
Cool. Yeah, I thought there was something more here. This is not that. This isn't of any interest to me. Is the film here? This isn't a biofilm. It's just a material. Cool. All right, we're good to move on. Thanks.
have a nice photo of this?
Yeah.
Oh, excellent, excellent.
Just left over.
He was waiting for the picture to leave.
It really did look like he was pausing for a photo. So.
Let me ask the audience something. Have you heard the term pom-pom crabs? And also see a comment on the brachiopods.
I think
I've been
Guillermo
watching
asked
them.
to catch the crab. He says it's probably a new species. I don't know if we want to go back or not. Just relaying information.
I'm actively looking for brachiopods. Yesterday was an amazing day, but I guess I was too concentrated on the hemicrab and I didn't pay attention. Well, if you see something, say something, especially when it comes to brachiopods, okay? Because my eyes are all over the screen. Thank you, I appreciate this. It was a hemicrab with a snail living in a snail cell, and the anemones were growing on the cell.
And we've got about 15 minutes more that we're going to be pushing up in this direction, adding a little bit of time for some of the stops we've made, and then we'll begin the turnaround. But we really want to give this an opportunity to
give it a shot for that crab. We have a request from the museum people. Sorry about that.
Yeah, I apologize. My WhatsApp for our scientist group chat is not working so great.
Saludos, Cristóbal. Again, I would like to emphasize that the majority of the collections we're doing, especially when it comes to bigger animals, is going. It will be. This is for the people of Chile. It will be going to a museum. There will be the specimens will be studied, curated, named, and being on display for the people of Chile. I would say all the big animals we're collecting.
Still just moving up, right?
Yeah. Can you zoom in on some of this?
Okay. That or? Okay. Okay. Whoa.
I was just asking for the same thing. Hey,
Yeah.
buddy.
You're interested in the bandings?
Yeah. Well,
Yeah.
She has a friend.
On a show for us.
Nothing unusual. Just a few sea cucumbers dancing around. Huh?
Oh,
Right.
Being that mud? A microbial mud?
It could be.
Yeah, it could be.
It could be a mat. How big is that rock actually? That's a big rock. So,
Maybe you can grab some of the smaller ones.
yeah,
The one in the foreground is pretty big.
yeah. What about like, would that one be?
The one behind it.
Or that one?
The one on the side. I think so, yeah.
Okay. You want to try? You want to go for it?
Yeah, why not?
Okay. We have a rock crab. Oh, what about that one? That's a pretty small, decent size.
Yeah. I'm interested in that banding, some of the growth on those.
The banding's the big rock. The small one, you want the small one or the little bit of banding?
So that's got a little bit of the banding there. That one's got a good amount.
Yeah, the big one's got quite a bit, but there's some banding on some of the smaller ones too.
Excuse me. That is very interesting. Cool. And they're discussing the grab.
Is that okay with you, this one?
Yeah, that would work.
Mhm.
That would work.
Yeah, that looks good.
Good question in the chat.
Good coloration or
There's a good question. Yeah, good question in the chat about coloration.
Does the coloration of these creatures signify danger to other animals or just a byproduct of what they eat that gives them color? I don't think that there's a universal answer to that. I'm not a marine biologist, though I'm a microbiologist, so.
Nick just logged out,
perhaps
left.
to weigh in on that, or Guillermo.
Oh, look at that.
That seems sturdy
Nice.
enough, so maybe we can place it in the rock box.
Yeah. Is that will that fit in the rock box?
Yeah.
Is it-
Will the box hold?
Do we want to try to take a smaller
Do we need
one?
to take
Is
a
that-
smaller rock?
Yeah. Is that pushing it to the limit a little bit?
Do you want it? How bad do you want it? How bad do you want it?
I mean, if there's the smaller one, we'll take a smaller one.
Yeah, maybe take a smaller one. I'm a little worried about that.
Yeah.
I need to breathe.
Yeah, there was a smaller one down a little bit.
Slowly up the rock. The animals were doing good bonus. Yeah, maybe something wrong.
Yeah. So like maybe that, would that break off? I don't know. It didn't seem like these rocks were very,
They're not as brittle as the ones
yeah,
further
they're not
down.
very brittle.
Okay.
This one? Yeah.
Oh, sorry.
That was a very good rock. You cannot always get everything. The bestest rock? I'm not so sure.
It came with some friends. 3
Yeah.
friends.
Not enough friends. anything below 6 phones so far.
I just filled up a cord. You have a marine biologist here.
It's in human nature to look for the most best as a rock.
Yeah.
You love our rocks?
That's what makes us human.
So, did it break apart or is it just covered in sediment?
No, I think it's just
It's covered in sediment. The sediment here is so fluffy.
I think we just
It just sunk into there.
Yeah.
They're just joining us. We're trying to get this interesting banded colored rock out of some sediment that we saw. We think it might have some interesting microbes. So this area's got a number of them.
Can you zoom out a little bit?
I mean, is that too big?
That might be too big.
Might be too big. We're getting organized.
Oh, yeah.
You get a friend for free.
How about it? Free friend. Question, are there significant amounts of carotenoids in the microbes? I mean, there are definitely microbes in the carotenoids. I don't know how prevalent they are in the deep sea though, off the top of my head. Microbes make pigments for lots of different reasons though.
Yeah. Well, and carotenoids can be for stress,
Yep.
but a lot of times it's oxidative stress. So.
Yeah, carotenoids are correlated to oxidative stress. So certainly in a more anoxic environment, maybe.
Mm-hmm. I'm
having a difficult time judging the size of these.
That one? Is that? Those are about the same. So,
Okay.
want to grab one? One's got brittle star.
Yeah. Grab the one with the brittle star.
This one right
That
here.
one looks a little smaller. No, the one.
This one right here, I
yeah.
think. Yeah. That one, it's got better banding.
Better banding.
Better.
A little something
As
for the
my
geologist, a little something for the biologist.
as my English skills break down, better even than. Oof.
Or the one next to it's even smaller, but I don't know.
That's
This one's good.
Yeah, seeing the question about photoreceptor cells of the predators, there's still a lot of interesting questions about what organisms in the ocean, what wavelengths of light they actually see. With very simple mutations, actually, you can change rod and cone cells to perceive different wavelengths of light. And for Miguel's notes, that's going into the rock box.
Rock box 6 is ready to go.
Do we want to grab a second while we're here? Geologists? Do we want a banded rock? Oscar says nay. So we are ready to move on. Thank you.
The geologists yearn for the methane seep.
We all yearn for the methane seep.
So we said we were going to turn back in about an hour. We stopped and do a little bit of collecting. What do you want to call it?
I would. I'd like to proceed to the next waypoint.
All right, next waypoint.
Yeah.
So yeah,
That one.
New waypoint, right? That's the new waypoint?
Yeah,
New
the new
waypoint's
one.
up there. All right, the 0, 1, 2, 2. Right.
And I recommend we hold off on any more sampling for just a little bit. Until we get further up.
Yeah.
Yeah. Yep.
Andrew.
Just get to that spot.
Let's move as fast as we can.
Okay. As fast as you'd like.
Minus lap of beats.
Yeah. We're scanning. Right, so we're going to continue on to our next waypoint. If you are just joining us, we are in the middle of a 20-plus hour dive. We started at around 7 o'clock this morning when we touched bottom, and have been following a pretty rapid move. We've moved several kilometers at this point, looking along this particular line that was selected in consultation with the team geologists being possible sites of methane seeps. We successfully identified one around 6 o'clock Eastern Standard Time this evening. Now we're beginning to move along this line a little bit to see if we can identify any additional ones before we turn around and go back and sample those sites more extensively. And this will help us make some decisions about where we want to go tomorrow. And everyone in here is actively scanning the monitors, looking for any additional evidence. So we're moving at about 0.6 meters per second. So we're kind of bucking it as we're moving through this area. So we're So some of our grad students who've been working on the back end of the project for a while now, the processing water samples and some of the sediment samples. So how do you guys feel about finding the seeps earlier today?
I'm really excited about finding the seeps. I mean, that's one of the main reasons we were searching today. So
Miguel, are you excited about the seeps?
Yeah, I'm really excited about the seeps. As a microbiologist, and finding seeps being a decent part of what I came for, although not all of it. I'm really excited about it, and to look into those microbial communities together with Lauren later on in the project. That will be amazing.
Does Amanda have headphones on anymore?
No.
Do you want to tell everybody what you think about finding the seeps?
Are you excited about finding the seeps?
How do you feel about it?
Then talk to the microphone about it.
Well, that's very exciting because we've been looking around for a few days, and yeah, and I think the area is not well mapped, so it's really hard to have any indication of where to go. So we've been, as Lauren said, walking with a flashlight, just hoping to find one. Of course, we have reference data, but yeah, it's very exciting and lucky that we came across this area, and hope we find more interesting sediments, water, creatures. Yeah.
Yeah.
there was something down to the right.
Yeah.
Right.
This area here. It looks like it's more snow. More biofilm potentially. Yeah. That could be mad. It's definitely not as much as we saw at some of the other sites. Could we possibly zoom in on it a little bit?
Welcome back to the watch party. Yes, it's definitely, you didn't miss much. We still have a few hours to go, so enjoy.
To answer the question in Twitch chat, we are in the Pacific Ocean off the coast of Antofagasta, Chile.
Yeah, this is. I mean, I think it's matte, but I mean it's pretty small compared to the other ones. Okay.
It definitely log as an observation.
Definitely.
I'm parentheses in that.
Yeah, who's logging right now?
Miguel's
Miguel.
Can you just log this?
It's already logged.
Oh, okay.
Yeah.
It was photos.
Okay. Yeah, I think we're good. We want to move on. Yeah, we're getting pretty close to that waypoint. So, there's little evidences here and there. Yeah.
Let's keep moving.
Hold on, we're going to have a brief conversation about what's happening next. Oh, wow. Okay, so look all along here. Look at that, guys. Have we seen that? this whole area. I think that's potential mat and shell. Yeah, because look right here. That looks like biofilm. It's like arrayed on the surface, like going down. Do we want to maybe get a zoom in?
Can we have a yes? We have a
Yeah, on
yeah,
this
can we
this
zoom in
area
on that?
here, please.
We're just gonna come forward.
Yeah. Kind of impossible to sample.
Wow, that's
This is really white. That's really. There's some different coloring in there too. Is it blue for real, or is that a function of the like filter?
Can we get a piece of rock here?
yeah. Take a piece of rock here.
Yeah. Okay. We would like to take a piece of rock. Perna, you want to point out what you're looking for?
Do you want to scoop of the sediment?
Yeah, let's take a scoop of this.
Okay. We just want to do a big sediment scoop. Just
Yes,
is
yes.
that gonna be loose enough? Well, that looks probably
But we remain and
Yeah,
then we'll
let
put
me
that
share.
okay. All right.
Hannah, do you want your own separate scoop or do you mind sharing? Because we can take 2 scoops. They can go
Like raisins.
Perché necesito una cantidad de muestra, pero ¿cuánto te lo hace? Sí,
One scoop.
one, 2 scoop the maximum amount, but 2 scoops. But if you don't mix with other samples, then if possible.
Okay.
Okay. I'm gonna try to do 2 scoops, focusing, I guess, on this particular part. We're collecting this particular area of some loose rock and sediment because there seems to be a biofilm, bacterial mat type material suspended here. But it's different than the ones we were looking at before, which had more of a cobweb appearance.
Can we take something like continental so that it doesn't get full of seeds?
It gets pretty, I think it's going to be kind of a good pull up, so I think we'll be fine. So we're trying to keep the surface layer as intact as possible for this. So I don't know if we can take the box. This is okay. Well, it's a sediment scoop. I didn't think of it as a rock. Can okay.
Where do we plan on pouring the scoops? On a coral quiver or?
Yes, the plan is to move the sediment into a coral quiver.
Okay, so then maybe we can just take 2 scoops into the same coral quiver and everyone can share them.
2 scoops in one coral.
That might make more
Will
sense.
that fit?
That's maybe it's better we take the maximum amount we can take
Okay.
from there, including the rocks.
Okay. So we want to try and get as full of a scoop as possible.
Where's it going?
Coral quiver 2.
Did you want to do bio box 1A or 1C? Probably more.
Are the coral quiver bad? Just for
We can do this. Is just yeah. You might need to go outside. So that's a screen scoop, right? You guys know that.
Yeah, it's a screen scoop, so
Do you want to sediment bag?
sediment bag will
Catch everything and then we turn it over ourselves in a bottle box.
Is that a possibility? Yeah, because yes, absolutely. Because otherwise, I was kind of looking at that and I was like, oh, worried. That's amazing. Right, that's what I'm trying to do the bag, because otherwise when they pull it up like that, it's just going to come right off. So this way they'll always be able to. Pilots are breaking out the sediment bag to try and move some of this more gentle sediment into it so we can collect it without disrupting this different biofilm on top. So this is a different mat than the one we've been seeing around other areas of the seep. So you want to try and get this area here. Hold my breath here for a second. I mean, there's plenty of it around, and we're there. And reposition.
Very cool.
Out of that color here, though, so the coloring is still there. So probably get the microbes. Excuse me. Burped.
That's a good score.
Yay.
I'm not going to make you guys do that again.
We want to
just go after.
We want to dump into one of the bio boxes.
That's a bean.
Whatever's
fold it over so.
Yeah, well, yes.
Yeah, I could just do that.
Okay.
Is that enough? Do you want another? Yeah, like we can do another pass and like really fill it up.
That should be enough, I think.
Whatever Aparna needs, and then we can just.
But I will be sharing with
Maybe another pass is fine.
Alright.
Ana, I think you have enough tools, chiquitos.
Is it
Look at
really?
it, look at it.
What is the size? What is the size of that?
Mira, la pinza de la... Mira, bájame. La pinza de la...
Ah, no, es suficiente, yo creo. No. It's fine, I think. I think it's fine.
Just in case.
You're only about this much in the bottom.
Mhm.
It's just, if you want, we can give you another scoop so you'll have about that much in the one.
I'd say since we're stopped,
We're
go
here
for it.
anyway.
Okay, we're already here.
Yeah, we're here and they're right there because also remember your blue layer is probably only like that. So. Awesome.
Now it's yes.
Yeah.
Yes.
Miguel, what's a good bio box for this?
I understood we were just wrapping it on itself. Can we just leave it in box 2, or should we put it in a bio box?
I think you said they turned it, in this case, you turn it upside down into a bio box.
Okay, it's pretty big, so we could try 2A, I guess.
Okay.
If they think that it fits in one of the bio box 1, in one of the...
No, it has to be...
No, it's not.
Okay.
Okay, then 2A.
We'll put in 2A, please. Amazing. And just confirming we're done at this spot?
No.
We're not done at this spot? We are done. Okay. We're good. We're ready to move on. We're pretty near that that waypoint. So thank y'all so much. So I think when we're thinking about one of the big discoveries or conclusions from the day, other than the fact that we actually found the active seep, it's that the methodology that we were using as a team, the collective we, in selecting the sites actually does seem to hold up. So this can help us on future days. Yeah. It seems to be. I think so, yeah.
Normally the sack will just fold on top of it and it stays in there.
Yeah, because it's the handle's weighted too, so it's gonna.
Yeah. So the other bag of tricks for you is, even though we've got that crowd and the sample boat, we've also So suck back to your mat, it'll pass past that. That's a good sign.
All right. We want to move on to that last waypoint. Okay, we are at the end of the waypoint. So,
We're almost to the point where we're trying to reach, I think.
Yeah.
oh, we're at it, yeah.
Yeah, I want to get there, and then I would like to. Whoop.
Take it.
I'd like to go along the bottom of this to get back around to where we were.
So that's this one.
If we went down along the base.
Here.
Yeah.
And we're trying to get up here.
Yeah, that's up. That's fine.
Yeah.
Yeah, just
So
move to the west.
everyone's come back around to this to sample, and then.
Yeah, so the distance here is about, okay. Yeah, it's about 8 meters.
Okay. I would like to give everybody the opportunity to take the samples that they want from that spot, and then we'll just keep searching around for the rest of the dive time.
So you want to go up here and then back down and then continue on?
Yeah. So where are we right now?
We're here.
Right here.
Yeah.
That's our old location,
yeah.
right?
Yeah. So the sheep was like back here, right? Yeah.
Yeah, but just keep in mind that this is not scaled properly.
Right.
So, and it's kind of, yeah. So if you wanted to get these samples from worms for some time, in my opinion, it would be probably better to go
Faster,
back
just go
down.
straight back.
And then while
And
we're
then
down
we'll
here,
go.
get your samples, and then we know we came off this line, and then on the way up, let's go over like 50 meters, and then you're actually scanning a bigger area for more seeps.
Sounds like a good
Because
plan to me.
if we go up here, now we're gonna move all the way up here, and then we're
Oh
gonna double
yeah,
back
you're
on
right.
ourselves. So
You're right. Okay.
that work?
Sounds good to me.
That sounds good.
Let's do it.
Yeah.
And in the meantime, we'll take a new route back down to here, so we're also scanning that area, so now you get.
Cool. All right.
Awesome.
I really like it.
So for everybody who didn't hear, we're going to be navigating back to the higher density site of the seeps that we originally found back about 3 hours ago. We're going to be taking an alternate route back there, so we'll still be scanning for new seeps. We'll collect our samples kind of for the evening and fill up, and then we'll kind of move to finish up exploring some new locations for the remainder of the dive time, which will still be, by the time all that sample collecting done, I imagine will still be about 4 or 5 hours, which means we'll still get a good amount of exploration done, and then we can always come back and look for fill up on some of those sites tomorrow.
I wanted to answer a question. in the chat.
Yeah, go for it.
So there was a question about the methodology. So this is based on pretty standard methodology for searching for seeps. So we looked through the literature on different methods of using bathymetry data, backscatter, subsurface profiling. There are more accurate methods of doing this, but we weren't equipped to do that on the ship. So we used what we had available to us, and then the rest was just following the lines that looked promising until we spotted something, which took quite a bit of time. We were ready to log 20 hours on this dive until we found something, and we found it in about 11 hours.
Yeah.
Yeah, and then looking for 6 more hours the other day. So realistically, we found things pretty fast because it can take days to find features like this, even when you have some data to go off of.
What's up, Jess?
Alright, so are we heading next week? In the interest of time, are we heading back?
From right here, the ship is not moving on this way. From right here, but remember, this is not an accurate representation, so right here it stays the same. So like more or less like that is the size of the ship. So I'll go to this one, which is south, kind of grabbing the other one. We're gonna go straight up. Okay, so let's compare the waypoints. These are 100 meter squares. That's typical. So, this one. If you grab, I'm gonna put a mark in there. When you grab the marker, you show this the last time.
Correction. The longer we spend doing this, the longer it's going to take us to get back to
Right, I know, it's just, the area has probabilities that are likely below where we are, so if we can go along that line to get back to the sea, then
Yes.
cover
I'm fine.
two different objectives at once.
Right.
On the same amount of time, we're just trying to figure out where to position Falco so that we don't get all Right, and I don't want to keep them up super late, they want to get their samples.
They're not getting
Well, yeah, but I mean.
I know,
don't want
they
to
went
be
to sludge.
Right, right. Yeah, no, we're gonna get them, I just want to make sure it makes more. Well, yeah, no, but if the area of high probability is on the way back.
It's on the
to
way
sea,
back,
we
right,
can
so-
just cover it on the way back to the sea. So, and then we can explore more after that.
Okay.
We've been having a bit of a discussion because we're trying to figure out what's the best way back to the original seeps and the new route we're going to plan after talking with Lauren and Jess, the pilots, is a trajectory that's going to actually take us past some high probability sites. We were just discussing how long it would take to go one way or the other. We're gonna go fast?
Yeah.
Okay, we can go fast.
Zoom!
Sebastian has the zoomies.
We're moving back to our original seeps under a slightly different path, so we have more of an opportunity to look.
I do it every time too.
So we got some potential here. Are we moving forward through this? Is that on our way? Yeah, there's not high density biology, native films, yeah.
The geologists are suggesting to inspect this.
Okay. The geologists would like us.
The geologists would like to see. I don't know if they want those.
Oscar? Oscar or?
It's not in here. That was Javier.
Both Javier and Antonio.
Okay, so Javier and Antonio.
We have a double to suggest.
You were asking were they looking for cores?
Yes.
Javier needs to take a sample of this. Maybe we could just like grab some and throw it in a bowl flavor.
Okay.
If it's carbonated, it'll break off pretty easily, I think.
Yeah. Yeah. That sounds like a plan.
And then we can just shoot straight back.
Coral quiver.
Which one are we on?
Two.
Which one?
Two.
So we're going to go for a coral quiver 2 if we can grab a piece of rock.
This one.
If it'll fit, yeah.
You want to grab a rock? The white fish. And just a small one to get in the quiver? Later you do 1 second.
Yeah, yeah.
So they want a piece of the white material, which is what we're doing already. So
Okay. The white material, I'm assuming is the. Yeah. A small piece of it. So it'll break off.
He's saying maybe more to the right or maybe better.
How about this? The part that zoomed in work? Is that
Let me ask.
We're in for confirmation here real quick.
I'm sending a message. Give me one second.
We're waiting on a geology confirmation quickly. Or not that quickly.
Javier says yes.
Yes. There we go. if at all. Little crack.
Is there a sponge in quiver 2?
No, they opened quiver 1 by mistake, so they closed it already.
Okay.
So just please.
Yeah. Oh yeah. Oh. Oh, yeah, it was like a rock.
It looked like it came with some friends too.
But what we were saying, I don't know if you were able to see, there is like a... Sí, pero miremos que pueden ser carbonatos. Todas esas estructuras que se veían. Did you see that there were like white layer going around? And I think we took a layer that was before that.
Okay, yeah. Director, because this is important.
What's up?
So maybe let her finish your stuff. So you see these lines that are quite white. And here I don't know if it's possible, but to the right there were no prominent, so I don't know.
You want to try and get a rock from over there?
Yeah. Okay. So in here it's probably the same thing, but you see that it's like kind of
Okay.
more white and linear feature.
Okay.
How do you? I don't know if it's possible to. That's why I'm not sure. Just we can try it, and if not, we can keep going.
Okay. So for everyone at home, what's the difference between these two?
We don't know exactly, but we were looking for carbonates because it's what happens when you have methane mixed with the water of the sea. So before now, we were not able to check on any.
Antonio, that's the end over there, maybe.
Which one?
Is that the same spot?
I'm not sure from this distance. I think maybe not, because you see in here, I think this is the. So I don't know if maybe someplace or maybe here. I don't know, because it's like.
Can you go?
Can he just, he'll break it, then he'll pick it up?
Yeah, it could be just a little piece. We don't need.
Antonio.
You just took a couple centimeters. Okay.
Yes. Antonio, remember what to know? Yes, it could be to the same. It could go to the same place. It doesn't matter.
Okay.
Is he gonna try? He's just gonna break a small piece.
Yes, just little piece.
So this can give us information about differences.
I mean, the way they look, they could be some fingerprint of previous seeps, you know?
Very cool.
You see it's quite more white.
Oh yeah! Wow.
than
Okay.
The other rock.
You see it?
That's very, very clearly. Yeah.
So,
Is that sufficient?
From far away, it looks quite similar because of how the surface weather.
The sediment and
And the
everything
sediment
else. Yeah.
and everything on
okay.
top, but from the distance you can see the. Yes, that's one.
There's some more down there.
If you can take it, yeah. All can go to the same place. Yeah.
So it could.
He's gonna put it there.
I think. Well, quiver 3 is
It
already.
can go
open
to
now.
3.
Yeah. So quiver 3 is fine. Awesome.
You want it in two?
Would you prefer it in 2? So it's all in one place?
It's okay if everything goes to the same place. We will be able to
Yeah, but-
identify them.
We already have coral quivers.
But it's,
Three
he already
open.
has 3 open.
Yeah, let's do
Yeah,
let's
that
do
one- that
that.
one's okay.
Vamos a
Tres nomás para seguir rápido.
To answer the YouTube chat, we are using the hashtag ancient Atacama. Sorry, Atacama Trench. Atacama Trench Ancients.
Andrew,
Yeah, I know.
Andrew,
I can't.
after this, we don't stop for nothing.
I know. I'm,
Thank you for reposting it.
That's a lot.
sideways and just wedge in the shell. So, but when you start, like, if you find like something really smelly, it'll be hot. Other stuff can hot. So they want to like suck that to your match. Okay.
Thank you.
So we're going to drop the last of this rock in Coral Quiver 3, and then we are moving back to the original Seep sites full speed. So we're going to try and get the remaining sampling for tonight done during that period.
To answer the question, is the white methane ice? No, that is carbonates. One of our geologists also let me know it's not possible at this depth for that to be white methane ice. So we believe it's most likely carbonates.
Okay, so regarding the question in the chat, no, CO2 is not solid at this depth, and it is actually dissolved in the water. And I think you may be talking about methane, which is CH4, and at these depths it's also not likely to occur. The clutter is methane ices.
Awesome. Cool. Right. So when you guys are ready, we're ready to just head back to the Seep site.
Okay. That's where we're going next.
Yep. In full speed.
Full speed ahead, please. Unless we find another seat.
So as we head back to the Seep site, I am actually going to step out now and take a break myself as we get ready to pick our samples, and I'm going to be turning things over to Antonio. This has been Drew Palmer from the Florida Institute of Technology. Thanks for hanging out with us for a couple hours, and I hope you'll stay with us. We're hopefully going to be continuing on till 4 a.m. Looking for more Seep sites after we grab some samples. All right.
Please let me know if you think Dr. Andrew Palmer has a very good voice for ASMR purposes.
Yes, he does.
He's got the right tone.
Hello everyone, I'm Antonio Molina again, narrating now from here, the local time. We are at 10 p.m. and we are going to be keep going until midnight. Here in the room there are lots of people. We have the pilots, we have the person in the crew that is taking care on the multimedia and all the streaming and everything else. And also we have some of the students. We have Miguel, Amber, we have Nicolas, and we have the two PA, Armando and Lauren. And now we are heading back to, if you have been following this for some hours now, we are heading back to the first Seep that we found that was active. We were transversing along this tectonic feature that we found that may be related to this first Seep that we found, but most of it seems to be already dead. This kind of Seeps doesn't last forever. They are active for just a few years. Could be hundreds of years, but when the methane stops, all the life that is feeding on those materials just start fading away, and that's why we are going back to the first one. And just in case maybe we will find another active Seep, we will stop, but otherwise the plan is to keep going and keep searching along this feature. And Nick, and you also told me that in here you can find very interesting fauna, right? That is related to those Seeps.
Yeah, absolutely. Yes, these are organisms that are adapted to this different chemical environment. Usually, these organisms are able to chemosynthesize, and they're usually in very close association with microbial communities. As an example, let's say the model or the poster child of ants, the polychaete family Seepage, they have lost the digestive system, and they rely completely on the microbial community to get energy for different sources of energy for their metabolism. it's definitely a special community. And up until a few decades ago, we didn't even know that chemosynthesis exists. We only knew that life can happen with the help of photosynthesis. So that was one of the major breakthroughs in biology.
Someone asked if we found any wrecks by accident. At least on the days that we've been diving on this Atacama Trench ancient expedition, we didn't find any, but it looks like we were in a treasure hunting for looking for these seeps, and we were lucky that today we were able to find some of those. Only one active by the moment, and a few others that are relics of past activity. Beside all the life, we can find also some trash, anthropogenic material, because it's very sad to see that even in this deep we found this trash laying around. Yeah. So one of the indicators of the seeps were actually some of those specimens. For example, these bivalvi, right, Nick?
Yeah, that's right. Yes.
Those feed on the methane in the seeps, and they only can live with feeding from them. So when you see them, it's one of the signs that there are seeps. But also we were able to see some microbial mats that also are usually found along these seeps of methane. We call them cold seeps because even though they are not actually really cold, but they are not as hot as the hydrothermal areas that we can find in other places. And for example, in here, I don't expect to be there more than few degrees more than the surroundings. The reason that I think we are seeing that much trash is not only because we are close to the coast, but also because we are transecting a long distance in a very fast fashion. So as we see more terrain, the more likely is that we found something. It's the same for finding new life or finding specimens, but also for when you are looking for trash. We are going following the bottom of this relief, and we can see some interesting geological features. It seems that there are some layering of them, and we just passed by this white layer material that could be actually carbonates. Carbonates form when the methane are in contact with the seawater, and this material precipitate, but also related to these seeps we can find sulfates.
Could we zoom in on a rock? Just
yes.
Scan the rock.
Which one can you tell me? Do you want to see over here?
The front side. No, just the front side.
In here?
The whole front side, yes.
So can we zoom in this area, please? So what is interesting in here? This small.
We're still actively looking for brachiopods and chiropods. Let's go. We just have to look.
Okay, is that okay, Nick?
Yes.
Okay, so we can keep going. What are those base-like creatures with white in color?
That's a hexactinellid sponge.
Mm-hmm.
Very common in the hexactinellids is one of the sponges are divided into the calcarea, demosponges, hexactinellids, and homoscleromorpha. The sponges that you are more likely to find in deeper waters than 100 meters are those vase sponges. And I mean they're beautiful organisms. Okay, so this is what you expect to find in deeper areas.
Mm-hmm.
Not a lot of species, but amazing seeps.
And why we are finding them in much shallower areas? Do you think?
Perhaps they have been outcompeted from the much more successful demosponges. Demosponges are the sponges that everybody can see while snorkeling and diving. If you have an original a real sponge for the bath, for example, that's a demosponge.
Mm-hmm.
Just to give you an example.
So as I was saying, we keep going through the lowest part of this relief. We still can find some of those crabs, these lobster crabs, white in color, and other organisms that, while we were looking for these seeps, seems to be some of the indicators of them. In here, it doesn't seem to be much more of that when we were in other places. But what is very indicative of those, as I said, is these bivalves that live through feeding from these methane seeps. Voy a comentar un poco en español. No sé si habrá algún hispanohablante en la audiencia actualmente, teniendo en cuenta que es un poco tarde, aparte de Miguel. Miguel, ¿qué tal estás?
Pues aquí estamos muy bien. No sé si nos quieres hacer un resumen de lo que llevamos haciendo este rato, qué vamos a hacer. Entiendo que lo vas a hacer en cualquier caso, pero bueno, me pongo yo mi granito de molesta.
Sí, efectivamente, para aquellos hispanohablantes que tengamos en la audiencia, hemos recorrido durante todo el día de hoy, durante bastantes horas, no sé ya cuántos llegaremos, buscando por estas emanaciones de metano que son muy difíciles de encontrar porque no se encuentran asociadas a formaciones muy evidentes ni a unas señales evidentes tampoco en el radar. Así que hemos ido buscando durante este transecto a lo largo de las estructuras tectónicas que encontramos en la costa de Chile, y finalmente hemos sido afortunados por encontrar uno de estas emanaciones. La primera de ellas ha sido una que parecía activa, ya que una de las maneras de identificarlas es por la presencia de una especie de mejillones blancos que se alimentan precisamente de estas emanaciones de metano. Y por otro lado también hemos encontrado esos tapetes microbianos que también son característicos de esas áreas, y que finalmente han sido estos dos indicios con lo que nos ha llevado a la conclusión de que efectivamente estábamos viendo esas emanaciones de metano, ya que no hemos sido capaces de ver burbujas, que es otro de los elementos que suele caracterizar estos seeps, pero en este caso la cantidad de metano no es lo suficientemente grande posiblemente para formar burbujas visibles, a pesar de que estas noches hemos estado haciendo el bubble hunting o la búsqueda de burbujas. Pues obviamente al no existir en este tipo de seeps, pues no íbamos a encontrarlas. Hemos seguido a lo largo del área en el que nos encontramos esta fractura que parece que sea a la que se asociaba esa primera emanación de metano, y hemos encontrado otros puntos que en efecto parecían haber sido en el pasado emanaciones de metano, pero que ya a día de hoy no eran activas. Eso lo sabemos pues porque esos bivalvos, por ejemplo, de los que hablaba, pues ya habían muerto y solamente encontrábamos sus conchas, y tampoco había tanta presencia de tapetes microbianos parecidos a algunos que hemos visto ahora mismo pasando a la esquina inferior derecha, de color blanco.
Saludos, Lordis de Puerto Rico. Estoy un profesor en Mayagüez, en el recinto de Mayagüez, para la última 21 años. Saludos.
Muchos saludos, Lourdes. Espero que estés disfrutando de esta inmersión y de la búsqueda de los seeps que hemos tenido durante el día de hoy.
Antonio, ¿te parece si hago un resumen del interés astrobiológico que tienen estos sitios?
Por supuesto.
Genial. Voy a poner yo mi granito de arena como microbiólogo, ¿vale? Entonces, buena parte de esta expedición consta de astrobiólogos. Entonces, aunque hemos estado intentando estudiar el fondo marino estos días, los ecosistemas abisales y bénticos de los fondos marinos suelen depender en muy buena forma de la caída de materia orgánica de la superficie, que es la zona productiva. Entonces, son ecosistemas muy dependientes de que haya mucha vida alrededor y de planetas o al menos zonas muy habitados, llenas de vida, y que no son tan relevantes a nivel astrológico, que son apasionantes a nivel de biología marina.
Can we get a closer look to this part, please? Is this the one that you say, Nico?
Yes. Peculiar shape. From far away looks like a...
Maybe just zooming in. Yeah.
Obviously these are tentacles, yes.
So what are we seeing, Nico?
That's a very good question. Not collectis, but I think it's a sea anemone on a protracted face. That's what it looks like. Also, the museum does not want too many anemones. Let's go. Let's
So
go.
we can keep going.
I think it's a sea anemone in a protracted face.
It's very pretty.
Comentan que es una
I'm certain.
anémona
Yeah, the way it is
retraída.
with the base.
Todavía una visión bastante curiosa y disculpa la interrupción,
Nada,
Miguel.
sigo.
Puedes
No
continuar
hay que disculpar
contándonos
nada, la anémona estaba ahí, no es su culpa.
el interés astrobiológico de estas zonas.
Sí. Entonces, las fuentes de metano, que es lo que estamos estudiando aquí, son surgimientos de metano que en general puede haber sido generado de forma totalmente geológica y sin interferencia biológica, aunque también puede tener fuentes biológicas, ya que muchos microorganismos son capaces de generar metano. Este metano supone tanto la fuente de energía como la fuente de carbono para microorganismos, lo que les permite estar totalmente separados de los flujos biológicos sin necesidad de ningún aporte externo de energía. Y estos microorganismos pueden ser consumidos por organismos más complejos hasta poder generar ecosistemas que están enteramente separados de lo que sería el aporte de materia orgánica de la superficie marina. ¿Esto por qué es interesante? Pues es extremadamente interesante porque son ecosistemas que podrían existir en otros planetas, en los océanos helados de lunas como Encelado y Europa, etcétera, y que requieren muy poco sustento para ser funcionales. Contener una fuente de metano y nada más, no hace falta luz, no hace falta nada, y agua son condiciones necesarias y suficientes para la presencia de microorganismos y la generación de vida. Esto hace que estas fuentes sean extremadamente interesantes a nivel astrobiológico, ya que, pues como ya decía, son extremadamente pocas condiciones para la vida, mientras que todo el resto del fondo marino requiere muchísimas más condiciones. Y en estas fuentes de metano se podría generar vida, no con relativa facilidad, pero sí de forma factible en otros planetas. Mientras que todos los organismos bénticos que llevamos viendo todo este tiempo requieren de ecosistemas muy desarrollados, órdenes de magnitud más desarrollados que lo que supondría simplemente una fuente de metano, que es eso y a vivir.
Hola, Lordis. La Universidad de Puerto Rico en Mayagüez tiene un departamento muy fuerte en ciencias marinas. Estamos en Isla Mayagüez, en Barriera, y si puede, si quiere, puede pasar con su familia para hablar con profesores y tu hija puede hablar con profesores o estudiantes graduados. Estamos en Isla Magueyes, en La Barriera. 7 días por semana, ¿ok?
Y la verdad es que la biología marina es una ciencia apasionante, así que no me extraña que tu hija tenga ese sueño, porque realmente es apasionante todo lo que podemos ver en el fondo marino. So, Miguel, me estabas contando, Miguel.
Sí, pero he terminado ya.
Ah, bueno, vale.
Sí, mi discurso ya.
Do you want to say that, or at least part of it on English?
I already went over it in English a while ago, but I can do it again. I was saying that the methane seeps are particularly relevant astrobiologically because methane itself can sustain life. It can provide both carbon and energy to microorganisms, which means that just supplying methane and being in an environment with water and the necessary elements for life is enough to sustain not amazing but well-developed ecosystems. And the conditions necessary for this are much lighter than those that might be required to have the benthic ecosystems that we have been seeing all this time, which require organic matter from the productive zone of the ocean. And it generally requires a much more complex and developed ecosystem, which we may not expect in other planets as just a methane seep.
Well, if you guys are offering English, Spanish, Hindu, I'm gonna go on a limb here and offer Greek, if we have any Greek or Cypriots
Sure.
on the audience.
Please do.
But I need to have an audience that is, Greek or Cypriots, so I could go talk to them. I gotta,
Some people may see this streaming later, so if you want to at least provide a little bit of context, or maybe...
I'm just gonna say hello. If I have a confirmation, I don't feel like, but thank you. Anyway, καλησπέρα σε όλα τα παιδιά. Άμα θέλετε να μιλήσω ελληνικά, να μου πείτε. Okay, that's it for now, Antonio. Just, I really just said hello. If you want me to speak in Greek, please confirm. I can say a few things.
Mm-hmm.
That's what I just said.
What I like about Greek is it sounds very similar to Spanish, but the way that you pronounce things is very similar to my.
What you see is what you get.
Yeah, that's it.
The phonetics of Spanish and Greek are almost
identical.
Identical.
Lo bueno estamos hablando con Nick, que él es griego, que realmente una particularidad de su idioma es que fonéticamente se parece muchísimo al español, de la forma en la que pronunciamos, tal cual leemos, ¿verdad?
Sí, así es. También tiene la consecuencia de que oír hablar a los griegos de fondo te hace pensar que se te ha olvidado cómo se habla español y no entiendes bien.
Como si no tuvieran acento, ¿verdad?
Pero luego lingüísticamente son idiomas español y griego bastante alejados. No muy alejados, pero bastante alejados.
Sí, el español está muy influenciado también por el griego, obviamente.
Como todos.
Hemos pasado hace poco uno de esos pulpos pequeños que vimos anteriormente. We just passed a little octopus that we just see a few hours ago. Very different from what we've seen in the last dives.
The fish was the common name. What people are using, rat-tailed fish, a very common fish in these depths. Yes, thank you, Lisa. Yep.
Mm-hmm.
That's exactly right. Rat-tailed fish.
Ratai. O sea, ese pulpito tiene el nombre de ratai. Realmente son así, no es que sea un ejemplar joven, ¿no? Simplemente que tienen ese tamaño reducido.
Lisa, if you keep going like that, the next half an hour is yours, okay? So, and thank you so much again.
De nuevo en esta zona encontramos una gran cantidad y variedad de pepinos de mar, so lot of sea cucumbers and other kind of organisms that we're used to find in these sandy plains. Maybe not sandy, but you know, these sediment plains. And we are getting closer and closer to the first place that we just found.
The red on by the hill. It's a hermit crab with a sea anemone on it.
Where?
To the right. I'm sorry. To the right. I'm sorry.
Where? Can you tell me?
Down and go towards the right.
Over here?
You will see it coming up next to a little hill. If not, we gotta go a little backward.
Here? No, it's a shrimp.
Now it's to the left. I'm sorry. If you back up a little bit, you will see it. Yeah.
They want to go to a little bit to the
Exactly.
left.
Back where we came.
Yes.
Yeah, I think we just passed. It's
That was the hill, so it moved out.
It was a crab?
Yeah, it was a hermit crab with sea anemones. It's behind us. We can.
So maybe we can find others later, I think.
Yeah.
Did you see it?
No, it moved downhill.
Okay.
Let's go.
Yeah, let's keep moving.
Yes, please.
Onward.
Onward, yeah.
The movie.
Again, this dive started as exploratory. We were looking for these methane seeps, but in the moment, there it is.
Oh, there he is.
There it is. I mean, now the water is not clean, but we can maybe get a closer look. We don't need to stop by, right? Yes.
We just need to zoom in on him for a second.
Maybe zoom in just a little bit, yeah.
Oh, wow.
Wow.
That's really cool.
Absolutely beautiful.
So we've seen yet few of those specimens that have an anemone on them. Right? They are, I don't know the name in English, but cangrejos ermitaños, right?
Hermit crabs.
Mm-hmm.
That,
but in this case, they are quite explicit, right?
yes.
Yes. So just try, you can maybe try to go through this rocky outlook, right? So as I was saying, this started as an exploratory dive, but as soon as we found this active methane seep, that is our priority. So we went a little bit around to check if there were others, and as we didn't find anything as active as that first spot yet, we're going back to this first point. En lo que empezó siendo una travesía exploratoria, en el momento en que encontramos esa emanación de metano, se ha centrado toda nuestra atención, con lo cual una vez fuimos capaces de identificar que al menos a lo largo de esta fractura que creemos que estaba asociada no hemos encontrado otras emanaciones activas, estamos procediendo a volver a ese punto. That's an interesting point because we are not sure, but it seems like they fit so well that they may be growing actually on the shell. Yeah, when the crab already have habited, right, Nick?
Yeah, I mean, this is nature, so they will grow until a point until there's a predation event or. Oh, hold on a minute. No, there's another line of thought on this. As you know, arthropods go through ecdysis because this is the only way for them to grow. So during ecdysis, I'm not so sure what is happening with the anemone. Ecdysis is the shedding of the exoskeleton. So, the crab and all other crustaceans, all arthropods, can grow. So I'm not so sure what's happening when the crab is losing its exoskeleton. There has to be a way that the sea anemone stays on it. The abdomen is soft, so it doesn't set. That's another thing. The abdomen of hermit-crabs is soft. It's the carapace, the legs, the head that has the exoskeleton. So perhaps the setting of the exoskeleton does not influence the sea anemone much.
So Miguel, can you? I don't know if you were listening.
Mm-hmm.
Can you explain for the Spanish speakers what just Nick told us? ¿Puedes explicar para aquellos que hablan español lo que Nick nos acaba de contar?
Lo del cangrejo, ¿no?
Sí.
Vale. La cuestión aquí es que en estas zonas se ha visto una relación simbiótica asociada, o comensal al menos, entre cangrejos como este que acabamos de ver y anémonas que crecen en su caparazón. Lo que se está preguntando Nick, es por qué, cómo se mantenía la anémona en el caparazón del cangrejo a lo largo de las distintas mudas que va haciendo. Son cangrejos que hacen mudas para ir creciendo, que como lo hacía, porque obviamente el exoesqueleto queda atrás y es a lo que se supone que está adherido a la anémona. Y Nick no lo tiene muy claro cómo es este proceso, pero entiende que tiene que tener alguna forma la anémona de mantenerse en el cuerpo del cangrejo. Otra opción es que se adhiera al cangrejo ya cuando tiene periodos de muda largo y aguante lo que aguante, pero es difícil de saber.
So thank you so much, Miguel, for explaining that for our Spanish speakers on the audience. Muchas gracias, Miguel, por la explicación.
Gracias. No de nada.
Gracias por darme las gracias. And yet we keep going through the coast of Chile, finding probably new species, but more importantly also being able to detect species that we didn't know they were there. Seguimos aquí buscando por quizá encontrando alguna especie nueva, desconocida, pero sobre todo también lo que es muy importante, descubriendo cómo la distribución de algunas especies que ya conocíamos alcanza esta zona sin que aún no se conociera.
Y ya no queda nada para llegar al Seep original.
Exactamente,
en 5 o 10 minutos estaremos allí.
Estamos viendo la topografía como estamos a punto de alcanzar
menos de 100 metros.
Ese primer punto en el que encontramos, de hecho ya se empiezan a ver a lo lejos esos primeros biofilms. So we are about to arrive to the first most active methane seep, and we are starting to see those white microbial mats. Yeah, some kind of biofilm, yes. But we should keep going, right?
Yeah.
Okay. So you can see some sea urchins.
Let's keep going.
I think that on the left is another crab, more cucumbers, but somewhere close we will start seeing those bivalves, those large, white, and living bivalves that are indicative of the methane in the coal seeps.
Well, I thought it was something.
This similar to the one that we just saw, right, Nick? But like laying down.
Yes, exactly. Wow, laying down, yeah, amazing.
Seguimos hasta encontrar esas bivalvos blancos que eran indicativos de actividad de metano y acabamos de pasar esa anémona, no recuerdo, que está como retraída, ¿no? Que vimos anteriormente, pero en este caso estaba como tirada en el suelo, tumbada. We can see here some interesting outcrops, but yet nothing really exciting. And it's interesting to see how being around here it doesn't look really different from what we already seen before, but yet we are about to arrive to that very interesting spot where methane is emanating from below the surface due to the tectonics in the area. The gas escapes through the little cracks that is made because of the compression and distension of the materials, and it changed everything around the new life, new microorganisms growing, and also even the geology changes. A pesar de que el paisaje no es muy distinto de lo que hemos estado viendo en las últimas horas, incluso en los últimos días, sabemos que estamos a punto de acercarnos a ese punto de actividad hidrotermal, de esa emanación de metano, en la que no solamente encontramos especies endémicas, sino también biología muy específica de esas zonas. Ya empezamos a ver ahí a lo lejos esas formaciones blancas. We are seeing these white bivalves from afar. I'm not sure if this is right, the ones that are alive, or I think they are. So our IP, principal investigator is giving some hints on how we should proceed, because what we want to do is to take some samples around the area.
This isn't the
I think
first
this
site
is
with
not the first one, right?
Exactly, that's what I think.
They don't look as alive as we.
This is the first site.
We may need to go closer.
Okay.
We can see clearly some empty cells close to us, but maybe in that hotspot in the middle we find some live specimens.
I believe we saw a source on the one further away.
You can actually see some of the let's say footprints of the SuBastian, those round marks that the core left when we land. Okay. So we are going to try to park to the left of this area, facing to those cells.
the people that are asking for the jelly, I just see two sea cucumbers right between the bivalves. I don't know if that's you're talking about. I didn't see a jellyfish. And that rat-tailed was very chubby there. I would like to see it. If I hope it's going to turn around. He looked a little peculiar.
So as this is a quite important place for sampling, both of our principal investigators are in the room, providing instructions to the pilots on how we will proceed. Hopefully the different teams that are working on this expedition will be able to take very exciting samples, both for studying the biological life, the marine biology I mean, also microbiology and the geology of the site. Actualmente, dado que hemos encontrado este punto de gran interés para nuestro proyecto, los investigadores principales se encuentran en la sala dando instrucciones sobre cómo vamos a proceder a tomar muestras de este área tan limitada en la que encontramos zonas interesantes tanto desde el punto de vista de la biología marina como de la microbiología y también de la geología. Sí, perdona, Nick.
Yes, there's a question if there are any Chilean scientists on board.
I was going to answer that, yeah.
And yes, of course, we have a very good representation. Dr. Guillermo Guzmán. Introducción, sí, por favor.
Acerca de estos moluscos, son animales que están obviamente muy bien adaptados a este tipo de ambientes. Ellos tienen una simbiosis con las bacterias para poder conseguir su alimento. Entonces, su sistema digestivo está muy reducido y estos animales en sus branquias albergan una gran cantidad de estas bacterias. Este grupo fue descubierto la primera vez en Concepción, hace 20 años, o sea, 20 años después estamos encontrando acá el mismo tipo de organismos en Antofagasta, a una distancia de, calculo, más de 2.000 kilómetros de diferencia.
So thank you so much, Guillermo. Muchas gracias, Guillermo, por...
Si puede introducir, porque ellos preguntarte, ¿hay chilenos científicos en board? Sí, okay, yeah. The genus name for the bivalves is Calyptogyena.
So I think first we are going to...
Calypto for the ancient... Oh, I'm sorry.
Later we are going to take a few courses with different octopuses, so I'm pretty sure of space. And I think afterwards we are going to collect some experiments for the animales logran dispersarse también en cáncer de order.
Water. Not all of the bottles, maybe just one, two.
So, as Guillermo Guzmán, that is one of the Chilean scientists that is on board, and he's specialized in the study of
clams. Okay.
Of the life of the.
Okay.
So what's going to be the order for sampling? Water, course, and then clams?
Sí, todo, cores, 4 especímenes de esto,
As ella said, we are going to sample as much as we can from this very limited and special place. and we are going to start taking water samples because we are actually close to probably methane sap. There is methane probably mixed in this water, so we will to try to take some of this water to understand better what's happening in these fluids.
Estamos aquí para 1 hora.
I think we can take at least one bottle because I don't see the mask.
And course.
Yeah, maybe.
It's cool. Bien, okay. Yeah.
So as I was saying, we are going to start sampling the water as these methane fluids mix with the seawater. We may be able to take some samples of water that contains probably not methane, but maybe some small particles that forms when they precipitate. The methane precipitate in contact with the seawater, forming carbonate. So we will try to filter this water and to see if we can find something in there. And then okay, we are going to collect some of these bivalvi that Guillermo Guzman just told us about how they are adapted their digestive system to feed on methane.
Yeah, actually.
And they were actually already found in Chile 20 years ago, but it's the first time that they are described this far, I think this deep, in the coast of Antofagasta.
Who wants it? How many people were course?
Geology.
Oh, okay, everything.
Five.
I don't know if there's enough film.
5?
6.
6?
So,
I don't think that's no.
I will take one and divide it with other 3.
We need one.
For yourself only. I will divide it with 3 others.
We've wanted our own core this whole time, so I don't know.
Okay.
But I don't
No, I
want
will
to get rid of
suggest
the
to
entire
divide.
map.
No, I'm not
That's
taking
my
the
concern.
entire list. So I will divide it with 3 others.
So in the room we are still discussing about how are we going to sample all these different materials and specimens. En la sala estamos debatiendo cómo proceder para muestrear este lugar de interés, tanto sus materiales como especímenes.
I can share it. As long as I get the files, I can share, of course.
We will try to take as less material and specimens as possible. We are going to try to share as much as we can, but again we need to be sure that we take enough so we can perform all of analysis and to achieve to identify both the organisms in here and also the materials, the different structures that we can find, so we can characterize this specific feature, as this is a very unique opportunity.
We should share. There are many here, 20. We're all sharing. So we have the fourth one.
I just see this, one of the bivalves just spit some sand to the right.
He's going to look for animals, right?
don't need to take a call here as long as I take
We will wait.
the other side.
Okay, thank you.
Maybe we can take a closer look to this rock to see what kind of organisms are there. Yeah.
Yeah, it's just a very small area. In geology management, taking something a little bit away from the actual
Animals?
On core is also for Daniel. And we take the core before. We take the core before in fossil methane to compare with the data with active methane seep.
Right.
How much? The wall, sí.
The wall.
Tanto.
Yeah.
¿Y para ti nomás?
For me and Daniel.
Ah, okay. Four. Okay, that's manageable. We can do 2 and 2.
We've been waiting for this for days. That's why we're trying to make sure that everybody gets what they asked for in the original proposal.
Yeah, we were lucky to find it. So
we've been working on this for more than a year.
That's
some
it.
more than others.
Yeah, yeah.
So it's
a complex matter.
This is our chance to study the system.
Okay. 4.
You can see there how the organisms, these bivalvi, are up close.
Each group gets individual copies.
Yeah, and you do.
That's we've been going back and forth.
They are filtering. They filter right through that protuberance, right, Nick?
We need the whole thing. We need the whole thing for the
I think Nick is also busy.
4 cores. Primero y luego 4 cores.
Again, we need to figure out how to proceed.
What that is the other one?
Number doesn't matter.
Soy de Concepción, pasaron alguna vez por esta costa de Chile.
And we are getting ready to take some course of material. The SuBastian is moving its storage in parts.
Here, show us the area.
And the pilots will start.
Here? Here? Oh no, not mine. 4. Okay, ready to see. Here? 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 I'm asking here? Okay.
So yes, the thing is probably these seeps are about to end soon or later. This is most likely dying area, so we will try again to disturb as less as we can. But
we have plenty of storage for specimens and sediment cores. The issue is priorities, time, and
also the balance between taking samples.
We try to disturb the environment here as little as possible.
That's it. It's a compromise. Estamos comentando como realmente a pesar de tener seguramente suficiente espacio para almacenar las muestras, también es una cuestión de prioridad entre los diferentes grupos y de intentar balancear la toma de muestras con perturbar el ambiente lo menos posible, a pesar de que esto no deja de ser una zona que según todos los indicios tenderá a perder su actividad más pronto que tarde.
So Nick, this cucumber, do you think is also part of this specific area, or it's just in here by chance? I mean, he's feeding on some specific...
The species? No, I don't know the species. It's a sea cucumber, but I don't know the species. No.
I think.
You don't think?
Because it's associated with this.
Danielle's asking if he can have a clam for him as well.
Yo creo. Bueno, sí, sí. Vamos. Pero el Seep está cerrado, ¿no? En el fin. Pero es importante, ¿no? Sí. Sí, pero cerca de esta área hay fauna normal, ascidians, lo grande ascidian, ¿tú sabes? Sí.
So we are a little bit focused, trying to sample of these very unique components. We are starting, we just took some water samples from the Niskin bottles, and now we are taking the first core of many. We can see how the core is quite complete. The pilots are doing a great work, going very slow so the sediment doesn't go off the core. You can clearly see how fine the sediment is, how it's moving all around the core. I think the fact that we are streaming this campaign is super important to make everyone aware of how science is done and they are, all of you can also be part of it. I mean, they are all discoveries that everyone needs to be aware of. So thank you for being here and to share for sharing this moment with us.
I just deleted here because I want to be in the middle. You want to be in the middle between me and Miguel and me? In the very middle?
Oh, we don't need this in the mount.
I want to be the one in the very middle.
No, as a team request anyway. Bye now. So we took the first core, number 2, and we are going to keep going. It was just secure.
There's no aquarium on board.
I don't think so. I didn't see any. But who knows? It may be in the kitchen.
But we'll take one of those sea cucumbers for the museum.
Yeah, we have 2 of them, right?
Yeah.
Maybe, I don't know what is streaming, but in the science cam we can see right in the front one of them, and the engineering cam, that is the one that provides us a top view. We have another one closer to SuBastian. Alguien en el chat preguntaba si teníamos un acuario a bordo. No creo que sea el caso, no, al menos ni yo ni Nick lo hemos visto.
La cuestión esencial es que prácticamente todos estos organismos que vienen de profundidades tan altas están adaptados a presiones enormes. Entonces, para el momento en que llegan arriba están muertos. Así que no hay necesidad de apoyo.
Sí, la manera en la que tenemos de almacenar estas muestras biológicas es básicamente almacenándolas en alcohol para evitar que sufran un proceso de putrefacción. So we're about to take the next core. If I'm not mistaken, it's the number 6.
Yes, number 6. Okay.
Vamos a tomar el siguiente core, el core número 6.
There to be much difference from like the center to around where the seepage is. I wouldn't go much further than the last clam.
I do think that what we do here inspires people and even kids. Mostly kids, I mean. This inspires people and especially kids. We just had this morning, or I don't know if it was the morning or already afternoon, one of the outreach activity that the Schmidt Institute promotes, while a school of 8 years kids were able to take a tour around, a virtual tour of course, around the boat, the Seep, I mean, the Falcor 2, and they were able to see what's the work, how the work is done here and how we are working.
No sé.
Someone in the chat is asking how many cameras have the submarine. What we have is a few cameras. The most important ones are the science camera and the engineering camera, that are the ones that are facing to the front. One right ahead and another from the top. But we also have another cameras that allow us to check on the different components and how they behave and how things are stored. So I don't know exactly how many, but in all the cameras I can see that they have lots of them. I don't want to bother to the pilots that are now collecting the samples, but maybe I will ask them. So please, if I forget, please remind me to ask.
No, I don't know what they call the secret. I guess that's another name, but we always call it geological.
Alguien en el chat estaba preguntando cuántas cámaras tenía el vehículo SuBastian, y las principales son dos, que son cámaras 4K, en las que podemos ver con alto detalle tanto de frente como desde un poco más arriba, justo enfrente del vehículo, pero contamos con muchas otras cámaras que enfocan a diferentes partes de este complejo instrumento de ingeniería y que nos permite ver pues cómo se están tomando las diferentes muestras, también en parte cómo se aterriza sobre el terreno, cómo están algunas de las fijaciones de los elementos. Todo eso al menos es lo que estoy viendo aquí desde la gran cantidad de pantallas que tenemos desde la habitación de control. Más tarde, no quiero molestar ahora a los pilotos, pero más tarde preguntaré para contaros exactamente cuál es el número de cámaras y sus características. So someone in the chat just said that the ROV has support for 50 channels of video, so maybe at least we may have as much cameras on board. Desde el chat también preguntan cuántos ingenieros están a cargo del vehículo. De hecho, hay una gran cantidad de miembros de la tripulación que se encargan tanto del pilotar el vehículo como de su mantenimiento. Una vez alcanza la superficie, es necesario tanto extraer las muestras como hacer el mantenimiento del vehículo, limpiarlo, endulzarlo, quitarle toda esa agua marina para evitar corrosión y volver a poner todo en orden para tomar las muestras y hacer la siguiente navegación. No estoy seguro de cuánta gente exactamente se encarga, pero la suficiente como para que podamos hacer estos turnos y no haya una sobrecarga de trabajo, sino que cada par de horas hay un relevo y al menos aquí en la sala de control, y también obviamente hay unos horarios establecidos para el resto de trabajos. Y no solamente
Aquí, más o menos, hay otro especies mismo.
No solamente estamos hablando de este mantenimiento, sino que también está toda la cuestión de la planificación, del manejo de todos los instrumentos y tecnología que contiene el Falcor 2, el Falcor 2 y por supuesto el SuBastian. Así que hay una gran plantilla de ingenieros que están a cargo de todo esto, con una dilatada experiencia y que son capaces de tomar muestras como la que estamos viendo ahora mismo, este Puscor de terreno.
Just a reminder for the audience, we have been in the water for 15 hours, starting 8 o'clock this morning, and we're going to finish at 3 or 4 o'clock in the morning. So we'll do a 20 hours to 22 hours worth of observing, taking photos, taking specimens, taking benthic. So we still have a way to go.
Good night for those who are going to sleep now in the audience. And as como estaba comentando Nick, hoy llevamos ya bastantes horas. En total van a ser 20 horas de navegación. Al igual que los ingenieros hemos estado haciendo turnos, pero algunos de nosotros también hemos estado en varios de ellos de espectadores. Y aún nos queda bastante tiempo, ¿verdad, Nick? Hasta el final de esta inmersión, esperando acabar a las 4 de la mañana, hora local. Alguien en la audiencia nos pregunta que cuál es la presión a esta profundidad. Permitidme que lo busque porque no sé exactamente dónde se encuentra esa información. Puedo confirmaros que nos encontramos ahora mismo a 2 kilómetros con 834 metros de profundidad. Para aquellos, for those who are going to stay with us for all this long dive, thank you for staying there. we'll try to keep it entertaining. Thank you for sharing with us these exciting moments. So I just check, and the pressure is about la presión. The pressure is about 2,080 dBars. I don't know in PSI. So la presión es unos 2.880 de bares. Así que nos encontramos efectivamente en unas condiciones de presión bastante grandes, y nos encontramos, como comentaba hace unos minutos, en las costas de Chile, cerca de Antofagasta, en uno de los sistemas tectónicos que se encuentran al sur de la península de Mejillones, en una zona que geológicamente es interesante por encontrarse en esta zona de subducción entre la placa de Nazca oceánica, que baja por debajo de la placa continental sudamericana, y gracias a esta tectónica, a esta actividad geológica, es por lo que podemos encontrar zonas de interés como estos emanaciones de metano que estamos muestreando ahora mismo. Aquí veis que estamos tomando otro core, en este caso con el fin de estudiar esos tapetes microbianos que acabamos de perforar y cómo es la relación entre todo eso. Estos microorganismos y estos organismos se encuentran adaptados, obviamente, tanto a estas altas cantidades de presión como para, en este punto concreto, alimentarse del metano, pero también resistir la ausencia de luz y unas condiciones relativamente frías. Estamos a 1,8 grados Celsius. So these organisms are not only adapted to these harsh conditions. Here we are in 1.8 Celsius degrees, but also with almost completely lack of light. But in here they are also adapted to live feeding on this methane that is leaking through the crust. So this course seems to going very smoothly. That's the third core on this side, and just you can see there how the material looks like in this transparent tube. Acabamos de tomar el tercer Fuscor de la noche y podéis observar el material que se está deslizando poco a poco. Oh no, this is sliding down, so we lost it.
So I still see the bar from the top. So
No.
Okay. Can we repush? I'm sorry.
Yes, the way that the dive went today was quite.
No has gafado del todo.
Ya lo sé. I jinx it. I don't know how you say that in English.
That's a good translation.
Sorry. So I was saying that the dive today went a little bit slow at the beginning. We were just going through these transects along the different fault systems. We were few hours of traveling relatively fast, and now this happened, and we all went very excited and wide awake. So yes, now is not maybe as exciting, but very. It's time to focus on getting these samples, and maybe we'll be able to take a good core, including this microbiological mat over there. This whitish, I would say, mucus that it forms the colonia of bacteria. Estaba comentando como al principio de esta inmersión fue bastante tranquila. Fuimos haciendo un trayecto a lo largo de estos sistemas de fallas y sin embargo una vez que descubrimos esta emanación de metano, esa tranquilidad se convirtió en me sale excitement, pero pues eso, nos despertamos todos y ahora estamos en una parte que requiere cierta concentración, ya que vamos a intentar muestrear de nuevo ese tapete microbiano blanquecino, que es una especie de mucosa que no deja de ser una colonia bacteriana, y veamos si esta vez tenemos más suerte y no se escapa como el último. So let's see how it goes if the next one goes well. I wouldn't say anything just in case I jinx it again.
Respecto a la pregunta que han hecho de cuál es la presión PSI, estamos a unos 2.800 metros, lo que equivaldría a unas 284 atmósferas de
Muchas
presión,
gracias.
que en PSI, que son pulgadas por... No, es presión por pulgada al cuadrado. He hecho la transformación porque no usamos eso por acá. Serían unas 4.200 PSI.
4.200 PSI. ¿Cuántas atmósferas habías dicho que eran, Miguel?
Pues es más o menos a una atmósfera por 10 metros, ¿no? Pues unas 280 y pico.
Estamos hablando de 280 y pico veces la presión que tenemos entonces en superficie. Imaginaos las presiones de las que estamos hablando. So we were just talking about the pressure at these deeps and how they are about 200, almost 300 times that what we can find on the surface. So some of the cells are broken, as you can see. We don't know yet how fragile they are, but let's see how we sample them. It may be a good idea to take an empty one before taking a
Guillermo is raising a very good point. The closer it gets to the bivalves, the more loose it gets, because the bivalves really change the profile of the sediment. Consider moving a little out, but that's your core.
Nick estaba explicando cómo cuanto más nos acercamos a los bivalvos, más suelto está el terreno, porque se encargan de perturbar esos sedimentos bajo ellos, así que es por ello que estamos teniendo estas dificultades a la hora de muestrear este tapete microbiano.
Blow it or blow that? Straight down.
So we are going to try again. Testing and probing this microbial mat through the buscar. Vamos a intentar muestrear de nuevo este tapete microbiano.
Es prácticamente idéntico a yo también me he compartido un poquito con la gente. Estamos ahora peleándonos con esto. Es que vete así. No está pasando por aquí. Todo parece ya. Una un poquito más a ver si te funciona. Con esto. Pues sí, yo también lo tengo bien con esto. Cuando termine con esto,
Bueno, Miguel, creo que solo nos queda hablar de qué hemos comido hoy o algo parecido.
¿Qué hemos comido hoy? Hoy había.
No me acuerdo.
Había unos macarrones.
Ah, es verdad.
Extraordinarios.
Yes. We have mac and cheese for lunch. Yeah.
Vamos a hacer un shout out a Greg, el cocinero, que hace unas salsas para pasta magníficas.
Yeah,
y
Greg.
tiene, yo no sé si tiene un libro de recetas mágico o algo, pero tiene una variedad de cosas que es extraordinaria.
Greg is a great chef, and he also has some amazing chefs working with him.
Sí, nos ha hecho pizza y sospechamos que la masa era artesanal.
Sí. We had some homemade pizza last night, right?
Yeah, we had pizza two nights ago, and then again last night because there was a slight malfunction.
The good thing is they also cook with leftovers, I think, and that's some very some creative cooking. I think is.
Yeah, it's pretty amazing, Antonio, that you've chosen to talk about this as one of the most tense moments of the expedition.
I know. But I want, to talk about something, and I cannot think another thing that food in this moment. I don't know why. Maybe because all those Bibles.
We want the biofilm, so I just destroyed it.
Can't reach for that.
Yes, that's
So
what
we only have so much reach.
Okay.
On the left side, so that's why we're focus on. If you want to do that, I'm going to have to do a different flow.
Tampoco hemos tenido suerte con esa último core, así que vamos a intentar muestrear otro core distinto. So we are moving on to the next core, I think. We may try to move a little bit.
That's all right. We can change arms. It's no problem.
There's
Yeah,
lots more
yeah.
biofilm now, so
Yeah.
so you can secure one later. I'm not interested in the specifics of the
Oscar, one of the
oil.
oilies on board, is giving some instruction on how we could proceed to taking a geological sample from this vent. And that's really exciting because when we analyze the data, we will be able to see how active is this methane scene was, and maybe even to analyze for how long it has been
Equal in the middle,
active.
so that way we have something from this site. There's, well, I thought I was trying this out. I don't know, it's working really.
Yeah, I think probably this cucumber is running from us.
Did you think that's too close?
They say all the movement, they are running for their life.
They're pulling up on the clams, I think.
Probably they are afraid to be taken.
He says the other side's okay.
So Miguel, if you feel like it, maybe we can talk a little bit on how we process the samples afterwards.
Okay, so it really depends on exactly what you're going to do with them and on which team you are. For instance, I'm going to take my samples through a set of processing techniques that no one else is going to, because I'm specifically looking for messenger RNA. Well, so first I'm going to have to do an RNA extraction, which is a specific process that can get quite complicated, and that I have to go through to get my transcriptomes. Other people, like for instance Lauren and Armando and other people, they will need to extract DNA, which is somewhat easier but not very simple. And geologists will have to do their own thing. People looking for metabolites, like Katie right next to me, they will have to do their phase extractions. And
yeah.
So it really depends on...
The biologists need to put the organisms in alcohol, for example, and to say, how do you say it?
I don't know what you mean.
A sediment, you need to put it through a stove?
A
sieve, that's the word, sorry. A sieve, so you can see the different organisms between the sediments, yeah. Alguien en el chat está preguntando que este metano de dónde proviene, y es debido de nuevo a que nos encontramos en esta zona de subducción, donde se produce el movimiento de una de las placas oceánicas bajo la placa continental sudamericana, de la placa de Nazca, como se denomina la placa oceánica que pasa por debajo. Y esto hace que haya unas compresiones tectónicas que fracturan todo el terreno y que parte de ese metamorfismo y toda esa actividad tectónica haya gases provenientes en parte del manto que llegan hasta la superficie, y es lo que acabamos viendo aquí. Es a través de esos sistemas de fisuras que se escapa ese gas hasta la superficie, en este caso hasta el fondo del mar.
Este sería el caso del metano generado abióticamente, es decir, por procesos puramente geológicos, que es en realidad el más interesante a nivel astrobiológico, pero en la Tierra también hay cantidades ingentes de metano generados por lo que se llaman las bacterias metanógenas, en comparación a las metanótrofas, que son las que consumen metano. Las bacterias metanógenas, mientras que las bacterias metanótrofas, las que se alimentan de metano, que son las que tendremos aquí, necesitan oxígeno para poder consumir ese metano. Las bacterias metanógenas no son capaces de vivir con oxígeno, entonces generan metano como producto de desecho en su propio metabolismo. Y ahí se generan dinámicas muy interesantes en sedimentos que no es en este caso, porque este caso tiene un aporte brutal de metano, pero en sedimentos que no tengan un aporte de metano claro, o incluso en láminas de agua en las que se genera en la capa inferior del agua, donde se está degradando. Esto ocurre, por ejemplo, en el caso de estanques en la capa inferior, donde puede quedar mucha materia orgánica de hojas y plantas atrapada. Se va a dar por medio de la acción de las bacterias metanógenas, y por procesos anteriores esa capa va a quedar sin oxígeno. Entonces el metabolismo de bacterias metanógenas lo que va a hacer es la materia orgánica degradarla y obtener metano como producto de desecho. Este metano luego va a subir por la columna de agua y va a permitir a bacterias metanótrofas encima vivir, generando un ciclo bastante divertido. Did we get the geological? Right?
Are we going to proceed with biological samples? I don't know the...
We're going to do it as soon as we get your score.
We're not taking a call from him.
What?
Our core's gonna come from the other spot.
Lauren, what's the course of action?
Some clams, yes.
That's the course of action.
We're done with course.
Okay.
All right, how many people wanted a clam?
Last time I was, I heard 4.
Disculpad que estaba discutiendo con mi compañero cómo tomar el resto de muestras, ya que se va a ir a dormir, ya que le toca en unas pocas horas procesar todas las muestras que tomemos. Efectivamente es relativamente antiguo este proceso, pero sigue estando activo en la actualidad, así que no sabemos por cuánto tiempo ha estado activo,
and I would suggest
pero sí
to
sabemos
give it a
que
try
se
with the
va.
arm. Straight up. You have guarantee you, if you get them, you...
3 clams from one side and 3 from the other, please.
Okay. All goes to the same container or to
They can all go in the same box.
Absolutely, that's a good idea.
And just cells, right?
No, not just cells, no.
Oh, the full thing?
Animals.
Okay, so
Yes.
you just take the specimen from one side and the specimen from the other side. They can go to the same biobox. I don't know which ones are.
Which box?
Which bioboxes are for 6 bioboxes?
I would recommend, taking into account that we're going to get 6 clams, I would use BB2B for all of them. It should be big enough.
So hello everyone joining just now. We just found this methane seep a few hours ago, and we just came back to sample it. Saludos a México desde Chile, desde las costas de Chile.
Yo creo que tenemos la oportunidad para procesar ellos, sí.
Sí, tiene que ser.
Él es duro, ¿eh? Duro. Va. Pero, ok, no con...
Today we
Bueno, el primero, dame...
are doing a really long dive. We are going to be diving for
No, está bien.
more than 20 hours. 20 hours of bottom time, but
Perfecto.
the
Eso es. Exactamente. Yeah, that's the way. Let's see if it's gonna stay.
Ah, esos los crabs también.
4 horas 12, el puerquito que está ahí, pero dicen 3 y 3, ¿no? Supongo lo que has dicho.
Vale, pero
Break
Creo que
easily.
entendí que dijeron ruido, confirmo. Confirmo. Vale. Puerquito,
A mí, idealmente necesitamos un crab para nosotros y yo creo para otro. Un crab grande.
vale. Vale, ven viendo por si se me pasa.
Todos los bivalves en el mismo lugar.
Yes. Because they need 4 for
Yeah, it's
characterization,
okay.
1 for demo, 1 for
I just want to confirm.
Yes.
No problem.
Sí, mira.
Deep?
2B is empty and I will put all the clams.
So we are sampling these Bibles.
Oh, ok, ahora entiendo por qué no. Okay.
We are going to take 3 from each side. Y efectivamente trabajar aquí es casi más complejo que trabajar en el espacio. Tengo la suerte de también haber estado trabajando en operaciones en diferentes misiones de la NASA y a diferencia de aquí, en esas misiones solo podemos planificar con suficiente tiempo, ya que no se puede trabajar en tiempo real como aquí, sino que hay que mandar todas las instrucciones debido al retardo que hay en enviar la señal que dependiendo de dónde se encuentre la Tierra Marte es de una media de 25 minutos, 24 minutos. Sin embargo, aquí tenemos la capacidad de trabajar en tiempo real, lo que, si bien hace que sea bastante más productivo, sí que lo hace más desafiante en ese aspecto.
¿Las mismas que has visto en otros lados o distintas?
No, al otro lado es el mismo. Pero el problema es que hay 2 o 3 especies de bivalves.
Some of the interesting stuff is not just the specimen itself, but also all the organisms associated to them, as someone in the chat is saying. And also, Nick in here is saying that we are not looking to just one species, but 2 of them at least.
There are at least 2 species. For example, in the main, you don't have the view, but in my view, to the left there is the second species, but
Which
it's a little
view
further
do you
to the
mean?
left.
Oh yeah, I have it in here, yeah.
Yes, if you put it there, you will see the left, you will see the cell of the second species.
What's the...
Calyptogena. Calyptogena is the genus of the...
What's the main feature that make them recognizable? I mean, why?
I'm not an expert in this group.
Okay,
at all.
okay. So how do you know that they are different species?
We have the... okay, in this particular case, I was talking to
Yes.
Guillermo, and then he was showing me species broken. And it's based, on size, for example. The one is a big size, the other is a small size. So that's one of the main characters, like really big difference in size.
Yes.
So, which is broke.
They are, even though the SuBastian claws are quite delicate, it seems that these bivalves are quite fragile, so it's very challenging to pick them up. We already successfully obtained one of those, but
Lauren, I have a suggestion. if another one breaks,
Scoop. All right.
So Nick is suggesting to scoop if it doesn't work properly this time. Como podéis haber visto, a pesar de que los brazos mecánicos del SuBastian son bastante precisos y tienen la capacidad de ejercer una presión bastante delicada sobre las muestras, estas bivalvos parecen bastante frágiles, y lo que está Nick, lo que están aquí en la sala sugiriendo es que si no somos capaces de tomar otra de estas muestras, directamente palear la parte en la que se encuentran para tomarlos de una manera igual más sensible, pero parece ser que gracias a la forma en la que se ha obtenido esta muestra, se ha respetado su integridad. So it seems that the pilot has the great idea of took those this way, so they are more they are not breaking. And another successful retrieval will work.
at the edge of the bivalves, these are small anemones. If the audience is talking about the pink chubby little things on the side of the or on the edge of the bivalves, these are sea anemones.
Okay. How old did you say they were? Or they could be?
No, they are sea anemones.
No, I mean, these bivalves, how old do you think they are?
Oh, that's a very good question.
I read, I'm not sure if that's true, but I read that they can live even decades and hundreds of years, right?
It could be, and there's a way to actually they can count the years, because the calcium carbonate cell is laid by layers.
Okay, so it's
you can
similar to the 3 logs, right?
Very similar, yes.
Estábamos comentando cuál podría ser la edad de estos bivalvos, y Nick me comenta que efectivamente, gracias a esos anillos que vemos en las conchas, se puede identificar su antigüedad, ya que se va produciendo
Antonio,
con esas capas. ¿Sí?
I do have a fact for the age of bivalves.
Mhm.
So the oldest living bivalves are actually, if I don't think I'm mistaken, in the great depths of the Atlantic,
Mhm.
about 400 years old.
Oh.
Yes.
So, lo que está comentando Nick es que los bivalvos más antiguos que se conocen en los mares del Ártico, perdón.
Atlantic, yeah.
Atlántico, sí, pueden alcanzar hasta 450 años.
Sí.
We are talking about living fossils here. Yeah. That's for sure.
Antonio, after we collect these, I need to push a cord because we're not going to stop at the next site.
Oh, okay.
Oh, we're not?
Because of the time?
I think it's because of the time.
Okay.
And I need a cord.
Sure.
I let my course go for the next site.
Yeah, me too. So maybe we will.
Can we stop at the next site? It's her call. There's multiple of us who want from the next site that we've held off from this site because we thought we were stopping at the next one. So we really need to stop at the next one.
So the way that these arms are operated is quite curious, actually. the pilots have this like a small piece of technology that resembles to the different parts of the arm, and they can move them, and the movement replicates in the arm. So it's, I don't know how to explain it better, but you know what I'm saying. It's like a small model of the arm, and I guess you need a lot of practice to do it right, because it's not easy, and you, I think, have this delay, right? You have some delay when you use it, right? It's not immediate.
Excuse me?
When you use the arm, you have some delay, right?
Very slight.
Okay.
Alright, what are we doing now? Alright, team.
Did we get 6?
We got 3 from this side. If we're gonna get any from this side, I have to move.
Okay.
Is that what you wanna do?
Yeah. Yes.
Alright.
We probably didn't took that sample because we are very limited on space, and we may need to keep only those who are unharmed, I'm guessing. But again, there are many people today in the control room trying to get what they need to understand better this methane seep that we just found. So it's been very complex to organize all this sampling here. We're still going to try to take more samples. There are like 2 groups of cells, and we just analyze and sample one of those, and we are now, I think, going to sample a little bit more in the other side.
Yes, to answer the chat, we're planning on.
No, you can keep talking. I'm just gonna let them know through the chat.
Okay, yeah,
that
the
we're
scientist.
gonna restart the stream. So
Oh,
but
it's not started?
You can continue talking about it. I'm just gonna check
Is this still streaming?
It's still streaming,
Oh.
but I'm just gonna let them know in the chat.
Okay, yeah. Yeah, the scientists are busy talking about where to sample and how they're gonna get all the clams and maybe more push cores. But yeah, we sampled 3 clams from one batch, and I think currently doing 3 from the other.
Just we want to announce that We are about to restart the stream soon.
That's my suggestion.
In YouTube.
The people I know that did not take is she had Katie had 3 failed ones and they won one. I said, not to
And yes, thank you, Amber. We just took, I believe, 2 specimens or 3 labels. Sorry, I lose the count.
Okay, so he's got
And now we are taking another specimens from the other batch. Let's see how it goes.
No, they told me, well, let's take the next site. Okay. And I said, there's no next site. Because we're limiting, we're getting limited time. Well, I gotta make sure I take my course. So it's not a big deal, you know. If that's what I did not hear it.
Again, this